less settled down. Stragglers come, they can. So the very first thing that we're going to do this morning, oh yes, thank you. The very first thing that we're going to do this morning is we are going to do an exercise. This is sort of a continuation of the talk that I gave last night. And also maybe a few more topics in filtering and analyzing, what data in the VCF are we actually going to use to make these decisions. And we'll be going in a little bit more depth on what the annotations actually mean than we did in the VCF eval talk. However, the very first thing I want everybody to do is go find the workspace that they were using yesterday. There's a couple of ways to do this. I mean, obviously, if the tab is still open, you can probably reopen it in your browser. However, there's an easy way to do this in Terra. If you go to the main page, terra.bio, you log in, you scroll down, and you go to View Workspaces. You should, assuming I set this up correctly, which I did, you see, you probably will only have 1 or 2 workspaces here in my workspaces. I have lots because I'm a developer, but importantly at the top here, you should see a list of all of the recent workspaces that you've used. So I can just click here and go and see exactly the workspace that I opened and cloned for you guys yesterday. So you should all be able to find where we were running data yesterday. In terms of the actual analyses that we're going to run through, if you scroll down and you go to 05 GATK variant refinement, I believe this is the correct one. Yeah, this is the correct one. And you want to open it up. And we're going to use the settings that it has here to define an environment. So this tutorial is actually going to feature a little bit of just old-fashioned hard filtering of the annotations. It's sort of unavoidable. You have to have some idea of what the annotations actually mean and how you can sort of look at them in a meaningful way. I would say, after having been taught how to use VQSR yesterday, it is a little bit like you know how to use chainsaw, and I'm teaching you how to use a hand tool to do the same job. But it is important to understand, and we're going to sort of talk about like how you can use the truth data that exists and how you can sort of some topics in what filtering looks like and what you're sort of aiming for with filtering. So if anybody is having trouble finding their workspace from yesterday or opening this up and getting this environment started, please feel free to raise the red flag. You can see at the top of this notebook, we have a very pretty picture. And of course, it's still thinking. And of course, I can get impatient. How environment is ready. Yeah, excellent. Okay, here we are. So just as a reminder, in the overall best practices pipeline, we're going to be dealing with sort of steps in the filtering stage and maybe some steps in the filtering stage that you would want to run in the absence of being able to have enough data to run a large VQSR or CNN score variance training on your data set. So we're down here in filter variance after we already have a combined GVCF of our whole cohort. And you can see a pretty picture that we'll get to how to generate this. We're going to just use the same standard settings that we've been using for the notebooks up to this point. However, we are going to be using R. So there's a very powerful plotting library in R called matplotlib. Sorry, am I mixing up? Is that the Python one? ggplot2. Sorry, ggplot2. It's very nice. It makes making these it's very powerful. It lets you do very fancy things with plots. And we can actually call out to it from Python. So we're just going to run some magic commands here at the top to import rpy2, which gives us the ability to just directly call R code within the Python script. So if you want to do any sort of fancy plotting, that's great. Once again, we want to make sure that our kernel here is still set to Python 3. If it's not, restart the kernel and then click through those installation steps one more time. And since it's doing a pip install, who knows how fast the internet is going to be today. But that should solve itself in a few minutes. Cool. So again, more boilerplate, just at the top of all of these workshops, we're going to set up our bucket environment. We're going to set up our workshop environment. We're going to set up some places where we're going to be downloading output to. So we can just run through all of those steps. And finally, we can check permissions one more time. And we can download data. So we should be able to get all the way through the whole one part of this module without actually thinking, because we've done all of these steps before. If any of these fail, or you don't see results that kind of outputs that look like mine, so you don't see this list of resource files, or you aren't seeing stuff get downloaded into the bucket, let one of my instructor colleagues know so we can fix that for you and figure out how to troubleshoot your workspace. So at this point, we have a trio VCF file that we've combined. We've run genotype GVCFs. Actually, all things considered, this is the trio VCF file or a very similar trio VCF file that we had in the tutorial yesterday morning. So this is the trio with the CEU trio, father, mother, and son that we were talking about yesterday. We are going to run this tool select variants. This lets us do some nice things, like we can, for example, in this case, pull out a single VCF file that corresponds to only one sample, and we can throw away all the other samples. So we just have what are the mother's variants and genotypes. And we're going to run some extra arguments here, like exclude non-variant sites. That's just making sure that if the site wasn't variant in the mother, we aren't also emitting it into the output here. And so we can run that and give that a minute. And we can also run a grep command just to see, what does this look like? Does this look reasonable? And the reason we're just pulling out the mother is just because it's a little bit easier to run some of these filtering tools on just one sample, because we can use just her truth set as the truth set that we're comparing against. So here you can see, here's what the output looks like. You can see all of these sites. If you look here in the genotype field, all of them are, heterozygous, or presumably there's some homozygous sites in here as well, but we don't see any no calls, which is good. And you can see we only have one sample here, the NA12878 sample. So we have now subsetted down to just the mother using select variants. Neat. But now we actually want to start to look at how we would compare this to the real truth call set. So for this, we're going to be using the genomes in a bottle call set. I think John talked about it a bit yesterday, but this is sort of the gold standard for human truth data, for at least certainly the samples that they have. It is a consensus call set. So it involves taking the same sample or cell lines of human samples and sequencing them many, many times across many different sequencing platforms and many different tools, and sort of building a consensus call set of, okay, what are all the sites that most of the tools sort of agree on? And it records where everything was where there was at least one tool that had some level of confidence. We're actually going to be using a much older version that hasn't included some of the newer data modalities that the new genomes in a bottle has, like PacBio and ONT reads, but the principle here should be good. So what we want to do, and you can see here, we have this resource. We're going to run the tool variant annotator. So that's going to take our variant file. It's going to spit out a new VCF where we've poked in extra annotations that correspond to whatever new annotations we want to add. And here you can see in this GATK tutorials workshop, germline resources, mother GAB SNPs. So this is just a list of all of these SNPs in the genomes in a bottle truth sample for our file that sort of overlap. And so we're going to be using that. This funky resource GIAB is just saying that is just a label that we're going to be using for the output. And so we should be able to just add to every single one of our output VCFs a new annotation that corresponds to the genomes in a bottle status for that site. So give it a minute. And if we go down here, we should hopefully, for these sites, if everything went well, see this. Genomes in a bottle call set equals 3, genomes in a bottle call set equals 6, genomes in a bottle call set equals 4. Great. So we've now added into our VCF file annotations that correspond to the GIAB call set. Now, why are these discrete integers? Well, as it turns out, this is just one of the the outputs in genomes in a bottle that we can use. This is the number of distinct call sets within the GAAB set that we have that have called this variant. So when we say there's 4 variants here, or the call sets number is 4 here, that means 4 of the tools that they use to build the consensus call set for genomes in a bottle called this variant. Now, this approach, generally speaking, biases you towards kind of simpler sites that all the tools see when you're doing consensus calling. But, if you work at it and genomes in a bottle has been, iterated on over the course of years, slowly it is becoming a very comprehensive truth set.
Is there patterns like identify only the position that is identified by only one tool set that be discarded?
Yeah, so genomes in a bottle, they publish what they call the confidence intervals, and those correspond to the regions where we are reasonably confident that we are correct. And that usually corresponds to the cases where most of your tools agree. And, generally the easy stuff, like stuff in the exome for the most part, and like the non-repetitive parts of the genome, most tools are going to get that stuff broadly correct. However, in situations where genomes in a bottle, there's a lot of discordance, but there's some agreement, like maybe 2 or 3 or 4 tools. In this case, there are 7. I think the newer genomes in a bottle, it's using all kinds of alternate data, and it's much more fancy than that now. But if there's like 2 or 3 tools that support it, it's just going to be excluded from the confidence intervals. And we're just going to say, you know what, we don't know if it's really variant or not variant here. And what we care about is building a truth set that you can use for validation. So if it's messy and we don't know what the truth is, we're just going to kind of wave our hands and not worry about it. Mm-hmm.
Let's say you have a true value in your sample today, there's low confidence that it won't be the
So I think, you're saying that if you have a true variant in your sample that falls outside of the confidence intervals for genomes in a bottle. Yeah. So I think the idea is if you can do well on the confidence intervals within genomes in a bottle, which, at least for these samples covers a lot of the genome, hopefully that your performance works well outside of those regions as well, but it's harder to measure. And oftentimes those regions, it's not an accident that there are certain regions that genomes in a bottle mostly isn't confident over, because it's hard to call in those regions. So it's, probably it's sort of a messy problem to sort of deal with. Generally, their solution has been to use cross-validation with like PacBio samples, or, circular consensus-seq with PacBio or ONT samples to sort of get coverage deeper into those messy parts of the genome. But, generally speaking, genomes in a bottle it covers most of the genome in terms of its confidence intervals, but it is mostly the easy stuff in the genome. I hope that's helpful. All right. So point is, we can see these discrete numbers. We've added a new annotation into our VCF field that corresponds to the sort of genomes in a bottle confidence for those samples. Now we're just going to do a bit of, extra work. We're going to run variance to table. This is sort of a not super necessary step, but just, in R, it's really easy to read TSVs and it's not so easy to read VCFs. So we're just going to put everything out into a tabular format. Each of these -Fs here correspond to taking one of our attributes from the VCF file and sticking them into a column. And then this GF here means that we're putting an attribute from the genotype into a column. And actually, if you have a wider VCF than just one sample, like we do here, like if we had 3 or many, it would make one column cross each GF field that it has for each sample. So now we can print this out. Great. And we should see it's a tabular format. Look, it's columns and tabs. It's great. It is, so, it's always useful when we're doing this sort of R processing to print out our data and sort of get a sense of what we're actually looking at, just to make sure something didn't go wrong. If we had a field that was all NAs here, probably we made some mistake or typo in our previous step and we're missing something. But you can see a list of all the annotations that we are going to use for processing here. So base quality rank sum, MQ rank sum, read pos rank sum. We'll talk about those in a few minutes about what they actually sort of mean. That cool. The next step is even more hand wavy. So we're downloading ggplot2, and we are also downloading some grid extra and a bunch of extra packages. And just to make your lives easier, here are a bunch of standard functions for drawing exactly the plots that we care about today. ggplot is powerful. If you ever want to make similar looking plots, you can steal these and use these as a basis for what you want to do. But this is just going to make it a little bit easier for us to like say like, oh, here's a density plot. Cool. So hopefully that works. And you'll notice now that we're stepping into R land, just in RP, this is how you indicate that the cell is now being run within R. Just this little percent sign R. And you have to put that at the top of all of your cells. You can see there's a bunch of warning messages. That's just R being R. Cool. So now we can start looking at plots and looking at those annotations that we just created. So let's go down here. Let's load our data in. So this is just calling into R, uploading the GIAB text document that we just created with the TSVs. And now we can go down here and look at a plot. So the simplest plot that you would generally want to do, as a reminder, when John was showing you VQSR yesterday, it's variant quality score recalibration, right? So the quality score of the variant is probably the first just annotation flag field that we would maybe want to think about when we're thinking about filtering. So let's plot that here. Sorry, this is in the wrong order. That button. Cool. So let's make a density plot for the mother genomes in a bottle SNP, and let's plot it. What does this look like? Let's shrink this so we can actually plot it. Cool. So you can see now here's a distribution across our sample of what the quality scores were for all of the variants that were present in the mother's sample. You can see there's a lot of outliers here, though. There's a really, dense peak down in the very low quality region, and then it sort of goes out for a very long distance. Generally speaking, we consider, when we're doing this kind of filtering and when we're thinking about what the annotations mean, we probably want to be a little conservative about whether we consider these annotations to be real. A site that had qual of 10 or 20 times the expected qual for most of your variants in a site, maybe that is an indication that there's some sort of artifact. I'll tell you now that the quality score annotation is more or less a measurement of all of the evidence that supports the alternate alleles at a site. So it is sort of approximately 30 times the number of reads that actually supported the alt at this site. So, if you have 10,000 qual, you probably have like hundreds or maybe thousands of reads stacked up at a single position in the genome. And, maybe we think there's something wrong with that. Maybe that's not representative of what our real data should look like. So we can try cutting off our quality score here by changing the comments and rerunning. And now we get a nice new, at least cleaner looking distribution of what our non-outlier data look like. This is great. As I said, though, it's still a little bit hard to interpret. I mean, the quality score goes out to 5,000. Obviously, this is nothing like the Fred scores that we've talked about before previously, because 5,000 Fred is meaninglessly confident. But you can see maybe there's some patterns that we're seeing. There's sort of a long distribution, but maybe there's two modes here. I don't really know what's going on with that. But how about let's sort of think about the qual. Now let's input the truth data that we have. So the genomes in a bottle call set data. So if I comment out this and I use the truth data, what do we see? Something has changed. So this is, using R to do what it's good at. we have now split out all of our variants into separate distributions based on what their genomes in a bottle call set value is. And so you can actually see, like, these sort of pink and purple colors here correspond to the samples that the genomes in a bottle is actually pretty confident about. So do we see any patterns that we might want to use for filtering, or at least for drawing boundaries in this data? Yeah. Yes, exactly. So typically when we do filtering, so we do have a published like best practices set of filtering for the output of like GATK and VQSR. Well, not like less VQSR if you don't actually have data. And typically we suggest that you filter, I think it's either 20 or it's either 30 or 40 qual. I think it's 30. And that just means like you're filtering out variants that you called with just like a handful of reads that were not very high quality. And so, yeah, it sounds like you would probably want to filter out based on quality. Now, the problem is the quality here is sort of multiplicative by the depth. And so you end up with this large distribution. And it's really hard to say, except for the sites where you have very, very low depth, that the quality was bad. Because maybe you had very high depth and the quality was still bad. And you're not going to get anything out of this. So now we can talk about one of our other annotations, which is the qual by depth. This is one that actually works pretty well for filtering in a lot of cases. So let's make the next set of plots. And you can see here, we get a weird distribution for the qual by depth. What do we think these peaks represent in this distribution? Any ideas? So this is the quality score, which is basically the quality, the sum of all of the qualities of all of the reads and their support for any given allele, divided by the depth.
The regions, like a quality score, just below.
Yeah. Well, I'll give a hint. One of these peaks is about 2x the other peak. What could that represent? Yeah. What? Yeah, exactly. So as it turns out, the majority of cases, the quality score that is emitted by at least this version of the Illumina pipeline that we're using for this data, the quality score for most of your reads is going to be 30. So it's going to be 30, 30, 30, 30. But if you're at a site that is homozygous variant, basically all of those alleles are going to be 30, and then you're going to divide by the depth, and you're going to get about 30 average quality for your site. But if it is a well-behaved heterozygous variant, you're going to have 30, 30, 30, and you're going to divide by half the number of alleles because you have a het. And so you end up with this peak in the center. But what about this guy? Probably bad, right? That means that the depth of your alternate allele is very, very low and potentially not very well represented in your sample. As it turns out, we can actually see how likely those are to be real because we can cheat and have truth data. And did that work? I really hope it does. Oh, we're getting our errors live. Yes. Apologies. Put here, split GIAB call sets into the QD field here. And that should be good. So just if you want to do this yourself, you can do the split, add the split GAB call sets. So that just gives us the nice colored plot where it splits it up based on that annotation. And here we can see a pattern emerging. That little distribution with like very unbalanced allele fraction on the left, well, those are mostly NA, which means they didn't show up at all in our genomes in a bottle truth set. So as it turns out, those maybe are some sort of error, and we can probably at least comfortably decide to filter based on qual by depth. Where exactly you draw that line is a bit of a question of deciding how much you care about potentially getting rid of real true positives that are maybe a little messy. I think our general pipeline, like our general best practices for throwing away quality by depth or QD scores is to cut them off at 2, which like stuff that's below 2 QD is like really garbage and like should not really be considered. But you can probably get away with a slightly higher score than this. But great. So what we've essentially said is stuff that doesn't look close to about 50% coverage, or that doesn't have a nice, well-behaved distribution of allele depths that corresponds with our diploid sample, maybe is an error. And we can use that for filtering potentially. So now we can make even prettier plots. Here we're going to make what we call, what in this toolkit we're going to call the scatter plots with marginal density. So just sort of, let's step back for a second. You can do this, you can play this game and you can build these plots with any of the annotations that are in that call set. Feel free to. It's very neat. This can be a very helpful way to sort of visualize what these annotations mean and what sort of good values would be for those annotations. But now we're going to make plots. It's sort of hard to say though, because these are sort of 1D plots. You're only looking at the annotations for a single annotation in absence of all of its other accompanying data. And maybe there's interesting patterns that sort of emerge from seeing 2 annotations cross with each other. That's obviously what VQSR in the presentation yesterday was really all about. Sometimes, there are nonlinear ways of drawing probabilities around, or truth around your truth set to get better results. So let's do that. Let's build these plots. Here we just have listed the plot at the very top. That's what we're making here. In general, here we're just going to, these are just our best practices standard filters for haplotype caller data that we use. I'll give you a bit of a key for what each of these annotations mean. We just talked about qual. We talked about qual by depth. Strand odds ratio is a measurement of how stranded are, actually strands odds ratio and Fisher strand are actually both measurements of how strand biased our sample is. And then the rank sums, the MQ rank sum and read-pos rank sums, those are essentially a statistical measure of if I have an alternate allele. So this is something that only gets applied at the hets. But if I have an alternate allele and I have this measurement of mapping quality or read position, is there a statistically significant difference in the rank in that annotation between my alternate alleles and my baseline alleles? I know that I made that sound more confusing than it is. So essentially, if it's 0, that means that those 2 samples, there's no real difference in, say, the mapping quality between your alternate alleles and your reference alleles. Whereas if it's the more negative that number becomes, the more bad our alternate alleles supporting reads become relative to the reference. And we could talk a little bit about how that is actually computed, but it's not super necessary. I hope this works. Yes, this works. But as you can see with our truth data and doing exactly the same trick with coloring the genomes in a bottle call set confidence, we can now look at this plot in 3D and we can get, along the axes, the 2D representation of that plot. But now you can see what we were talking about with the quals, the very high qual score depth samples and the very high qual by depth samples, is that there's actually like a relative cluster of what good alleles look like in the genomes in a bottle truth set, at least for this sample. So, if you wanted to draw hard filters, you would maybe think about like, okay, well, maybe I can draw some lines to sort of cut around this distribution. But as we've already shown, VQSR is sort of the correct tool to think about these things. Though oftentimes you can be in a case where you don't really have enough data for running VQSR, or you have a single sample. And in the single sample case, you do want to sort of think, or if you have very few samples, you might want to think about using hard filters. But you do want to be careful because playing this game, it's really easy to get hung up on like, oh, I've made a pretty distribution where everything clusters well and I can get rid of all the stuff that doesn't fit my cluster. You know, running this with the genomes in a bottle truth set is meant to sort of give you an idea of, to sort of ground you, right? Like you can, like with this truth, you can get an idea of what you are actually looking for and what good data sort of looks like. And that's sort of the idea of bringing this up. Yeah. That's the depth, yes. So the number of reads. So you can see, so actually just to add to that, you can see the depth goes up to 250. This is a 30x sample, or at least most, or actually 50x sample. Most of the depth fields for most of our alleles in this sample go up to about 50. So stuff that's up to 250 maybe is an outlier, maybe it's not real. As it turns out, those sites, there's, you know, in BWA, just because the reference is not totally unique everywhere, when you map a lot, there's sites that, you know, just because they're homologous with stuff that isn't represented in the reference, or just because they look better than a lot of other sites, BWA will just pile on tons and tons of reads that don't belong. And so you end up with like really kind of messy sites that you can't make any calls in. And so calls that you do make there probably are very likely to be artifacts.
How many variants VQSR need to run?
That's a good question. So I think VQSR, the primary benefit that you get from it is using population statistics. I think you can run VQSR. John, if you want, if you know the answer to this question, the question is, how many variants do you need for VQSR to function correctly or reasonably? So generally speaking, we use VQSR in a, like if you're doing a multi-sample experiment, so you're doing joint calling, we use VQSR for our single sample pipeline, especially the one I'm about to talk about after this. The recommendation, like VQSR doesn't help as much. Like you need that population information to really like hammer home the benefits of VQSR. So sometimes this hard filtering is sort of all you can do if you're in a non-model organism or something.
Can we say just we need to have a whole exome and not a green panel? Or...
Yeah, so certainly with a whole exome, I would expect it would be like that might be well below the threshold for VQSR functioning well, I suspect. But we put that question in the document and we can maybe get back to you once we talk to some of our colleagues. Cool. I believe that's almost everything. Now let's just talk a little bit about running filters. So we saw the qual was bad. Let's try actually putting quality, like, you know, 30 was a cutoff that worked reasonably well in one of those first plots. And actually, if we look here, either of these are quality. We can actually just run variant filtration to just spit out that annotation. It should be noted when we filter variants in the variant context field, generally the pattern is that we will do something like we'll use this filter expression and then we name the filter what type of filter we actually used. And that means that when you're looking at your VCF, you actually get a list of all the filters that got applied. And that can be very useful if you say, you know, had very strict filters on your variant context file. And then you realize you've thrown away all your data because, you know, these filters were just too strict. You can easily just roll them back and look at what all the raw data actually looked like later in analysis. So you can see here, everything looks to be passing, but if we do a grep for qual 30 and we run, yeah, see, in the first 100, there was at least one sample that had lower than qual 30, and it had 27. So it didn't meet our exacting standard of 30 quality for this. Now, if you wanted to run multiple filters, we can do exactly that. Again, this is just our standard set of filters that we generally recommend that you use for most like haplotype caller variant calling data. And if we run it, you can see, again, most stuff passes in the sample. But there should be some filter fields in here further down. Here, we actually have been running this, like all of those plots we were just showing you, and this whole game that we've been playing with the pretty lines and drawing cutoffs against genomes in a bottle means that we actually have good truth data. So why don't we just say, what is our genotype concordance against that data for this sample? So we can just do that here. First, let's run it for this. So what we're using as truth is that same genomes in a bottle SNP sample. Here, as we talked about before, we're using as the -L, so the intervals that we're using, the mother high confidence bed file. So again, as I said, we're subsetting to just the regions where the genomes in a bottle concordance is high. So probably where there's like 7 or 6 or 5 tools that all agreed on the calls, and not the places where there were only 1 or 2 that put those in. And then we're going to run that, and we're going to spit out concordance. And then we're going to run this again on our simple qual QD filter. And we can see what the effects of running these various filtering approaches are for our data. And you can see the precision, so the recall precision. I wish I can, if you double click, you can cause it to fully collapse. Excellent. So you can see here, up here is what our general best practices filtering does to the data. So there's no indels. We filtered out, we subsetted to only SNPs earlier in this pipeline, just because SNPs are a little bit easier to see this, and filtering for indels is a little bit harder. But you can see here, when we use our best practices pipeline filters, we have only 99 false positives in this whole call set, but we have over 1,000 false negatives. So sites that were real sites that we threw away because we were too strict about filtering. However, when we use the less strict, just quality 30 filter, the false positives go up. So we are not filtering out as much of the garbage. However, we have much higher recall. We're not throwing away as many of the marginal sites that were real. This is generally kind of the trade-off you want to think about when you're doing filtering of any kind. You know, when you're filtering, there's always a risk that you're throwing away some of your real data. And you know, in bioinformatics, depending on your application, generally speaking, we tend to err on the side of being very precise, because you know, if you call a variant and that variant looks real but isn't, and you act on it in some clinical setting, then that's really, really bad. Or you paid a lot of money to do Sanger sequencing on a site that wasn't real in the first place. So generally, you know, this is a trade-off, and you want to think about this trade-off when you're doing any kind of filtering. Are there any questions? Yes. So we talked about this last night, or last evening. So recall is a measurement of the true call or true positive accuracy. It's the number of true positives over the number of truth calls in the whole set. So when we have a recall of 98.4 or 0.984, that means that we've recovered 98.4 of all of the true positives in that set. And precision is measurement. I would have to look at the cheat sheet to tell you the exactly correct answer, but precision is measurement of how accurate the calls are. I believe it's yeah, exactly. Yes.
So, is it the same GTK concordance with the happy tool?
Yeah. So yeah. So GATK concordance. So there's lots and lots of tools. Happy, which I think maybe calls into RTG at its base.
Yes,
Yeah, exactly. So there's many tools out there to run concordance. For the most part, you're going to get very similar results. However, you know, at the margins, there's some noise. Like some tools do realignment of indels internally and try to build the haplotypes that correspond to what read groups you get, because there's a lot of representation issues that make it really difficult to say that two calls were exactly the same, even if they were, and they were even called correctly in both cases. In general, I think we probably use happy more often, but you know, it's convenient to use GATK in this context. Any other questions? We're only a little bit behind schedule. I'm going to switch us over, and we're going to talk a little bit about DRAGEN-GATK. And then after this is done, we'll take a break, and then we'll do a little bit of a demo of what how to actually use DRAGEN-GATK and run it in the cloud. All right, so hello again. I'm going to be talking a little bit about DRAGEN-GATK. So this is sort of our the latest, at least from our end, in germline short variant calling, and is a pretty big upgrade to haplotype caller as it stands. So what is DRAGEN-GATK? Who is Dragon? Dragon is a pipeline that was originally developed by a company called Edico, but then they got purchased by Illumina. And they sort of built their own accelerated version of the GATK4 best practices. And their sort of purpose was to build specialized fancy boxes that they sell that do your variant calling very, very quickly. They eventually got purchased by Illumina, and then we at the Broad started a collaboration with them to develop what we call DRAGEN-GATK. And this is a collaboration from both houses where we work together and develop software that is sort of under the broader umbrella of DRAGEN-GATK that sort of takes the best of both worlds. Transitions. So what is DRAGEN-GATK? What is this sort of collaboration that we have made with them? So as it turns out, there were actually a bunch of scientific improvements both in GATK and Dragon. Developers for the GATK have actually worked directly with developers from Dragon to share code and algorithm improvements sort of across the two pipelines. And so actually there's a lot of new algorithms that have been ported into GATK that were scientific improvements over the existing haplotype caller algorithms. On top of that, they've also released an open source version of their DRAGmap aligner, Dragon DRAGmap, I guess. That does perform better in certain circumstances, particularly on like it's better alt-aware awareness if you're on HG38, for example. They still have their fast accelerated FPGA algorithm, but we now have released a set of best practices that is part of our new variant calling pipeline that is available for everybody to use, at least for human data. The DRAGEN-GATK best practices. So you can do short variant calling and get results that are sort of functionally equivalent. And I'll talk a little bit later about what that means to the Dragon results in such a way that they are, at least to our minds, good enough that you can sort of mix and match call sets if necessary. You know, there's also some other details, you know, Illumina, I believe Dragon is available to be run on AWS, but we obviously support Google and Azure. Or Google and Terra, and then also Azure in general. And so, you know, this is making these algorithms and variant calling improvements available to more people in general. So why do we care? As it turns out that DRAGEN-GATK, or sorry, Dragon in general, actually, like, you know, when they went off and started developing their own tools, they had actually made some significant scientific improvements to variant calling just in haplotype caller. They were doing a better job, at least in the single sample case. And if you do better at single sample calling and are more accurate to start with, then all of the data that you feed into VQSR and all of the data that you do joint calling with is just going to be more accurate as a baseline. And so it helps to have better results. So we have released a new tool, DRAGEN-GATK, and we have demonstrated that we were able to very closely match their new results. I will talk a little bit about how to read these plots later, but you can sort of think of this as a rock curve if you're aware of what that means. So how does our pipeline differ? What have we changed about our best practices to sort of support DRAGEN-GATK? So DRAGEN-GATK sort of slots in not really exactly to the same pipeline we've been talking about for the last several days. It slots into our single sample best practices, which is very similar. It's just, you'll notice we produce maybe a GVCF and maybe we run CNN score variants, which can be run on a model for a single sample, but we don't actually ever do any sort of joint calling improvements. Dragon, as it turns out, is also very much optimized for the single sample use case. And so these improvements are sort of designed to make the most accurate possible single sample calling you can get on human genomes. And so, you know, these are all basically the same steps that you've been looking at for a while. We use haplotype caller. Eventually, maybe you'll use Funcatator, you use CNN score variants. Great. What does the Dragon pipeline look like? Mostly it's equivalent to the GATK pipeline. You still run an alignment. You'll notice a few things are missing. And then there's this extra step, the PCR training model step on the right here that takes in reference STRs. But you'll note, if you remember my presentation from yesterday morning, there's these steps like detecting active regions, assembly, HMM, and genotyper. Those all sound familiar because those are the steps of the haplotype caller. So they are broadly doing the same things that haplotype caller does. They just have some extra steps that are doing things better. So what are the differences? As I say, DRAGEN is hard to run for some people in some circumstances, just because it's proprietary code that you need access to an expensive FPGA machine for. Maybe you can run it in the cloud, maybe that's not so hard, but they also use DRAGMAP. There's some differences. And so we at the Broad wanted to sort of unify these differences and make a pipeline that could sort of handle both, that can output equivalent results from both, so that if you are a researcher who wants to, who has some DRAGEN data and doesn't necessarily have access to a DRAGEN box easily, you can use the DRAGEN-GATK as a stand-in for actually running DRAGEN. And so here are the differences in the pipeline. You'll notice like there's no BQSR. We've made some major structural differences to our large pipeline to accommodate being close to DRAGEN for these results. Cool. So what does the new DRAGEN-GATK pipeline look like? This is publicly available. I'll show you after the break where you can find this and where you can run this if you want. But generally speaking, it's the same steps. We used the DRAGMAP, the open source new version of the DRAGMAP aligner that they've published to map our reads. We still run marked duplicates. That's basically hasn't been changed in DRAGEN. But now we skip BQSR because it actually is not necessary given some of the genotyping changes that they've had. When we get to the variant discovery step, we've added in that extra little bit on the right, the STR model, which we'll talk about. And we sort of learn an STR error model on a per sample basis for each of our samples using the tool Calibrate Dragster model. And then we call variants with haplotype caller. So we're still using haplotype caller. We've just added in a bunch of extra configurations and arguments that correspond to the DRAGEN algorithms that have come about as part of the DRAGEN-GATK pipeline. And then finally, alternatively, you can still use the CNN. The results are not totally equivalent for the baseline CNN score variance 1D model. There is some wrangling that you would have to do, but in principle, you can do whatever post call set refinement that you were doing for your DRAGEN samples with DRAGEN-GATK samples. So what are the actual algorithmic improvements? Why did we go through all this trouble? And what was all of the work that went into this spent on? So you'll recognize this slide from yesterday. This is a representation of what all of the haplotype caller steps are. Just to hammer home, we're still doing haplotype caller in this pipeline. We're still identifying active regions, assembling plausible haplotypes, determining the per read pair HMM likelihoods using a hidden Markov model, and then we're genotyping, right? The DRAGEN changes primarily affect these last two steps. So they have updated models that do slightly better and do slightly better out of the box without a lot of expensive post-processing on the data to give you better scoring. So what are those models? There's three. There's the STR error model, which fits into the HMM. It's that pre-processing step we talked about, where you sort of learn a model of what STR errors look like in your sample ahead of time, and then you sort of use that learned model to update your weights that you use for the hidden Markov model for actually generating the likelihood of read versus haplotype. And if that is too in the weeds, we don't have to go into too much detail, but you should know that it makes the HMM much more accurate in cases where you have STRs in your samples. Then there's two other models, which are sort of updates to our genotyper. At a high level, our genotyper sort of makes a read independence assumption. So it assumes that if a read has an error and two reads have an error, that those two read errors were totally independent of each other, and that the chance of them having made the same mistake to emit exactly the same wrong base is just going to be the quality of those two bases, right? That's not actually how sequencing works in practice, right? There's certain reference confidence are reference contexts where, you know, the sequencer reads across them, and then all of your qualities for all of your reads sort of drops out because the sequencer just sort of can't handle that reference base. So we actually have, as part of DRAGEN-GATK, baked this directly into the haplotype caller genotyping model, is a bunch of extra models to model this exact possibility. So reference confidence, reference context sensitive linked base quality dropout, which helps us sort of cut down on false positives that are artifacts due to just messiness in sequencing technology that isn't really well characterized. And then also we deal with foreign read detection. This is just an extra model that we've added in that does, I mean, it's not so terribly different than the like existing contamination models that we have, but it does help cut down on artifacts that come from mapping errors. And importantly, it can also sort of bump homozygous reference calls into heterozygous calls in the case where we think the reference reads were actually wrong. Great. So what is an STR? I know I've used that term a lot over the past couple slides. So STRs are short tandem repeats. These are common motifs that are found all over the genome, where you get a repetitive section of like, you know, a couple bases. So for example, there's this repeat up here, TC, 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 TC. Sometimes you have, you know, a meaningful repeat of 5 of those, and it's easy to call with short reads. Sometimes you have, you know, dozens or hundreds of them repeated in the genome, and it becomes really hard to call with short reads. But these are all over the place, and they can come in many shapes and sizes, right? A TTG can also be an STR. And so the way that, and these are very hard regions to call for lots of reasons, right? The mapping becomes very difficult because, you know, maybe that TC motif is repeated all over the genome, or maybe it's really hard to tell exactly where to put your read on top of a TC motif. But also just the PCR, like these sites are very messy in PCR biologically just because of PCR slippage concerns. And so when DRAGEN and now DRAGEN-GATK is doing its pre-processing step, we run this tool called Calibrate Dragster model. So in addition to our reference, we have a side input that is a list of all of the STR sites across the whole genome that you can pre-compute using a tool that is also published. That basically lets us go out to the genome and look at all the sites that are STRs of a particular variety. So, you know, repeat length 2 and length 20, or 20 is actually fairly long. Repeat length 2 and length 5, right? And we go to those sites and we count up how many reads after we've aligned and duplicate marked and done all of those pre-processing steps, and count up how many reads actually have errors at those sites. And we use those errors to build a model for every type of STR error across the genome. How likely was it that those reads have errors? Using essentially a very similar assumption that we did in BQSR, that anything we see at this stage is going to be an error because variation is actually fairly unlikely. Once we do this with some math that you can ask me about after if you really care, we generate a new penalty for gap open and gap continuation penalty inside of the HMM that we then use to say, okay, well, we're at an STR in the reference. When I have my reads, I'm going to be less confident about the read map coming from any given haplotype that has a particular STR, just by virtue of the fact that I know in this sample there was a lot of messy PCR error. And what's neat about this model is this is sort of, you can do this for a single sample and actually do a pretty good job of learning the STRs by just counting up and subsampling all the STRs across the whole genome, because there's more than enough of them to do a pretty good job at modeling this. And this helps because, you know, PCR errors are sort of specific, and oftentimes they can be very noisy in your sample or not very noisy at all. And it's sort of hard to know ahead of time. And this gives you a model of exactly that in a way that functions a little bit better than BQSR does at exactly the same problem. Cool. So what about the genotyper models? This here on the left is an adaptation of some of the haplotype caller slides that you guys saw yesterday morning. This is our toy example that we walked through, and you can go look at those slides if you like, where we have an AA or A to C potential allele SNP present. We have added into the genotyper as part of BQD and FRD an entirely new potential outcome of genotyping. So instead of just saying that the sample was homozygous reference, heterozygous, or homozygous alt, we also add these sort of error models where we say, okay, well, maybe even though this looks like a head, maybe actually what happened was it was homozygous reference, and all of those alt reads were actually error reads in some capacity. And if we sort of build a model and do some sort of expectation maximization internally, maybe we can sort of say, actually, we can better describe the data this way by saying that, okay, all of those alt-C reads were bad, and we can distinguish that they were bad at this level and cut them out. And so when we go to genotype and we go to actually emit our results, we just take for each of the homozygous results, whatever is best amongst each of the baseline genotyping models and the error models. And so here, in this case, using entirely fabricated numbers, we have now upgraded our old genotyping call to a homozygous reference call with slightly less confidence, because there's the chance that that C allele could have been an error, which is great. This works pretty well, and I can talk about the details. If you want to ask and get an earful, you can ask me about the details later. So what are the two models? So what is BQD actually doing internally, and what sort of signal is it trying to filter out? As it turns out, so this is an example site. If you look at this in IGV, you would be suspicious about this site, right? So there is, a pretty good looking T allele on the left that looks like a reasonable SNP. However, on the alternate strand, there's a very messy G allele that is sort of very hard to it's kind of crappy for a lot of reasons. It doesn't have very high depth. It maybe has some low base qualities present, but you can also see all of the reads, or at least most of the reads that have that C allele, also have all kinds of other messy errors going on in them, right? You can see this read here. It's got lots of, low quality bases here. There's a lot of like low quality C's present here that also are G's present here, sorry, that also have a lot of like low quality bases that are present with them. And this is exactly the model that BQD is attempting to sort of build internally, is you're penalizing, if you can meaningfully draw a line internally within your alleles for reads that are different somehow. When you're genotyping, you can add that into the genotyping model as like, a read error model that is penalizing those bases and gives you a better idea of like saying, okay, well, maybe filtering is going to catch this later with the VCF annotations, but maybe not. Maybe the VCF annotations aren't sort of precise enough to capture this weird kind of evidence of like, there's a bunch of extra soft clips at the end of my bases, but the actual bases that correspond with the messy site are good. So I can't really rely on the base quality filtering. That's all sort of baked in. And so it is really just a new genotyping model that is built on top of the old regular diploid model. And you can see it even just uses the regular diploid model plus some like error terms to just sort of make this call. And it does it in a strand dependent way. And hopefully this results in better calling just at sites where, particularly for, NGS Illumina sequencing falls over and doesn't do well. The next model is FRD. It is very similar to BQD, but it is much more complicated. So I'm not going to show you the math or even try to. But generally speaking, it is looking for evidence that is a little bit more slight. It's looking for things that are unbalanced allele depth. Actually, this model doesn't do so well if you're in a non-diploid case compared to the other ones. But also as part of it, it sort of reaches into the very low mapping quality reads that normally we just filter out completely at the start of haplotype caller. And so when you're building a model of like, oh, are the reads that support this allele more likely to have lower mapping quality? it's going to look at all the reads that are at mapping quality like 2 and 3 and 5 that normally we would just ignore. They're crappy reads, but maybe that's enough evidence that we can use to say, okay, but also all the reads that do have this allele are more likely to have come from that low quality site. And so you sort of use that to sort of filter out your reads at the haplotype color level and produce better genotypes. So what does this mean for your output? It influences the hard filtering a little bit. The quality score that comes directly like out of DRAGEN-GATK and DRAGEN, it fairly closely matches between the two tools. However, the pattern is very different. So you can see here, here's like a distribution of what the variant quality scores look like. No longer is it kind of the raw, like, you know, about 30 times your depth that we were talking about in the last presentation. Now you get this weird peak at around 50. That is by design. Both of these models, just based on what we were talking about, essentially penalize heterozygous calls and sort of upgrade them into homozygous alt or homozygous reference calls. And as it turns out, just because of the way the math works out, it sort of ends up hard capping the quality at 50. And that is sort of by design, just to make it sort of less confident. The quality score actually becomes much better as a baseline filtering threshold in DRAGEN and DRAGEN-GATK than it is in regular GATK. So if you're doing a single sample call set, you can maybe trust your, or if you're doing something with a single sample and you don't want to use VQSR, it's a lot easier to use just the raw quality score. And it's now been sort of brought a little bit closer to earth. That's what this plot on the right is showing. So how do you run DRAGEN-GATK? All of the extra algorithms and code changes that are part of DRAGEN-GATK are totally configurable. You can mix and match pieces. You can run FRD and not BQD or the STR model. However, there's one unified argument. It is simple. If you want to run the DRAGEN improvements in haplotype caller, you can just run --DRAGEN mode, put it in. It'll spit out a list of everything that it changed in its configuration. So you can say, okay, well, maybe this one isn't good for my application. Maybe I'm doing a non-model organism, whatever. And you can spit that in. If you have a Dragster model file that's been learned, you can just use this extra argument here, pointing to the model file that got learned with calibrate Dragster model. All of this is available on Terra. We publish a featured workspace that shows you exactly how to use this and all of the extra steps in the pipeline, including like running the drag map aligner, if you think that's better for your data, etc. And it should be noted, we currently target DRAGEN version 3.4.12 equivalency. We are in the process of upgrading to 3.7.8 equivalency. We are targeting the version that all of us is using, just because they're using those versions of DRAGEN for actual processing. And so we're sort of upgrading in lockstep with them. And I believe UK Biobank also uses for their calling 3.7.8 at this point. Great. I know it sounded like I was done and I still have another section. So this is the functional equivalence section. We're going to sort of speed through this one at the end here. But I do want to say, so DRAGEN and GATK and DRAGEN are not sort of exactly matching in their results. It's actually really hard to take two code bases that have diverged years apart and are written in different languages and unify the results so they're byte identical. So the question becomes, if I have two call sets that are close but not identical, how do I sort of say, you know, in a scientifically validated and reasonable way that these two tools are close enough that I'm not going to cause horrible batch effects if I try to mix and match the data? And it turns out this is kind of a really hard problem conceptually. Most of the time when we generate call sets, we're like generating them all from like one pipeline or one sequencing center and passing them through the same parameters and hoping that everything matches up. And it becomes very difficult to say like, okay, well, what if I want to use these two different tools? So this is the problem that we solved. We built this functional equivalence pipeline to solve. The basic idea behind functional equivalence is, at least in like the multi-sample genomics perspective, what we care about is sort of keeping the difference between the two tool call sets less than the difference that's sort of inherent in sequencing bias that you get from just doing any sort of genomic data analysis statistically. So the idea here is you take one sample, you pass it through two completely different library prep pipelines, and you get two technical replicates that are the same underlying cell line, but were sequenced twice. And you run for each of those replicates, both, in this case, DRAGEN and DRAGEN-GATK, but you can really compare any two tools by this standard. This is sort of designed to be very modular and apply to many other applications. But we run DRAGEN and GATK on both of them. And what we want to sort of say is that the difference between DRAGEN and DRAGEN-GATK for that one replicate is less than the difference of if I took the same tool across two different technical replicates, right? So if I just sequence the same sample twice, are these results, are we more similar than having just done that process? So how do we actually measure this? We use sort of two standards. We use concordance, which is, we'll talk about in a second, and we also use the sort of difference in quality. Just because, again, DRAGEN, for their hard filtering, the output of DRAGEN and now DRAGEN-GATK, we use sort of a standard 10 approximately quality filter score. So we use the difference in quality as the standard here, that you can actually, in theory, use any annotation that you want to sort of build those plots on the right. Well, something got broken. There's a plot of, I swear there's a graphic here on the right. Maybe it clicks in. Yeah, it clicks in. Great. Excellent. So this is the first measure that we use is the concordance measure. Effectively, what you want to say, you just want to do the same raw concordance that we did before with a truth set, except we sort of play this game where for every sample, we take one of its other technical replicates and call that truth. And then we just use this Jacquard score, which is actually a symmetrical measurement of concordance that we can use, and we can sort of build a plot of how concordant are these two things against each other. So if we have, we can sort of measure for any two technical replicates, how concordant are DRAGEN and DRAGEN against each other versus how concordant are DRAGEN and DRAGEN-GATK against each other, just using a raw accuracy score or Jacquard score within concordance. And so in theory, when we're running functional equivalence, we kind of want this plot in the middle to be consistent with what we've been saying, to be above these two little bars on the right. Great, fantastic. And then when we actually do this across many regions and measurements, we actually do this across a bunch of samples. When we say that DRAGEN and GATK are functionally equivalent, we have a bunch of representative samples that we've sort of decided on. They include CHM, they include a bunch of NIST samples, they include some exomes. We do a bunch of stuff and we cut things down, and you can run them using whatever subdivisions of the genome you want to do for your analysis. Great. So I've been showing a lot of these ROC curves. They're receiver operator characteristic curves. Essentially, you can just think of them as on the bottom is the false positives and on the left is true positives. And if I have some sort of classifier, which you can think of genotype, just variant calling as a classifier, you can, if you become less precise, you tend to include more true positives. And so generally speaking, you kind of move up. If you lower your threshold for quality, you're going to include more variance in your sample, and you're going to include more false positives in your sample. So the idea is, if you want a good quality score, you kind of want to be in the top left of this sample. And we can measure this. We can there's a score we can use to look at any given point along this pipeline, which is the F1 score, which is computed in this way. Great. So again, when we have truth, as it turns out, just looking at the concordance between those two tools is not enough to say that these are functionally equivalent to the point where we can just feed them through the same pipeline and get equivalent results. Actually, as it turns out, the quality score matters a lot, and we targeted this for this analysis. We also generate these plots for comparing DRAGEN-GATK against each other. So, even if I have two plots, one tool could be systematically calling everything at quality score 10 above the other tool. And so if I try to use that as my threshold for filtering, I'm going to get different results, even though the overall call set is concordant. So how do we fix this problem? We actually produce these plots on the right, which essentially computes the F1 score for what the call set looks like for DRAGEN or GATK if we call the hard filtering cutoff that exact value. So if we call it, say, quality score 10, which is what we use in practice, what is the sort of distribution of F1 scores against truth? So in this case, against the genomes in a bottle call set. And is the intra-tool difference so is our like DRAGEN GATK versus DRAGEN on the same replicate tool difference less than the difference of using two technical replicates? In general, because visual rules of thumbs are always easier to remember, the green bar wants to be below these two other bars. Great, fantastic. So if you're using this pipeline for any sort of comparison, you just want to visually look at these plots. All of this is available on Terra. We're going to talk about this a little bit later, and I'll show you how to run this pipeline. But if you want to run functional equivalence between any two things, we actually don't just have in this workspace an example of DRAGEN and GATK. We also show an example that GATK4 and GATK4 are actually not functionally equivalent, even though they are highly concordant in their results. And if you have any questions about DRAGEN GATK, please go to our forums. This is actually a more general comment. Go to our forums. If you have any questions, we would be happy to help you. Great. I think that's all that I have for now. Are there any questions? Yes. So DRAGEN GATK is out now. The new version is going to come in, so probably a few months for the next version. Actually, most of the code is there. It's just there's some pieces that are missing, and we're sort of going on. So we'll publish some blog posts about it, I'm sure, when it's available. Anything else? Great. So we have a break. All right. I think everybody should be settling down soon. We've had a short break. I am going to, in this section, this is a guided demo, so this is distinct from the exercises that we walked through before, where you were walking through Jupyter Notebooks. This is going to be more of a demonstration of how you would typically use Terra to, run many samples or do, general genomics analysis. And I'm going to do that through the lens of these DRAGEN GATK and functional equivalence workspaces that we've been talking about. So starting from the beginning, if I want to go find a workspace that's a featured workspace, it should be relatively easy. If I want to say pick up and this is not just if I want to run stuff on Terra, but this is also a really good way to go find whittles for pipelines that I want to run that are sort of the best practices whittles for running those pipelines. So if we go here to view examples, we can see these are all the featured workspaces on Terra. Featured workspaces generally have very, very good descriptions and documentation and are intended to be sort of our best foot forward of like, here's what you should be doing for X, Y, and Z type of sample processing. So if we want to go find the featured workspace corresponding to DRAGEN GATK, which is what we're going to do now, you would just type in DRAGEN. And for this section, I'm going to be doing some like baking show switching between finished jobs and stuff. So like, don't worry about following along. The recording will be up available later. And actually there are other recordings demonstrating exactly how to use this specific workspace. So you can see here, I have found the DRAGEN GATK whole genome germline pipeline. That's a mouthful. This is the DRAGEN GATK like proper best practices workflow that we recommend that people use if you want to run DRAGEN GATK in its most validated, best foot forward way. You can see here there's a lot of documentation at the top. I'll go through a little bit of it, but this workspace is actually very well documented. There's a lot of comments and tutorials that are actually linked in directly from here, where you can go look at how people use this and what our advice is on how to run these things. And you can see here is what our quote unquote new best practices workflow looks like for single sample using DRAGEN-GATK. And again, this is if you're doing single sample human data and you have, all of the benefits that Dragon brings for human data. So you take your raw and map reads, you run drag map aligner, you do all these steps, and then you run haplotype caller and calibrate SDR model, and you get your GVCF, and then you do whatever you're going to do downstream with that. Maybe you've hard filtered, maybe you haven't. That is configurable. So as I say, there's another video of somebody else, possibly more coherently than me, describing to you how to run this. So, you have an option if you forget any of the information contained in here. There's some description of what is actually going on here. We can see that we're running the Dragon mode generated VCF. But importantly here, you can see what does it require as an input. So DRAGEN-GATK is basically just a modification of our baseline whole variant or whole genome single sample human calling pipeline. So what is the input to that? Well, the unmapped BAMs that come out of our alignment, or actually before our alignment software, that have been base called. So if we want to run DRAGEN-GATK on our samples, we just feed it in those UBAMs, and then it'll go through all of the pre-processing steps that we normally run for pre-processing before running DRAGEN-GATK. Now, if you have something that's in a different format that's not a UBAM, there are actually a bunch of tools, not available here, but you can find them also on Terra or on the cloud, to just run the revert SAM step to just turn your crams that have already been aligned back into UBAM, so you can do the processing from step one as though nothing was wrong. So what do you get as the output? And we're going to go over this a little bit in a minute. The output for DRAGEN-GATK is sort of three things. You get the reblock GVCF output, so the actual variant calls. You get some summary metrics, and importantly, you get these CRAM files. CRAM stands for compressed reference alignments map. So they're like BAM files, they're just reads, but they're compressed against the reference, so it's smaller. And so if you actually want to see like, oh, hey, I have some samples and I want to see what drag map alignment does to these samples, you can just run them through here and you will get out the CRAM file at the end that is the alignment using drag map. And you can also go find the WDL that this is using to run that specifically if you care. No, this is what I wanted to sort of highlight here. There are two example workflows here. If I actually go over here to my clone of this, to workflows, you can see there's two workflows. There's WGS functional equivalency and WGS maximum quality. So this is sort of if you want these are for two different sort of use cases. As it turns out, when we did the FE, like when we did all of the work to make ourselves functionally equivalent against the version of Dragon that we targeted, we actually had to strip away some of the improvements from GATK that weren't present in the Dragon pipeline, or at least that version of the Dragon pipeline. So to be functionally equivalent meant that we had to sort of disable some like real scientific improvements that we had, specifically some genotyping improvements that involve like spanning event genotyping, where you have long deletions, and also a few other just like QC control things. Actually, this workflow or this workspace has a pretty detailed description of all of the changes. Just first of all, pretty detailed description down here of all of the changes and what the differences are between those two workflows. So in both cases, we're not using BWA, we're using the drag map aligner, though that is configurable. You can choose to use BWA with both of these tools if you so desire. You're running Dragon, you're not performing BQSR, you're using Dragon hard filtering. However, this is the spanning event genotyping, which I just told you about, and then also this unmapped contaminant reads. This is just another piece of our pipeline that isn't present, at least in this version of Dragon, where if we see lots of like low quality contaminant reads that look like the presentation that Derek gave on Monday or Tuesday, those, lots of just like very shortly, poorly aligned reads that have lots of soft clips. Generally speaking, we throw those away in our pre-processing step because those are probably like, off target, wrong species contamination that we want to get rid of. And, that just improves our results for those messy, like large data sets because contamination happens fairly frequently. Okay, so how do I actually run DRAGEN-GATK? So I'm going to show you here the functionally equivalent version. first, though the other one is actually really not that simple. Really simple. You just change an argument. And I want to highlight a few things. So again, we're back in the like standard. I know we've showed you a lot of Jupyter notebooks, and that is one very nice way to use to do processing and just do all kinds of work in Terra. However, a Jupyter notebook is not very good if you have to run a dozen samples or 1,000 samples. This is sort of like, if you want to do most processing in Terra, generally speaking, this is the approach. Is you're going to make a workflow, you're going to, maybe put your WDL in DocStore, you're going to use a publicly accessible WDL, and you're going to change your settings. You'll notice here, this whole genome germline single sample workflow here is actually the just basic warp, how like this is our best practices WDL that we publish that you would want to use for just regular GATK4 variant calling. We have baked it in, so there's just one WDL. You just change you can enable the Dragon features, and you can a la carte turn some of them on or some of them off if you so choose. But since we're in Terra, we can actually click here and go into DocStore if we are so inclined, and you can see a lot of information about this WDL. You can see, when it was last updated. You can see where it's hosted in GitHub, and so you can go see where the WDL actually lives. Or if you are so inclined, you can go over here to files, and you can actually see the code. So this is the outermost file. So this is what we actually run when we run the WDL. But there's a bunch of subdirectories that we call into that are listed at the top, right? So you can see here we're importing this tasks brode unmapped to align BAM. If we ever get confused or we want to sort of browse through like, oh, there's this complicated messy WDL, and I want to see what each of the individual components are, well, we can just go here to select file. I can go tasks brode and scroll down here and go to exactly that task. So unmapped BAM to aligned BAM. And it has more sub BAMs. These are like spider webs of like lots of modular pieces that get sort of put together. So it can be a little messy to read through these, but DocStore can be very convenient if you want to go read through, what is in this WDL. And importantly, if you're like, oh, hey, I want to be able to run this tool that we talk about in this workshop, but I don't necessarily feel the need to use Terra, well, you can always come here. Basically, all of our WDLs are publicly hosted somewhere, and you can go find them on GitHub, or you can go find them in DocStore. The workflow says exactly where the workspace comes from. But you can see, exactly what arguments we're using for Dragon. So let's go back to our workflow for functional equivalence. So, okay, we've done that. You can see it's running on this read group set object. Just a reminder, Terra lets you hierarchically group data. So over here in read group, if we click on what the read group data is, you can see that we have all of this data here. All of these read group IDs, like read group, some Germain, some, Byzantine letters, and then, NA12878 downsampled. So we've actually, like, just in this workspace for the sake of making it easy to clone and not cost a lot of people a lot of money, we've downsampled this to about 3 gigabytes of data. So it's about 1/10 of the normal size. And we've just published one NA12878 sample that's like a standard, Illumina genome that's publicly available data so that you can run it. But you can see all this is just a bunch of unmapped BAMs corresponding to each of the read groups. If we go over here to the read group set and we want to actually look at how it works, this is actually, the hierarchical data storage in Terra. We have a read group. It also has its ID as being NA12878. And we are storing in this list here, 24 entities. This is just a list of all of our read groups. And so we're sort of binding them together through this set. If you're like, you can see we have an output CRAM and output VCF. Terra, these tables can often get very wide. I've already sort of done the work to make this more readable. But if you ever are like, oh, it's hard to read and scroll through all the stuff that's present in Terra, maybe there's other metrics I care about, you can always go here to settings, and you can see a list of everything that's in this data table, and you can just click, I want to see these contamination results and these cross-check fingerprint metrics, and you can click those in, and they will appear over here. Yeah, these two, they appeared at the front. So you can see what the contamination estimate was for this sample, if you're so inclined. Again, Terra really is quite convenient for running like a lot of samples through very repetitive pipeline tasks and storing all the data on a per sample level in this database. So going back here, going back to functional equivalence. So how are we actually keying? So as I said, I told you that this is running our basic like general publishing best practices pipeline for running haplotype caller and running through our like whole genome single sample pipeline. Again, we're just adding in some flags that say run Dragon mode here. So what is different? So you can see here, what are the inputs that we actually need to fill in to make this sort of runnable. There's some POPI settings, don't worry about those too much. There's this big object for references. These are just all of the possible side inputs you could maybe need for any reference for any tool, except for the Dragon tools. And then when we're actually inputting our sample, we are using the sample and unmapped BAMs binding, which is just an object, and it's storing here the read group set ID and these read groups.flow cell unmapped BAM list. So we're just feeding a list of like, here's the name of the BAM, and here's what all of its unmapped BAM like BAM inputs are. So we're just going to go through and we're going to realign those as part of the DRAGEN-GATK pipeline. And then there's some other settings down here. But if we scroll down, and here's sort of the meat of this, if we want to run in functional equivalence mode or best quality mode, we go here. So it is just a Boolean argument. You just set the functional equivalence mode to true and the quality mode to false, and vice versa. And it's sort of that simple. Somewhere in here, there is also a Boolean just saying I'm running in Dragon mode. The only real difference in terms of input that you need for Dragon is this table here. You need to input, so the drag map aligner has its own like set of binary like indexes and special files that side inputs that it needs for running its reference. There is a tool that is part of the drag map code base that will spit out and generate all of those files for you. We have publicly hosted the H238 versions of all of these files for exactly the functional equivalent, like, masked version of the Dragon reference. So if you ever want to like say, oh, I want to run DRAGEN-GATK, I want this, we publicly host these files. So if you ever need to like find, what should I be using to run DRAGEN-GATK or drag map aligner, even if I'm using drag map for another application, you would go here to just see an example of what files are necessary. Cool. So that's sort of a run through of what's in here and what all the data and what all the arguments look like. And again, I can see what the script looks like by just tabbing over here to script one more time. But if I want to actually run it, I just go to read group set, which again is my binding that says all of those individual read groups were part of the same sample. And I hit select data, and I'm only running here NA12878, just because that's the only one that's in here. And I hit scroll down and hit OK. And I hit run analysis, and I hit launch. And it should be running. That will take 4 hours or so, though call caching, I think, should make this really fast because I ran this recently in this workspace. So hopefully I can tab over here and we can talk a little bit. So what is the maximum quality mode for DRAGEN-GATK and what difference does it make for our calls? So these are more of our rock curves from before. On the left here these are specifically for SNPs and the CHM samples that we use for truth validation for Dragon and GATK. And you can see on the left here, when I showed you the DRAGEN-GATK plots, actually, I was a little bit disingenuous when I showed you those plots because the colors were not quite exactly what they really mean. There are 4 things on this plot. There's legacy GATK, Dragon, and then there's this 4th one, which is Dragon-GATK with spanning deletions. You can see, maybe a little hint of there being red under this line, because Dragon and GATK actually, if you look at the rock plots for just plotting a quality score, using quality as a metric of filtering, it is very, very highly concordant with Dragon. You're basically getting exactly the same results. But if you want to enable spanning event genotyping, you actually can do better in some cases. And in particular, if we go here, so HCR and LCR stand for high complexity and low complexity regions. They're just kind of ways of subsetting the genomes in a bottle truth into some, with some knowledge of like how difficult the reference is at each place. And so we have a list of like a standard list of HCR regions and low complexity regions that mean like, they're highly repetitive and they have mapping artifacts and they tend to be more difficult for tools to call. As it turns out, with the spanning event genotyping and GATK, we can do much better. And DRAGEN doesn't look as good at those sites. Those sites aren't representative of the whole genome, but maybe, there's a reason why we publish the maximum quality mode, because at least in some circumstances, if you're looking at SNPs, if you're looking at certain versions of the tool, you really do, maybe DRAGEN-GATK with modifications, so it isn't functionally equivalent anymore, but with those modifications, you get better results overall. So, again, I just want to emphasize this is all modular. You can enable or disable any of these arguments in tools that are present in DRAGEN-GATK. And we actually have started doing that for some of our own internal pipelines that still use GATK. So, and again, it looks less good if you look at indels for this particular set of arguments. The spanning event genotyping can sort of create a little bit more noise in indels and GATK. There's a trade-off. This is sort of why we have this maximum quality mode and we sort of emphasize the FE version, because that is functionally equivalent against DRAGEN. So I did not mean to flash that. Great. So now we're back here to our workspace. This is now running. This should be good. Yeah, so it's submitted. As I say, it should take a while. It's probably going to be thinking for a while. We maybe don't really have to concern ourselves too much with it. I can assure you that it will probably succeed and we will get results out. However, you can see what a finished run looks like. It cost 95 cents for this sample. The workflow ID is here. And if we want to look at the dashboard, we can get some interesting information about how long it took, when it started and when it ended. We can look at the job manager and have to authenticate. And we can see what are each of the tasks that got run, what are all the inputs, what are the outputs. And here we can actually see the outputs. This is, we can see all the outputs of this tool, which is actually a lot of things, right? But the important outputs that we care about for DRAGEN-GATK here are this output cram, which is the input to haplotype caller. This is not the BAM out that we talked about before with all these haplotype caller outputs. This is actually the cram that has been duplicate marked, that has been, not BQSR recalibrated, because we actually don't want to use the BQSR recalibration for DRAGEN with those new variant calling models. That has been realigned with DRAGMap. And so this can be useful for you if you want to try using DRAGMap. It is better in some cases. It's useful in some cases as an aligner. And then here's the output VCF. And you can see this is hard filtered. I can choose to have it hard filter the outputs. Remember, when we hard filter anything, though, we are just putting a flag that is replacing the pass filter field in the VCF. So you can always unfilter something that has been filtered. So those variants still exist. If you want to use a different filtering metric, you're totally welcome to. That is available here in this VCF. So great. And there's a lot of outputs. If you care about the timing diagram, you can see how long everything took to run. Don't concern yourself too much with these. These can be very confusing unless you know what the whittle you're running is. Okay, so one more plug I guess I'm going to make to sort of close the loop on this. So that was actually running DRAGEN-GATK. If you ever want to run DRAGEN-GATK or even just our regular whole genome germline single sample workflow, like best practices workflow, that's all available there. You can find it. You can find the whittles. You can run any of the tools that are present there for your samples. However, if you want to run functional equivalence, whether it is on DRAGEN and GATK, just to sort of keep us honest and see that the results that we sort of present forward are correct, or you want to compare two different pipelines. Maybe you want to compare, I don't know, GATK3 and GATK4, or Freebase and some other tool, and you think that you've sort of mashed them together enough that they should look similar, well, you can run functional equivalence. So again, this is a featured workspace. If I type in functional equivalence. I get here and I open it up here. And you can see here, again, more documentation, more data. We generally like to have featured workspaces be self-documenting and sort of easy to understand. You can see actually the functional equivalence workflow is a little bit more expensive to run than what we just showed you with the downsample DAM. That's just because it's running a lot of crosswise comparisons of RTG of like every single replicate against every single other replicate and every other combination. So you're running a lot of like inefficient RTG tool calls, but the expense is not too high. But how do you actually run functional equivalence? You can see here we have sort of two pipelines here. We actually have present in this workspace two entirely different workflows, one of which for running functional equivalence. This is just a sort of failed control test that we wanted to show of two things that are not functional equivalent. So in this case, GATK3 versus GATK4. GATK4 is not equivalent to GATK3 because it's better, or at least we think it's better. But we can also show exactly the correct version of functional equivalence for DRAGEN versus GATK. So just to hammer home, how do we do this comparison? If we go over here to data and we look at sample, reminder, so when we run the real functional equivalence for DRAGEN and GATK, we use many replicates across many samples. However, for this, we've published the actual 3 replicates of HE002 that we use for this that are public data. Now, a reminder, if you want to run functional equivalence, you need at least 2 replicates of any one sample. Just because in order to do that intersample comparison, you need to have some power to say how variable is that sample. And generally, you maybe want 3 as kind of the sweet spot. Once you get beyond maybe 5 or 6, it becomes really expensive because you are doing a lot of like basically a combinatorial number of comparisons, and it's sort of not meaningful. Like it's not helping you past a certain point. But you can see here, we have here the confidence intervals for truth. We have the data set, which is the same for all these samples because they're all HE002. Then we have the DRAGEN 3.4 VCFs, the DRAGEN VCFs, or the DRAGEN GATK VCFs rather, GATK3 and GATK4. Great. So here are the workflows. We have functional equivalence. And as you can see, as the input, it's publicly available just on GitHub. We're just using the main version for now. What is it operating on? It's operating on these sample set bindings. So when we take inputs, we want to run on many samples at once. So we want to run on the sort of superset of all of those things. And how are we running these tools? Well, this is an example of just all the things you want to sort of input. It's using HapMap. It's using a bunch of sources of truth to sort of make a very specific comparison between, in this case, GATK3 and GATK4. And it's using just that GATK3 VCF and GATK4 VCF. And so if I wanted to run this, I just go select data. I go select my one sample, and maybe I have more samples. And I scroll down because of that. I hit run analysis, and I hit go. And this is not my clone of the functional equivalence workspace. And so everyone will see that run forever. Great. You know what? We're doing it live. But if I want to see what the actual results of functional equivalence are, I can just click into these workflows. It actually spits out the data into here in our sample set. So I can actually read the plots for what the comparisons of all my tools are. So if I wanted to see, okay, well, what do the FE plots, so those like box plots, look like for GATK3 and GATK4, how like Jacquard score concordant are they with each other? Well, I can just read that. That's in here in the FE plots. And I can click here and Google will show me a preview of what the actual PNG file looks like in Google. And I can download this, I can do whatever I want with this. You can see if I compare GATK3 and GATK4, they're actually very highly concordant in the variance that they output on exactly the same sample. They're doing broadly similar things for calling, but they're still not equivalent in a mix and match way that isn't going to cause batch effects. And the reason for that is if we go back here and we look at these F1 plots, you can see the F1 score, the variant quality score that we output from the two tools is not equivalent at all, right? This green line is all over the place, which says that if I sort of mix and match the two tools, I'm going to get a quality that is totally inequivalent and not as accurate between the two tools in a way that's not going to cause batch effects and cause weird artifacts. The tools are fairly consistent with themselves. When they report that something is 10 quality, it is pretty similar to what other 10 quality calls look like across the same sample of different replicates of the same sample, but across exactly the same technical replicate, you get totally inequivalent results. So that's saying something is not functionally equivalent. However, if we want to see what this looks like, and mostly if we want to use the functional equivalence results to actually see, see it as a report, see all the summary. And if I have many, many samples, maybe I want to see all the samples in one place. I can show you the results. So this is running that other pipeline. This is comparing DRAGEN, the version of DRAGEN that we target for functional equivalence, some VCFs that have been run through DRAGEN in exactly the correct configuration to get functional equivalence against GATK, and exactly the correct version of GATK to run functional equivalence against DRAGEN. So if we want to see what the results look like, I can just go here to Job Manager. I can go down here to Outputs, and I would highlight here, there's this HTML report object. So I can click on that. I can go to Report, and this is just the Google Cloud Storage bucket, like UI. If you do a lot of stuff in Terra, you'll sort of get used to this. It just lets you browse and view the data. You can see where it's actually stored. Here's your FireCloud bucket. Here's, where the stuff, here's what, Cromwell has sort of put all of this data. But we can click here on the authenticated URL and we can see, oh, hey, here's a full report. And at the top, because I'm lazy, I can just read that they're functionally equivalent and they can wash my hands and be done. But if I want to go look at the results, I can go look at these box plots. I can see they're fine. I can go look at these quality score plots and I can see, oh, well, we're doing great. Maybe the indels in the HCR, so the high complexity region indels are a little bit more marginal, but they're still close enough that we can call them functionally equivalent. And we're going to say that they're good. And all of this is configurable. We, as a standard, use high complexity, low complexity region, and all SNPs and all indels as our sort of standard for calling, just for diagnostic purposes, because we care if something's wrong, is it wrong in the bad stuff or the good stuff? But you can actually put in any other ways of sort of subdividing your samples into this, and you can have it spit out any kinds of plots that you like. This is just sort of a framework for that we thought was rather helpful for doing this project against DRAGEN and GATK for comparing two things that are like but not exactly alike. And I think that's it. Are there any questions? So the question is, what should we look at when we're concerned about batch effects? So generally speaking, it's really hard to mix two data sets that have been made that are inequivalent. Oftentimes you're going to see, if you try to train a machine learning model, right? Oftentimes you'll see that the model just learns which call set it came from before learning any actual real truth. Generally speaking, if we're using something that's sort of equivalent like qual, and if we're when you're doing functional equivalence and you're trying to decide is something mixable, you want to be thinking about what the downstream applications are. So the reason why we targeted these annotations and this sort of mechanism of calling something equivalent for GATK and DRAGEN is because, the goal is that we're going to have something that's equivalent. If you take BAMs that come out of large call set pipelines that are run on DRAGEN, you should be able to run the GATK, DRAGEN GATK on the same UBAM input and get something that's very close and that's usable for whatever your other project is. So that's why we targeted these things. You would want to look at annotations for the VCF. Often those can be very different between tools. There are some annotations that are not equivalent between DRAGEN and GATK still, that mostly don't matter too much, that maybe we don't use for BQSR or what have you. But it is hard. It is, this downstream from calling part of the talk, it gets sort of into the artistry of the work that we do. Any other questions? Yes. Exactly, correct. Yeah. And so the agreement with GATK and Dragon was actually like much looser than the one that we held ourselves to when we did the work. We originally agreed that if you took two different technical replicates from the same cell line, sequenced at two different sequencing centers, and combined them, then you would be able to play this game and have them look functional equivalent. That is a much bigger ask than just saying two different replicates from two different library preps run on the same sequencers, right? Which is what we do. Did that answer your question? Okay. Cool. All right. Well, unless there's any further questions, I believe we have another break.
All right, everyone, it's about 5 to 12, so let's get started again. This will be the last session before we go out to lunch. And I'll be going over just an introduction of some of the things that we're going to see after lunch. So it's an intro to somatic variant discovery and a little bit of background on somatic variants and why we have to have separate pipelines for the somatic case. So when we're talking about somatic variants, we're talking typically about the cancer case. So we've got normal tissue that eventually starts accumulating these mutations over the course of an organism's life. You have a driver mutation that then creates heterogeneity in the tissue that becomes this subclonal population of tissue with that driver mutation. And we accumulate passenger mutations along the way, but those are not really driving these new subclonal populations. They're just along for the ride, hence passenger. And over time, we'll develop a few more of these driver mutations and a few more passenger mutations. And each time we do, these become sort of distinct subclones of whatever the tissue that this cancer is growing in. And this may not even be cancer yet. It could be precancer. It could just be a normal extra skin growth that you have, or it might not even be detectable. You might not know. It could be totally fine. But the main point is that over time, we're accumulating these mutations that effectively result in differences in the tissue that it's growing in terms of the cell population that you have. So each one of these different colors is going to have a different set of mutations in it that may or may not be contributing to cancer. So how can we help make the problem a little bit easier to solve if we've got all of these different populations we need to look at the same time? What we can do is we can have the samples from both, the normal cells, so some healthy part of your body, and we can have samples from the tumor itself. And if we do that, then it becomes a little bit easier to distinguish between these two things, even though the tumor population, as you'll see in a moment, is going to be somewhat heterogeneous. And over time, there's a number of cancer determinants that can lead to these tumor cells having these alterations in them. There's sort of background genetic variation that can lead to a predisposition to one type or another type of cancer. And there's also these accumulated factors that can be genetic, like if you smoke too much, or if you smoke at all, really, or if you've been exposed to a virus that can lead to certain cancers like HPV or something like that. Even sex can be a determinant for how likely you are to develop different kinds of cancers. Like most commonly, women get breast cancer, but men can get breast cancer too, and that's very well known at this point. Similarly, to stick with the breast cancer example for a moment, we have these high penetrance genes and high penetrance alleles that we know are really strong driving forces behind cancers, like the BRCA1 and BRCA2 mutations, but those account for a very small percentage of cancers overall. So we really need to be careful and take a look at all of the different mutations and all of the different variants that we find in samples. And again, the best way to do that is to sequence from both the normal and tumor cells so that we can make that determination. And as I said, it can be pretty complicated because over time, you end up with many small populations of different cells in whatever the tissue is you're looking at that have different variant profiles effectively. And so when you're looking at doing a screening for cancer on a genetic basis, usually you'll end up getting a biopsy of whatever that region in your body is that you're looking at. And the biopsy will be a small chunk of tissue that gets taken out and looked at under a microscope for morphology, and different morphologies can be specific to different kinds of cancers. But when we're talking about sequencing, we then will take that biopsy and put it into a sequencer so that we can see what the genetic profile is. And one of the issues that we run up against is the margins that you get for your biopsies are never completely clean. You can never only get cancer tissue and exclude all of the normal tissue. In fact, you might not want to do that because in a lot of cases with a biopsy, you're excising an entire tumor as far as anyone can tell. And in fact, the makeup of these tumors can be a little bit different from one another as well. So we have, this biopsy from the left set of cells here, that's really primarily this single line of cancerous tissue with one single subclonal population that's also cancerous inside this precancerous or dysplastic tissue, and then surrounded by normal tissue. So in this particular sample, you're going to have primarily cancer reads when you've actually done the sequencing. On the other hand, here, you're going to have, again, primarily cancer, because that's where most of the area in this biopsy came from. But they're different subclonal populations that are cancerous. They're almost 9 individual cancers in and of themselves. That is also, again, surrounded by this dysplastic tissue and some normal tissue around it. And even some of these cancer populations have subclones inside of them. And this heterogeneity makes it incredibly difficult to find the specific variants that are driving these mutations or driving these cell growths in this cancer. And in addition, we have some normal tissue there as well. So you could think of that as normal contamination. We don't have a pure cancer sample in any case, even when you have these really strong, large populations of cancer tissue. So it becomes really difficult to go in and just call these things without taking both the tumor purity and the heterogeneity into account. And there's one other complicating factor. We still have all the germline variants to deal with. So we talked about germline variant discovery already. All of the same germline variants that are present in an organism are going to be there in that organism when and if they develop cancer. So in addition to discovering these somatic variants, we need to make sure we don't count the germline variants as somatic variants. So what this all leads to is that effectively your signal to noise ratio in germline variant discovery is much higher than your signal to noise ratio is in the somatic case. And this is because of all the things that I mentioned: tumor heterogeneity, tumor normal contamination, other mutations that might be there that aren't really driving the cancerous growth. And this is just sort of a schematic demonstrating that, but it also means that we can't just make the same assumptions that we would for germline calling. So here, for example, in humans, at least, we're diploid, so we're expecting 2 copies of everything. And if we assume that, then if we have, a single mutational event that changes something in the body, we can assume pretty easily that there's only going to be about 50% of your reads that will support that conclusion, that there was a variant of that position, because we know that as diploids, we have 2 copies of our DNA in each cell. That's great. And this is the allelic fraction, not the allele frequency. AF is a little bit overloaded, I'm afraid. In the somatic case, it's much more complicated because we do have all of those subclonal populations that are a little bit different, and the driving mutations in each one may in fact be different. So rather than being able to assume this diploid makeup of each cell, we can have, some normal contamination that's diploid, that's fine. We can have two populations of tumors, one of which has only mutation A, and then we have, a progeny of that that's accumulated another mutation in this B allele. And maybe there's also been some copy number variation there as well. So we've, instead of having diploid cells, they're now tetraploid. And if this is the case, then 20% of your reads from this sample will support allele A being a variant, and only 10% will support allele B being a true variant. So when we've built up these somatic callers, all of this has been taken into account so that we can have good calls, even though there's very little supporting data for each of these somatic calls. And this is really hammering that home. So as I said, all of the considerations we've made when we're doing germline variant calling still apply to somatic calling or tumor calling. We have all of the same artifacts we need to pull out: batch effects, cross-sample contamination, sample swaps, sequencing artifacts, all of that stuff. And then we have even more problems because we have, the tumor normal contamination, tumor heterogeneity, and there's also a bunch of different mutational signals that happen in different kinds of cancers. So they can be, one specific cancer is caused by a SNP, another specific cancer can be driven by a copy number alteration, and even structural variants. So it's incredibly complicated, and each one of these cases really requires separate considerations. So there are many different tools to call these different kinds of variants. The overview today and what you'll see after lunch is for copy number alterations and small variants, so SNPs and indels, or single nucleotide variants and indels. Yes. It's a good question. So the question is, what is the gold standard for normal tissue? Is it blood? Is it mouth swabs? What is it? What I've seen is typically it can be blood. Usually what will happen is that you'll get a tumor biopsy, and then you'll have a biopsy from the same tissue type in a similar region to where that came from. Or if you can't find something there, then if it's muscle, for example, you'll take a sample from muscle in some other part of the body, ideally from the same muscle, but that's because you want these things to be as similar as possible so you can pull out the normal variation from whatever the tumor variation is. So blood will work in a pinch. I think it's probably best to have matched tissue types and ideally co-localized tumor normal biopsies. So really this is a quite complicated problem, especially considering how relatively easy the germline calling was, because this is additional complications on top of that. And so to really drive that home, here's a karyotype of a breast cancer cell line, HCC 1954, and a normal cell karyotype here. And what you can see is, these are nice and diploid. They've got 2 copies of everything. Here we've got a whole bunch of copies of chromosome 1. That's, like 5.5, because the other ends of these 2 are wrong. 4 copies of 2, I suppose. And it goes on. And you can see that there's been some really strange structural variation in a lot of these things, where you've got basically a full rainbow of different chromosomes all ligated together somehow in this unfortunate, well, it's a cell line, but if it were a person in this unfortunate individual, this is really kind of showcasing how complicated some of these things can be. And this is not even at the small variant resolution. This is at a really high level at the chromosome resolution. So you can imagine that all of those coverage concerns that I mentioned earlier and those ploidy concerns are very much valid in this particular sample. And I don't know if this is necessarily typical, but very often we see signals that are similar to these sorts of things when dealing with the somatic case. And, as an aside, this is a breast cancer cell line, and Rosalind Franklin, who I would say did more than her fair share of work discovering all of this stuff, in fact, died from breast cancer. So, ovarian cancer, which is similar to this in that it was a copy number variation driven cancer. So, yeah, it's fairly important. So let's talk a little bit about the workflows. Again, I'm not going to go too much into detail here because after lunch, we're going to hear about both the copy number and small variant calling workflows and processes in detail. But this is what the workflows look like. These should look vaguely familiar, as I think we've shown these a couple of times already in some of the overviews. But roughly, we have pre-processing and then variant calling and filtering for the somatic small variant discovery. And we do something similar. It's not quite pre-processing and variant calling and filtering in the CNV side, but there is some pre-processing that has to happen before you can actually find these copy number alterations. And then you do a little bit of analysis afterwards, and then you call the segments themselves. With copy number analysis, you're, instead of producing a SNP or a small indel or something in a VCF file, you end up getting what regions of the genome have been either duplicated or deleted or partially deleted. So the output's a little bit different, and these are much larger events typically. So for the somatic case, I mentioned these matched tumor normal samples that you're going to want to have when you're doing this kind of calling. And this is an example of a tumor normal data set for the somatic calling case. And what you can see is the normal on top and the tumor on the bottom. And we have a germline SNP that's present in both the normal and the tumor sample. This is exactly what we would expect because the tumor is effectively a progeny of normal tissue. So it would have all of the mutational events that had occurred in the normal relative to the reference. The difference here is that there's also this really small signal here for this variant, and it's only present in the tumor sample. Now, if we did the same thing that we would do with a germline sample and just tried to call this in absence of anything else, we'd probably say that both of these were variants because we don't have the context that this was present in the normal. So having a matched normal for your tumor really helps to do this filtering. But it is possible to run in tumor only mode. Yeah, do you have a question? Yeah, no worries.
What about when you like take from
Yeah, so the question is, if you have a lot of samples that have been preserved like FFPE, how can you do tumor normal analysis with them? The really short answer is, if you haven't collected a matched normal at the time you've collected the tumor sample, you just don't have that data. You just don't have that information. You can do something like this, which may help, but you'll still need to do a lot of filtering afterwards. And so really quickly, what you do in tumor-only analysis is you run MUTEC2 in this case in tumor-only mode. And then if you're doing this, you want to have a panel of normals that will help you do some of that germline filtering that you found at the population level. Now, this is good to do in general anyway, because it can certainly help. In this case, you'd want to match it as closely as possible to the samples that you have, because you don't actually have the normal tissue sample that you would like. This will still produce a lot more variance than if you had the tumor normal matched samples, because there's still not, you'd get the population events out of your tumor sample, but you wouldn't necessarily get all of the individual germline events Now, germline events are fairly rare, so there shouldn't be that many of them, but you're still not going to have as good germline variant removal as you would if you had the matched sample. As an aside, FFPE samples are notoriously difficult to get good data out of because there's been a lot of degradation already. So there are some extra concerns for FFPE samples that you need to make when you're calling variants on them. I'm not very familiar with all of those things, except to say that it's significantly harder, but we can talk afterwards and I can direct you at some resources that might have more information on that. Yes. Good question. So the question is the expectation that you build your own panel of normals, or can you use a publicly available panel of normals that might be appropriate for your sample, say for FFPE or something else? There are public panels of normals that you can get. I don't think that they would be specific to FFPE, although maybe there are some. The preservation in FFPE, I know there's a particular set of signatures that that kind of preservation inserts into the DNA over time, but I don't know if a panel of normals for FFPE data would fully recapitulate all of those changes. But there are certainly panels of normals that you can download. We host a few of them actually publicly that you could just get that could be appropriate for something like this. They're human samples, which I like. If you're doing human data, so much the better. If you're not, then there may be panels of normals out there for other organisms. But when we're doing somatic calling, most often I've seen it done in humans because that's where we study this stuff primarily. Yes. I'm sorry, I'm having a hard time hearing you. Isn't there a bias between? Let me make sure I have your question right. So the question is, would you miss similar mutations between the tumor and normal with this kind of processing? Okay. The mutations that we really care about for the somatic use case are these driving mutations that really cause divergence in the cell populations. So if there is a mutation event in the normal tissue that's also present in the tumor tissue, we would expect that to be kind of normal variation, if that makes sense. Does that answer your question? Yes. Yes. Yeah, there's no reason you can't do that for these particular tools, or for the small variant detection tools for MUTEC2. It allows for input of a matched normal and a panel of normals. You can't do that comparison directly in the tool. As Mehrtash was saying, that's kind of one of those steps in tertiary analysis where then you would do this extra comparison to do extra filtering or, event discovery or similarities between precancerous, normal, and cancerous tumor cells. Yes. Mm-hmm. Yes. Yeah, so the question is, if you have tumor biopsy and normal cells from the same tissue, similar, but a little bit further away, but there's some kind of contamination in there, can you use an additional set of data to remove that contamination? In this case, what kind of contamination? Do you mean contamination from the tumor or? Yeah. Mm-hmm. Yeah. Yeah, it's a good question. but hopefully there's enough normal tissue in your normal sample that the variance in there will override the variance in the tumor. If I guess one of the problems with it is when you give the matched normal, it's sort of assumed that most of that matched normal, if not all of it, is going to be pretty clean from the contamination standpoint, that most of the contamination will be in the tumor sample. By adding in this panel of normals, both in tumor only mode and in or actually yeah, so I guess the short answer is if you have significant contamination in the normal sample, that will reduce some of the variance that you would find even with a tumor normal comparison. Yeah. So, but the follow-up to that is you can give this panel of normals, but that won't reduce the contamination variance. That will reduce the common genetic background variants that are present for that sample, but aren't necessarily present as germline variants. So if the contamination is specific to that tumor sample, yeah, having the normal sample will help, but if there's significant tumor contamination in the normal sample, then you're going to have degradation in the variants that come out. But as I was saying, the panel of normals is extremely helpful as well in this case, because you can not only reduce, some of the background genetic variation, but in this case, the panel of normals is also removing artifacts and systemic artifactual or other errors in variant calling from whatever your sequencing modality is. So if you're doing everything on NGS, on an Illumina, NextSeq, or whatever, you'd want to have a panel of normals from that same technology with the same kit so that you can remove those technical artifacts. Because when you're doing somatic calling, you want to have increased sensitivity so you don't miss things, and because the signal is so low, this is extremely helpful to filter out some of these artifacts that might show up as variants if you don't have knowledge of them ahead of time. And so I'll give you an overview of the somatic CNV discovery case. Again, after lunch, we're going to go over both of these things in more detail. So we're looking at copy number variation, and it's really about proportional coverage and then normalizing this proportional coverage to define where we have these regions where there's an increase in, the number of variants, or really the amount of DNA that would map to that region. So we've got all of these regions, or this big set of DNA regions that we've segmented here. And over time, you can see we've grouped them into different sets, but overall, there are two main copy number events here. There's a duplication event and hemideletion event, so half deletion event. And the way that this works is we have these raw coverage plots binned over, in this case, the y-axis is a region in the genome. We put them through some filtering and standardization so that the ranges are the same and they're actually comparable and things like that. We go through a segmentation phase where we're actually trying to figure out where the boundaries might be between candidate copy number events. Then we actually call those events and plot them along this deletion or amplification axis. And then we determine what the minor allele fraction is for each of those events. And again, this is very high level, but this is roughly what the pipeline looks like, so that we go from kind of just coverage to, this region of your genome has been duplicated. Now, it's important to note that this doesn't say where those duplications actually occur genetically. It could be that you've duplicated a segment of chromosome 3 and it lives in chromosome 5. It could be that it lives next door to its original location. There's no way to know because we're actually mapping all of these things back to a linear reference. But we are able to tell that that location on chromosome 3 has been duplicated. And where that lies relative to the rest of your genome may not be important. It might be, but it may not be important for whatever disease phenotype or other phenotype we're looking for. And once again, we want to use a panel of normals to pull out some of the artifacts and other potential false positives that we might have. This time, it's not a panel of normals from a VCF file. It's a panel of normals for coverage over the genome. So it's a similar idea, but it's distinct in that it's not a VCF, it's not small variants. It's in fact looking at, again, regions that have increased or decreased coverage. And you would use this to also decrease some of those technical artifacts I mentioned earlier because they're still present in the data that we call CNVs from. And that's everything for this talk. Any questions before lunch? Yes. Oh, I see. So the question is, when doing copy number analysis, are you able to tell whether it's inherited from maternal or paternal DNA? And I think the quick answer to that is no. Mehrtash, James, Derek, correct me if I'm wrong.
The quick answer is no, but it's certain.
Yeah. Yeah, that would fall into that tertiary analysis again, where you knew what the regions were that were duplicated or deleted, but you'd have to then get samples from the parents and then like call variants across all of these regions and see what the allele fractions for each of them are and try to match up the regions that are duplicated with the allele fractions in there with each parent so that you could actually figure it out. It's similar to some other like genome assembly things that you might want to do, or even haplotype calling, not in the sense of haplotype caller, but like long connectivity haplogroup building. Yes. I think they should work okay for exome, but it's better to have whole genome sequencing because you'll detect a copy number event if you only look at the exomes, but you won't have as good boundaries on it, obviously. So you'll still be able to make these determinations, but you won't have as good breakpoints. Anyone else? All right, then. That's it for the first half of the day. We'll break for lunch and come back at okay, 1:35, which gives about an hour.
Welcome back from lunch. Before we took our break, Jonn was going into detail about somatic variant discovery, and it seems like there's a lot of interests in that topic. So you're in luck if that describes you, because we've got a lot more about SNP and Indo discovery in somatic cells, particularly using Mutex2. So we had a good primer beforehand, and now we're going to go a little bit more in detail on this topic. And then finally, we're going to be able to explore Mutex2 using our workspace in Terra. let's get started. So Mutex2 is included in our GATK toolkit, genome analysis toolkit, but we have a very handy implementation of it that is accessible on Terra, which we'll be demoing today. Mutex2 is designed for somatic SNV and indel discovery. So the most likely use case that you would use it for is for looking for variants arising in cancer cells. At the most basic level, I mean, Jonn has already covered a lot of this information, but at the most basic level, the way that our pipeline is going to work is that we are going to start with our analysis-ready BAM files that is using, it's already gone through the pre-processing stages that we talked about at the beginning of this workshop. And then from there, we are going to just look for all the variants using Mutex2 that we can find. There's going to be a lot of information there, but we're going to need to enact several layers of filtration to get rid of any false positives that may arise. And so then we will, at the end of this, have a collection of variants that we can have very high confidence are true positives. So before we get our hands on the workflow itself, let's take this as an opportunity to learn more about how the pipeline works in more detail. So you've already seen a version of this that was shared by Jonn. And so this is our diagram for our optimized workflow for SNP and indel discovery using Mutex2. So we didn't go over it in detail beforehand, and I understand that now it looks a little bit daunting, but we'll break it down piece by piece and it will make a lot more sense by the end of this presentation. So but in essence, there are three main stages, as I was describing before. We've got our pre-processing stage, we've got our variant calling stage, and we've got our filtering stage. And this is very similar to some of the other pipelines we have for Germline, for example, if you were using haplotype caller, or if you were going to be using DRAGEN-GATK, it looks very similar to this, but the details are where it really matters. So on the left-hand side, that's where we start. We're going to have our analysis ready reads that we're going to be looking at. That's our BAM file. We're not going to be creating our BAM file for this demo. We are already assuming that you have created some kind of BAM file for your tumor samples or your normal samples. But in the next step, for variant calling, we are going to use Mutex2 to call variants and provide annotation. And so for inputs into Mutex2, it is going to be accepting several things. First of all, it's going to be taking TN pairs. Those are your tumor and normal pairs. These are the two different files representing those two different samples. And these are going to be used to more accurately identify the variants that are going to be found exclusively in your tumor samples and not found in the normal sample. So we got a little bit into the details of that in the previous presentation. Mutex2 can also accept, optionally, it can accept a panel of normals, and it can also optionally accept a germline population allele frequencies file. So with both of those, those are going to be used to help you skip sites which would otherwise constitute false positives. So we're going to go into a lot more detail into false positives in the upcoming slides and how we can get rid of them. And I'll also take this as an opportunity to say, as a side note, that a lot of our users initially find their way into the Mutex pipeline because they are interested in finding loss of heterozygosity events. Those are LOH events. And so LOH is where you have heterozygous somatic cells that become homozygous because of one of the two alleles getting lost. So on a surface, Mutex2 sounds like a good tool to use if that's the type of thing that you're interested in. However, in practice, it's really not the ideal use case for Mutex2. Instead, we have another tool that's much more qualified to deal with loss of heterozygosity events, and that is called allelic CNV. And so if that is something that you are interested in, you should look up allelic CNV, which is part of the GATK. So moving on, we've got our variant calling done from Mutex2, and it will give us some outputs as well as some metrics, which are going in the other direction there. But once we have our Mutex2 output and the metrics files, then we will move on to filtering. And so in our filtering stages, we will put all that together. And I should also say that some of the metrics info that we're going to be creating is optional, but we have the option to collect it or exclude it if we want to. So in this particular figure, we're representing the filtering of the variance as this tiny little square here, but in reality, there's a lot going on in that little box. So we are going to go into a lot more detail in those filtering steps in this presentation. Okay, and so once all that's done, once you've got your variance, you've got your SNPs and your indels, and they're ready for analysis after all of this, then you can go take care of some more downstream analysis using a tool like Funcotator, which will functionally annotate those variants and give you a lot more information about what's actually going on. So I'll go back. But before I continue, as I was saying, during the filtering stages, we want to make sure that we have a lot of information about which of the variants that we are going to be finding are false positives and which ones are the real positives. So naturally, we want to make sure that the workflows that we're using are going to be very sensitive in distinguishing signal and noise. And so therefore, we have to make sure that we're having a low false positive rate in the samples that we're creating. So then the next question is, how do we actually determine where false positives are coming from? So when it comes to false positives, at least for cancer genomics, there are three main sources of false positives. The first comes from artifacts, the second comes from germline events, and then the third comes from contamination. And we'll be talking about those in the next slides. So false positives. So I've got two areas here. We've got false positives due to artifacts and false positives due to germline events. So they're blue and red to distinguish them. And so here we've got for the artifacts side of things, we've got an alignment with 4 events that are represented in the tumor, and all the reads are in the same direction. And there's no events that are represented in the normal. So that the normal is on the bottom and the tumor's on the top. So these kind of false positives can arise when the sequencer has misread some bases or has created some other artifacts, although really a lot of that may depend on the origin of your tissue. So in other words, if there's some artifacts that came about from the sample prep, then they may be represented in your tumor samples. And so that would be something that we would want to exclude, because that is not really something that is real. It's not something that we would want to be identifying as a variant. It's something that we should be filtering out. These can also arise from issues with the alignment or the liner creating artifacts. So for example, if you have repeated or repetitive sequences in the genome, then those might also show up and have an effect on just creating artifacts in your tumor samples. So on the red area of this plot, we also have our false positives from germline events. And so this is the second type of false positive, and it typically occurs it occurs at germline polymorphic sites. So in reality, this is actually a major source of noise, and it typically occurs when we don't have enough coverage at a site. So what you can see is in this example, we've got very little coverage in the normal, but we have a lot more coverage in the germline event. And this is something that you may be likely to experience, because in reality, we have a limited amount of money to spend in doing sequencing and getting our samples. And so we're more likely to spend or invest more in high depth tumor coverage than we are in getting really high depth coverage of our normal samples. So we can also incorrectly conclude that an event is going to be somatic if we observe that there's a non-reference base in our tumor samples, but then we end up missing it in the normal. And so then that would lead us to think that a particular variant of event is somatic, even if it's not. And I'm not going to cover it here, but we can also get false positives from contamination. We will go into that later when we're talking about filtration, but contamination can come from a lot of different sources, and it really is something that you are liable to find it later on, but it's not something that we can definitively know where it came from unless you had access to the library prep, which you may or may not have had. So how do we combat these artifacts? Well, first of all, we want to help our tools to filter these artifacts and germline events by incorporating certain practices into our workflows. So the first thing that we can do is use base quality score recalibration, which we have already talked about earlier in this workshop. We can also use panel of normals, which we talked about in this previous in the presentation before this one. And just to reiterate, panel of normals are derived from the normal samples from the typically or ideally from the same batch of samples as the experimental samples, and a panel of normals is going to help us filter additional batch and aligner dependent artifacts out of our sample so that we can identify where our true positives lie. We can also use a database of known variant sites such as dbSNP and exact for germline cells or cosmic for somatic cells. And so that can be very helpful if we find that an event is in a polymorphism database like these ones is represented, but it's not available in cosmic, then we can ask for more support from the normal sample and then use that to disregard that event. So with all of these filtering safeguards, you might be wondering, why do we even need a match normal in the first place, since we already have, there's databases that tell you where these SNPs might be, where these polymorphic sites might be. So the issue is that even if we exclude commonly known polymorphic sites, the rate of novel polymorphic sites is still roughly 50-fold higher than that of the somatic mutation rate. So with numbers like that, it means that having a tumor normal matched analysis is absolutely necessary. So our standard approach is typically to calculate the evidence for an event in a tumor and then calculate the evidence for not seeing that event in the normal. And then we should be able to set some appropriate cutoffs and filtration for those scores to ensure that we still have a very low false positive rate. So in GATK, the statistical analysis is going to be more powerful than simply just subtracting the variants that are found in the normal from those that are found in the tumor sample. We talked a little bit about that earlier today. Instead, what we are going to be doing using MuTech2 in this pipeline is to pull data from both the normal and the tumor samples. And so by doing that, we are going to filter out the variants that the tools are identifying as being statistically likely to be in the germline. The values that we have can be tweaked in order to adjust for either greater or less sensitivity, depending on what our outputs tend to look like at the end of this, and also what your data requires. Going back to the panel of normals, I can give a little bit more detail on this, although we did get a fair amount during the previous talk. But basically, the panel of normals is a VCF of calls that are made from a set of unrelated normal samples. So they're not the exact same samples that you're using for your experiment. We also use the panel of normals to eliminate recurring technical artifacts from our databases, or sorry, from our data sets. And ideally, we should be processing our normals using the same data generation techniques that we use for our tumor samples. That's the ideal case. So if you use a specific exome capture kit on your tumor samples, then ideally you would be using the same method of, or the same kit for your normal sample as well. As another example, if you were using a particular sequencing platform for one, then you would use the same platform for the other. And in general, that's just good experimental design to eliminate as many possible variables from our different samples as possible. But in reality, that's not always possible. Sometimes you don't have access to some normal samples that you can use, or at least not under the exact same conditions. So in cases like that, it is possible to run the MuTech2 workflow without a match normal at all. And that's why we have something called tumor only mode, which lets you run the code without a panel of normals. However, in general, we do not recommend doing this. And the reason for that is because even using data from a normal that was not collected under the exact same conditions as your tumor is still way better than not using a panel of normals at all. And during the exercise, we will actually be able to see the difference between using a panel of normals and not using a panel of normals. And it's on the order of hundreds of, or 500x difference in the amount of variance that we'll be calling. So we'll be able to see that. And just in general, even having a crappy panel of normals is better than just not having one at all. So it's always worth looking for one, or even just using one of the panel of normals that we provide from the Proto Institute. Are there any questions? Great. So now we're ready to actually get our samples and put them into MuTech2 now that we have a little bit of an idea of what we're trying to filter out. So let's get on with it. So when we're passing our samples through Mutex2, it's going to first be identifying any active regions that it can. So the tool that the tool Mutex2 is going to be using a modified statistic threshold, and it's going to be favoring the reference model at its core. So the tool is then going to be filtering differently based on whether or not it's looking at the tumor sample or the normal sample. So if Mutex2 is looking at the tumor sample, the filtering that it's going to use is going to be incredibly strict. The mapping quality is going to be, I think, greater than 20. Did I have it written down? I don't know. There's, if you know, you know. And the tool is also going to be disregarding any discrepant overlapping reads as well. In comparison, when Mutex2 is looking at the normal sample, the filtering is going to be very permissive, and any mapping quality is going to be allowed. And in addition to that, the tool is also going to keep alternate reads from the discrepant overlapping reads. So for the tumor, the filtering is very strict, and for the normal, the filtering is very permissive. That's the takeaway. So next, we're going to be covering a brief example where we can compare BWA and Mutex2 and see how they compare. So most people are familiar with BWA, and it's fine to use it in a lot of cases, but it might not be the best tool for the job in this particular case, as we're going to see. So here we have some BWA alignments for tumor and normals, which are in possession of 120 bases, or sorry, there's a 120 base deletion here. And as you can see, or as you can't see, there's evidence that there's some sort of event, but it's not really possible for here in IGV for us to actually recover what is that's happening there. So the insert sizes of the supporting reads are within a normal range, but this means that the event wouldn't be able to caught with a discordant read pair color like what we're looking at here. However, Mutex2 is able to assemble haplotypes using a pair hidden Markov model, which it uses to determine variance on a per read. Is able to determine per read likelihoods. And so for the active regions that it's looking at, and for any of the kept reads that it has, Mutex2 is going to be able to assemble those haplotypes. So specifically, the tool is going to reassemble reads into possible haplotypes using only the high quality reads that it can find. And then to account for any somatic events at low allele fractions, Mutex2 has a lower tolerance or an increased threshold for discarding those reads than it does with haplotype callers. So ultimately, this means that it's going to keep more variance than haplotype caller would, and then we can do our filtration after that. So here is a similar case study. We're going to show an example of how the method of reassembly is going to affect our ability to find the deletions in the alignment. So when we compare our previous example with this, which is Mutex2's reassembly, then we can see a difference. So not only that difference is there, but it also discovers these reads that are unambiguously phased into the normal haplotype, as well as a haplotype with insufficient likelihood, which is showing up there. So in this way, Mutex2 is superior to the previous method that we were looking at, but we'll have an opportunity when we actually use our tools in the demo section to actually see how all of this comes together. So I think this is the most important part. After we run our samples through Mutex2, then we are actually going to cover filtering for our raw variants. So what we want to do is pass our variants through several, or at least 3 different levels of filtering. So these are, and I'm going to cover them one at a time, but it's important to note that more or less these are occurring concurrently. But first, we're going to be looking at Mutex2. So Mutex2 is going to be treating our samples as diploid, unlike haplotype caller, but we do this in order to determine if the allele is either a heterozygous or homozygous, and we'll only be considering one alt allele at a time using this method. We're also going to be disregarding any ploidy arguments. So Mutex2 is always going to be assuming diploid normal for the inputs that you're putting into it. And so what that means is it really shouldn't matter if, or at least for this tool, whether or not we are changing the ploidy of our samples. It's not really sensitive to the ploidy of your samples, and it's just agnostic in that sense. So we've also, in addition to Mutex2, We have a variety of hard filters and pass filters, which we'll be discussing in the next slide. But our ultimate goal is to start with hundreds of thousands of variants from our sample that are being identified, and then do these filters to get rid of the false positives that that we don't trust, and then get just a small handful of variants that we do trust that we can then look at in more detail. So after we've passed our variants through mutex 2, our second filter that we're going to be looking at is hard filtering. So here are some examples of hard filters that we can use. The filters are here on the first column, and on the second column we have what characteristics those filters are based off of. So for example, we can filter based on base quality, or we can filter based on fragment size or position. I'm not going to go over all of these in detail. I will note that all of these filters are explained in our mutex 2 documentation, which is available on the GATK website. So when it comes time for you to use these tools, you can go ahead and get that information and interpret it when that time comes. However, I think the important thing to note is that anytime that one of these filters is run on your sample, it is going to, in the output, is going to add information about that in the metadata of the VCF. And so just having it run through, just having a filter run on your sample is going to add a little tag in your headers that says that that particular filter was run, but that doesn't necessarily mean that that filter was successful in filtering anything out. It just means that it ran without error. I think it's just important to note that to keep that in mind when you're looking through your headers, and we'll look at an example of that in the demo section. I think it's also important to note that some of these filters are going to require you to invoke specific options in order for them to work properly. So if you are running your samples using these filters, then I encourage you to look at the documentation so that you know what it is that it's looking for. Otherwise, you may find yourself with some errors, or maybe it just won't be filtering properly at all. So along with filtering our variants using mutex 2 and hard filtering, we can also filter using model-based filtering. So with model-based filters, we will be reducing the false positive rate using machine learning. So that's the end goal here. So how does that work? Well, first of all, mutex 2 is going to process our raw variants, which are going to be passed on to filter mutex calls. And this is a tool that we have that's going to be doing the filtering itself. So from there, the hard filter, filter mutex calls is going to be filtering, it's going to be using hard filtering on the variants and then discarding the ones that it finds. So those algorithms are going to be calculating the error probability for the inputs that it's finding, and then that's going to be used to look for normal artifacts and strand bias artifacts. The algorithms will then be taking that information and then resubmitting it and recalculating the error probability, which will then be used to look for bias artifacts like it was doing before, and then back to recalculations, and it's going to be doing that through several iterations over and over again. So when the variants are finally chosen, they're going to be chosen in order to maximize the F-score, which is a statistical measure of the test accuracy. So the machine learning is going to be looking for the optimal error probability for each of those variants and discarding the ones that don't fit that criteria. So finally, the process is going to output the called variants along with filtered variants. And this is what you're going to end up getting when you pass it on to your next step in the pipeline, whatever that is. So just like before, here are some of our various model-based filters that we can use and what they do. And as with before, just because the header of your VCF lists one of these as a filter, it doesn't necessarily mean that that particular filter was successful in filtering anything out, just that it was used. So here are some annotations that you are likely to encounter after using hard and model-based filters on your samples. And these are the annotations that you're going to be able to find in the info field of your VCF headers. So all of the information about these tools and the filters is also available in our documentation, as I said. So for example, if you see like MMQ show up in your headers and you don't know what that is, then you can go do a search on our website and then find out that it's mapping quality and get more information about how to decipher that information. So finally, here we have some additional mutex 2 features. We know that everything works using a panel of normals, but it can also work using tumor-only mode. However, unfortunately, that means that your bacterial contamination might also be working as well. And so that's one of the reasons why it's better to have a panel of normals than to not. We can also joint call multi-samples over time for the same tumor, if you're interested in that. And then finally, we can also run mutex 2 on mitochondria, although this feature is currently in beta, and so use at your own risk. So that's it for the presentation portion. And now we can move on to our demo, which I think is the exciting part. Are there any questions so far? Yes. Yes. So my gut tells me that you wouldn't be able to differentiate it so easily because you're using the normal as the control. Do we have anything that would help differentiate in that case, or is that just a case of getting more, like getting better normals?
Things that you find.
Yeah, that is one of the steps that we run anyway. We use that tool in the demo, for example, but I don't know how well it would work if you have tumor contamination in your normal samples. Yep. I think there's another question there. So is it possible to do what with your tumor and normal samples? Sorry, I didn't hear.
Yeah, like.
Oh, so you mean, but they should be separate samples. What do you mean by deconvolute them? Maybe I'm misunderstanding, or maybe. Oh, I see. Is there a way to do that? I don't know. No, I'm sorry.
Yeah.
Okay. Well, then I think that we can move on to the demo section. So please bear with me while I shift gears. Oh no, I don't need to because I already I'm ready. So, okay, why don't we all open up our Terra profiles again? So go to app.terra.bio on your machines, whichever one you were using for the previous exercises. And then what we are going to do is go into view examples. So we are going to be using our Mutex2 featured workspace, which is available here in the featured workspaces section. Let me just double check that I'm logged in. I am. And just do a search for Mutex2. And it should be the one at the top called Somatic SNVs, Indels, GATK4. So just click on that and make sure that the workspace that you open up looks like mine or has the same title as mine. If you have any issues, please don't hesitate to put a pink sticky up and we can get one of our other instructors to help you. So as with our other exercises, why don't we go ahead and clone this workspace? I will clone it, and you can go ahead and give it a unique name that you can find later. I'll call it Derek. Oh, sorry, it's called Mutex2. Just type in Mutex2 in the search bar of the featured workspaces, and it should you should be able to find it spelled like this. And just make sure that your workspace has the same title as this one, because there are several workspaces that are for Mutex2. So it should say GATK best practices for somatic SNVs and indels using Mutex2. Just want to make sure that you're doing the right exercise. So yes, I'll go ahead and clone and go back to putting my name in here. Don't use my title. I called dibs. And go ahead and use your, what is it? Workshop GATK 2304 is the billing, and just clone it. Okay, so we are all set. And we should all be project owners of our versions. So as with our other exercises, we have all of our information about what this workspace actually does on the dashboard. So if you go here, it should give an explanation of the data that we're going to be using, resources that you can use. There's a forum article here with information about tutorial and just a lot of in-depth information, the likes of which I was covering earlier. But we're not going to go over any of that. What we're going to do instead is go to the analysis tab and then make sure that we have our notebook ready. I guess while we're doing that, maybe this may take a few minutes. One thing that we are going to do as part of this demo is to use IGV again, like we did yesterday. So I guess we could take this as an opportunity to open up IGV on our respective machines and make sure that you are logged in to your account as we were able to do last year, into your Google account, the one that you use for logging into Terra. So if you don't remember how to do that, first of all, make sure that at your menu, you have this Google menu bar item. If you don't have that, you're going to need to go over to View and then Preferences. And then in View and Preferences, you will have to click on this checkbox that says Enable Google Access. And then once you do that, this little menu should pop up and allow you to log into IGV. So we will do login. When you do log in, just make sure that you have a, you've checked this box, view your data in Google Cloud Storage as we did yesterday. Otherwise, you will find yourself with some errors, and then click on continue. And so it just says, okay. So if I go back to my IGV, then I am logged in. And that's essentially all that we have to do. So I'm just going to refresh this page, or maybe go to my data page and go back. I'm still waiting. Great. Am I the only one waiting here, or how many people have the notebook already loaded in the analysis tab? Everybody's waiting? Okay. Okay. We'll give it a little bit more time. All right. I've got mine. Just refresh your page and it should show up after a few minutes. And once you have that notebook, go ahead and click it and make it open. And this will be our exercise that we do. So again, we have all the information here that we're going to be running through as part of this demo. It should be Pretty self-explanatory. Each of the steps are explained in a lot of detail. Actually, for that reason, I like this this tutorial. I think it's very well explained. But you can see here that we have our Mutex 2 workflow that we were going over in the previous presentations, and this is what we're going to be doing today. However, in order to run this demo, we are going to need to spin up our environment, our Jupyter environment. So as we've been doing before, go ahead and click on that side bar on the little cloud icon, and then click on settings, and then create new environment. And from here, we're going to need to make a change. I'm going to double check. It actually says here what we should do. Just make sure that your settings align with this one. I think the only thing that we need to change is our CPU to 4. So once we do that, just click on this, and then go to CPUs and change it to 4. And then create. Am I going too fast? I just want to make sure that everybody's in the notebook at this stage. Who is not in the notebook? Okay. You're still waiting? Okay. Okay. For the rest of you, just see if you can start up your Jupyter environments. It should take a little bit of time regardless. So I think that we should just be patient. It was a lot faster for me when I was doing this demo by myself. And I think that's because I didn't have 50 people trying to do the same thing at the same time. So let's be patient. So I guess while we're waiting, I might as well just start explaining the next steps that we're going to do, so we don't actually have to wait for them to load when we are ready. The first thing that we're going to be doing as part of this demo is just setting up our notebook environment. So this isn't this is necessary only if you're running GATK from within the notebook, but if you're outside, if you're not doing this analysis inside of a notebook, you would obviously need to be setting up your directories on your own. But that's essentially what we're doing here. So as we've shown before, if I click on this, I'm going to open this up in a separate tab. But if you remember, our data is stored in this workflow, or sorry, in this workspace in the data tab. And down here, we have workspace data. There's a lot of information here that we'll be able to access using our analysis, our notebook. And then we also have additional files in here, including the files that are going to be saved by the notebook itself. So in the workspace data section, we've got our panel of normal, an index for our panel of normals, along with the panel of normals itself. We also have an intervals list and an index that we got from Nomad. You remember Nomad from the presentation yesterday. And we will also be getting access to some BAM files that we're going to be downloading. So I'll return to the workspace. My cloud environment is ready, so I'll wait for the rest of you to catch up. But in the meantime, I'll just start explaining what we're going to be doing. So the first thing that we need to do is set up our workspace bucket variable for this particular notebook. So this is going to, I'll just enter through this and it will do it on its own. The next thing that we need to do is to set the workspace variable to have access to the most recent materials that we're going to give it access to. And then finally, we need to create all the directories for our files to live inside of this notebook. So those directories are going to be housed locally inside of that data tab that I just showed you. And when your cloud environment is ready, as with our other examples, you just need to be able to click in here and you can click Shift and then return, or Shift and enter, and it should just run that. I'm sorry, I skipped a step. You should click open, and then you'll actually be able to run these steps. Okay, here we are. Right, so when you're ready, you can just click on one of these and press Shift and enter, and it will go into the next step. Okay, and just for future knowledge, when you are starting one of these notebooks, you can always check to make sure that you're in the right environment or the right kernel environment by looking at the logo that we have up at the corner. In this case, we are supposed to be in Python 3, and so it tells us that we're in Python and that we also have a little Python logo. However, if you are running a notebook that needs another environment, like an R environment, you can always just change it by going to kernel and then selecting the one that you want to give it access to. Great. So I have been able to create these additional directories. In fact, if I go to the data tab, I should be able to see those directories as they've been generated down here. They should be saved someplace. Well, it's not important. I'm going off script. I'm a madman. Okay. So the next thing that we're going to be doing is to check the data permissions. And so for this tutorial, we're going to have everything in the Google bucket as we've been doing. And so the first thing is we need to make sure that our data is actually accessible to the notebook. And so we're going to run this gsutil command, and then it should be able to tell us if there are any errors. And it will tell us where that is. So here it's giving us a list of the data that actually has access to. This is going to include our BAMs and our CNA inputs, also some of the pre-computed data that we have, our references, and some other resources. And so if you do run this and you do find yourself having an error, we have an additional step here that you can just uncomment, and then it will allow you to install Google Cloud Storage, and you can try that again. You'll have to restart the notebook, but or restart the kernel, but it should work after that. But hopefully, I mean, things are working, at least for now. The next step that we need to do is to download our data directly to the notebook. So we will just run this. These are some more gsutil commands that we're going to run. And there we go. This will take a little bit of time, but we'll be able to tell when it's ready because the command here will turn from red to yellow. And our next step is to set up IGB. Fortunately, we have already done that. However, while this is loading, we can return to IGV since we have a little bit of time, and just make sure that we are on the same reference genome. And the default, at least for me, is HG38. This is what we need to use for this particular demo. So I know that yesterday, I believe we were on HG37. So if you're still on HG37 or you swapped over to a different default, I'd recommend that you look for HG38 because that is what we are going to be using. Right. How are we doing? Are we good, or are we getting errors? Good. Excellent. Okay, so while this is going, I can review the next steps. So that's going to be the end of our notebook setup, which is the boring step of the tutorial. After that, we can actually start running our samples on Mutex2. So I'm not going to press enter on any of these until this is actually completed. So I'll wait until this turns yellow to actually hit anything, but I can at the very least go over what these commands are going to be doing. So here we're actually going to be calling our Somatic SNVs and indels and generate a BAM out file that we're going to use later. But as we can see, here is our GATK command using Mutex2. We're going to be inputting our reference assembly. This is a FASTA file that we have given access to from within the notebook. We are also going to give it two inputs. One is going to be our tumor BAM file, which is here, and the other is going to be our normal BAM file, which is here. And it does seem that this has finished copying for me, and it completed without any issues. So at the very least, I can continue. Hopefully yours will be able to finish soon. So I'll continue. So we have our reference, we have our inputs, and we also are going to give it access to a panel of normals. This is one of the files that we gave it access to through the resources that we downloaded and copied into our notebook. And then in addition to that, we are going to give it access to our interval list. And so this, in addition to the panel of normals, is going to help us properly filter out what we don't want to carry over into the VCF. And then finally, we've got our output here, which is what we're going to be generating, along with this BAM out, which will be carrying over into a later stage of the analysis. So I'll go ahead and press enter here and let it do its thing. Yes, of course. Oh, yes. Okay, so in this particular example, we, just for time, we're only going to be looking at this particular region in chromosome 17. I should have explained that before, so good catch. But if you are going to be looking at your particular samples, then yes, of course, you can run the entire thing. We're just doing this because these are large files, and if we're just looking at the section, we'll actually be able to see the samples in IGV like almost immediately, whereas if we did the whole genome, it might be here for a few hours. But good question, thank you. So of course, if you have access to a panel of normals and the interval list, then we would recommend that you use it to get the best filtration and results at the end. However, that's not always possible. So as we were describing before, it is possible to run Mutex 2 and this pipeline without a panel of normals or without the interval list, but it won't, you'll end up with a lot more variance at the end of the day. And so you may have to do additional filtering either manually or using some other tools outside of this workflow to help you narrow down. You might end up with, several thousand variants at the end of this.
Like some documentation about generate.
Actually, I'll show you how to do it from within this workflow. So that's going to be one of our stages I can show, or one of our steps. I can show you how to do that once we are ready to move on. The interval list is going to, what's the technical, like what is inside of the interval list? What's it looking for? Okay. So it's essentially like a bed file. It has positional data that the great. I saw a hand up here. Yes, question. Can you have more than one normal? Yes, well, if you have multiple normals, you can put them together as a panel of normals. So that would be part of a step that I'm going to cover in a bit. You could also run the pipeline multiple times comparing. Yeah, could you make it sequential, like run the analysis multiple times using different normals and do a comparison that way, even on the same tumor? Yeah, of course you could do that. But you would end up with multiple VCF files that may have different results because they're being based off of different normals. I mean, they're being compared against different normals. So they may differ a little bit, but it depends on your use case. Yes. I suppose so. Off the top of my head, I don't know why not. What are the?
All right, if you have a normal file with two replicates in it, but they're different sampling, could you then just put both of them?
Yeah, I think if you've got BCF and SNVs. Yeah, you can use that as well. You're sure.
But that would be a VCF though. Could you include two BAMs as an input? As from like two different normals? Was that your question? Yes. So two different BAMs, two different normals. Yeah.
That's two different.
Yes. Yeah. Right. As long as the regroups information is maintained, then you would be able to separate it out in the end. Okay. Okay, at this point, I think that we should. Oh, sorry, were there other questions? I didn't remember if I saw another hand. Okay, so moving on. Also, okay, so talking more about the inclusion of a matched normal control. There's a very good example here in this workshop, in this workflow, where you can actually see the difference. So here, I'm going to zoom this in so we can all see it. But here's an example of mutex2 calling calls with a panel of normal and also one without, or sorry, with a matched normal and without a matched normal. And what you can see is that there's a stark difference in the amount of variants that are called. So without a matched normal, it ends up with something like 50,000 variants at the end of this pipeline. Whereas if you included a matched normal, you would end up with something closer to 5,000 variants. And so 5,000 is a lot more manageable to look through. Whereas if you don't have a matched normal, that's way too many. You would require additional filtration steps at the end of that. Yes, question.
Sorry. Will be the case that a variant reporting in Newman,
Okay.
or the panel normal, which is like a recurrent mutation in the population, driver mutation, something like this, to be excluded if we are going to use the Newman as panel of normal, or when it's a normal tumor normal area.
Okay, so you're if I can rephrase, the question is it possible to exclude variants that occur naturally within the normal sample from the tumor sample?
If I'm correct, the Newman database also includes some variants already reported in cosmic as I don't remember exactly, driver mutation or recurrent mutation, like in RNA-seq. And if these variants are inside Newman, the Newman that we are using,
yes,
and we are using the Newman as a panel of normal,
exactly.
Is it possible to lose them, or is there a way to avoid this possible?
So long as it's not if it's included in the normal samples, then it should be able to be filtered out. If it's inside of the if it's in both the tumor and the normal, it would get filtered out because it's being found in both locations. Whereas if it's in only the tumor, then regardless of what nomad is going to be showing, then it should show up as one of the potential variants.
Is there a way to avoid if it is a germline driver mutation, something like this?
So you're interested in keeping a particular set of variants in the tumor sample that you know is.
Yes, to have the output with an annotation that say, okay, it's present in the panel normal, but when I do the functional annotation and I see it at the cosmic ID.
Yes, you should be able to, at the very least, merge that information back into your output after running through this workflow. I don't know if running this tool itself, if you can dictate specific areas that you're interested in keeping, aside from just running this on, I mean, you can give it, for example, BAM files that don't include those particular regions, and then those would be excluded from the analysis, which then you could take those VCFs that you've done before and then merge them back into the final VCF. So you would have that as well. Then you could run the filtration steps on that information.
It's possible from the bed file.
Yes.
To filter the bed file to exclude the no mutation reporting cosmic.
I believe so. I mean, so yeah, in that way. So I mean, if you use it wouldn't be the bed file, it would be the interval list, but in that case, it would be, I mean, you could filter that out if I'm understanding correctly. We can get you, I mean,
yes, in the panel of normal to exclude this variant.
Yeah, if I'm understanding correctly, yes, I think that you can do that, but we can get back to you with more information later.
Yes, sorry.
Okay, great. So moving right along. So to return to, I believe it was your question about how do we make a panel of normals. Fortunately, Mutex2 includes argument that we can use in order to make one, and there in our instructions here. We are not going to make a panel of normals as part of this demo, simply because it takes a very long time. And so we already have a panel of normals that we are including. However, there are instructions on how to include or how to make your own panel of normals using the following codes here. So the first thing you would want to do is run Mutex2 in tumor only mode for each BAM individually, each of your normal BAMs individually. And then you can see here that we're running this sequentially. So we're running this Mutex2 and outputting a normal 1 VCF and then a normal 2 VCF, et cetera, until we have normal 40 VCF in this particular case. We're going to have 40 samples in this example. And then we're going to create a genomics DB database from the normal Mutex2 calls using this argument. We're going to use a genomics DB import is the tool that we'll be using. We'll give it access to a reference FASTA, as well as an interval list of areas that we're interested in. And then we will point it towards all of the normal VCFs that we generated in the previous section. So 1 through 40. And we'll do that. We'll give it access to all of those normal files. And then finally, we're going to combine all of those normal calls using the tool create somatic panel of normals. I hope you can see that. And yes, we'll just be running this command, putting all of that information together, and then outputting a panel of normals file. So that is something that we would be doing before actually running Mutex2. I mean, we could do it before and then do it after, but if we want to include a panel of normals, that would be a good way to do it. So after we're done with that, after we've finished filtering, calling our somatic SNPs and indels with Mutex2, then the next step, as we were discussing, is actually doing the filtration and filtering for our confident somatic calls. So the first thing we're going to be doing as part of this is to use get pileup summary and calculate contamination. Calculate contamination is the tool that Mehrtash was bringing up earlier during the questions. So first, what we're going to do is use get pileup summaries to summarize our read support for a set number of known variant sites. So in this case, we're going to be using a human population germline resource, Nomad, which we were learning about yesterday. And we're going to be running the tool on the tumor and on the normal samples. And we're going to be doing that separately. So first, we're going to be using get pileup summaries on our tumor samples, inputting our tumor BAM, and then running it on this particular section of chromosome 17. And then we're going to be outputting get pileup summaries table at the end of this, but only for our tumor. So I'll go ahead and press enter on that. And then while that's happening, or it's already no, it is happening. Then we can go ahead and do the same thing, but for normal. So the code that we're using here is exactly the same, except instead of the tumor BAM, we're using the normal BAM, and instead of naming our output as a tumor get pileup summaries, we're going to name it a normal get pileup summaries. So I'll go ahead and press enter on that. So when it's ready, it's ready. Okay, I believe that it should be done by now. And so each of these the commands that we have here below, this head command is going to produce a 6-column table. So I can it's going to be pulling some information off of the files that we generated, the get pileup summaries table. So I'll just press enter, and then I'll press enter on the other one, or shift enter in this case. And you can see that it's generated a table with 6 columns. The formatting is a little off due to the sizes, but what we're interested in looking at is the allele frequency section. And so we can compare the last column between our tumor get pileup summaries table and our normal get pileup summaries table. And we can use that to refer to the reads that are going to support all the other alleles. And I believe there is, okay, so for us, we are basically going to be targeting ranges that are between 0.01 and 0.2. So if we just look at this, we can see that it's within that range. So that is what we should expect. So things are working as normal, as we've described here. So then after this, we are going to use our calculate contamination tool. So this is the tool that we're going to use, GATK. Yes. How do we adjust it? That should be able to be changed in one of our arguments. But I don't know off the top of my head, how do we change which tool would we use if we wanted to change the population resource ranges that we would end up with at the end of the get pilot summaries? Is there a way to change that? If we wanted to make the so at this stage where we are trying to look at homozygous variant sites, could we be more or less stringent based on the parameters that we're using, or how would we adjust the parameters at that stage? Or is this something that would just come about as part of our subsequent filtration steps? This is after we have done Mutect, but the question is, where in Mutect, what argument would we use if we wanted to make it more permissive or less permissive? Okay. Yeah, so I guess ultimately the answer is look at the documentation for Mutex 2. I know that has a lot of different arguments. We didn't go through all of them. We only used a handful to get the basic outputs here. But if you look at the technical documentation on the GATK website, it shows every single argument that we have, and you could probably find the one that would help you. There are explanations for each option that you can include. So continuing on, we're going to run calculate contamination so that we can get an estimation that is going to inform downstream filtering when we use our filter mutex calls. So I'll just enter this now. I don't need to go into detail. I thought that there was a question. Was there a question over here, or just stretching? Okay, that's okay. So this is we can use this to determine how much contamination there is between our samples. So contamination of a tumor sample into the normal sample, the tumor sample into the normal sample. So we should be able to exclude that from our end result.
This is due to something like something.
Yes, just because sometimes it's difficult to separate samples if you're taking a sample from an area that has a tumor and like a biopsy, for example, and might have a normal sample in there. So
if you're taking two separate samples instead, so rather than skin, for example, you need to run.
Yeah, in theory, you wouldn't need to. Okay, so that should be done for me, and then I'll continue with here's an example where we're going to generate. This is a site. We're going to go on a little side here because we can use an additional feature of the tool, which is to provide both the tumor and the matched normal pilot tables separately, and so that may allow for us to have a slightly more accurate estimate. So what we can do is just change our settings in the original calculate contamination sample. I'll just go ahead and run that, and it should be able to create a table very quickly. Yes. You're comparing it to the normal, and it's going to be comparing them and then finding out or trying to determine how much contamination there is between those samples. So So after we have created our contamination table, then we can go ahead and export both of these, or open up both of these tables. We're going to take a section out of both of them, and then we're going to be able to see, compare our contamination and our error, in one case using a matched estimate, and the other not using the matched estimate. And so you can see here in our one version where we're only using, only looking at the tumor sample, we've got a contamination rate of 0.019 with an error of 0.0023, and we can compare these numbers to when we are including a matched estimate, in which case the contamination is 0.0115 with an error rate of 0.0219. So that's a lot of numbers, but we can just see that it is the contamination rate is less when we're using the pair, and then it's we also have a lower error rate as well. So it's just something that you should be aware of when you're choosing how you're going to be setting this up. Okay, continuing on, we're going to apply some filters using filter mutex calls. So here we're just going to hit enter on this. We're going to give it access to our, we're going to be using the annotations that are already within the call set if we've provided that. And it's also going to use the contamination table that we were creating in the previous steps. And so here we are with just the default settings that we have, and we're going to try and output our somatic filtered file. And at the end of all this, it should be able to, it's already finished, and it should be giving us our filtered file, the one that we can use for analysis. So now that we've had everything finished, what we can do is grep out all of the headers that have a filter that's been used. And this is what I was saying, where if we look at the filters that are used, it tells us exactly where things failed or passed. So we have a filter here, it passed. It says that the site contains at least one allele that passes filters. This is obviously a condensed version of the header. I'm just pulling the information out, but here you can see what passed and what failed, and it gives you information about that. But as I was saying before, just because a particular filter shows up in here doesn't necessarily mean that it did any filtration. Just because it passed doesn't mean that it was able to exclude any additional variance. It just means that it was able to be run without having an error. Obviously, if there was an error, then it would show up and you would be able to identify that. So I want to continue with actually taking this information and putting it into IGV so that we can take a look at it in a little bit more detail. So by this point, we should already have set up IGV, which means that we can go ahead and use our gsutil argument here to give us some information that we can use. Yes. Yes.
Basically a set of parameters that are already built in.
Yes. Filter mutex calls. Yes, it has. It will allow you to run all of the filters that we were discussing during the presentation, the hard filters, the
best bulk filters.
I believe that in the default, it's going to give you a subset of the filters that it is able to run. If you wanted to choose specific filters, then you would need to look at the documentation to find which argument you want to use to exclude or include. So, okay. Great. So continuing on with our IGV, I'm going to make sure that we have HG38 loaded as our reference genome, and then we are going to I'm going to click on this, and it's going to give us our list of files that we're going to want to input. Is the tumor files and the normal BAMs that we're going to include. So what we're going to do is use in IGV, go to file and then load from URL, and then input one at a time these 4 samples. So go ahead and click copy, go to here, file and load from URL. Move this so it's closer, and then paste. Press OK. That is going to be our filtered VCF that we created in the previous step. I'll go ahead and copy our tumor normal BAM, and then do the same thing. File, load from URL. Press OK. Continue on. And then the last one. And at this point, we can navigate to the location where our variants are. If you remember, we are not using the entire genome for this example. We're only using a particular region that is located in chromosome 17. The location that we used in this example is included here. It's this gene TP53 to locus, and so we can, that's the location of the locus. We can go ahead and just copy this and paste it into the viewer so that we can go to that region. And so here we are. You'll see that we've got our read showing up. We've got everything. We've got our tumor BAM file, our normal BAM file, and then we should have our VCF here as well, the one at the top. So if I zoom in, we should get a little bit more of an idea of what we're looking at. And there you can see that we have our, I'm going to scroll around and look at our information, but we can see that there's a variant that's appearing over here. So I'm not going to go into this into too much detail. We can play with this detail or with this data on our own, but later we can, if you want, you can revisit this and look at information about what types of variants are showing in this particular example. And there's an explanation in the notebook of what's going on so that you can get a better idea of how to interpret your own results when it comes time for you to do that. So if you remember, after we've finished with our running Mutex2 and then filtering our samples after running Mutex2, then we can do a little bit of annotation using Funcotator. Funcotator is a tool that lets us annotate our SNPs and indels. And so we'll just be running that right here. And there is, and the output is it's going to generate is this VCF file that we have right here. I should note that after using Funcotator, we have a choice of either outputting our files as a VCF. We can also output as a MAF. I think it was written down here, MAF format, which is a mutation annotation format. It's an older format. I think people like to use VCF more, but if you have a tool that you want to use downstream, then all you would need to do is take this code and then replace this with MAF instead of VCF. And you should be able to get a file in the format that you need. So at this point, we can examine the annotations of our particular mutation that we've been looking at in this demo, and we can just rip out the regions in chromosome 17 that we're interested in. This is just, it's pulling information from our VCF file, our Funcotator file, that's in chromosome 17, and then we're also getting information at this particular region 7674220. And so what we're going to be looking at here is, well, what we can notice immediately is that in this region, there's a lot of missense mutations. So there's missense. You can see it all over the place. And if you look closely, you can see that a lot of these missense regions are here with this R turning into a Q. So that's a glutamine missense mutation, or sorry, it's an arginine to glutamine missense mutation. So that's one example, but you can see that it's showing up in a variety of regions here. So yeah, so it's annotating this for us so we can then interpret it later. And then the last thing that we want to do is, before ending the demo, is going ahead and counting our total mutation records that we have. So when we began, we had well, let's just go ahead and take a look at all of our mutation records. I'll go ahead and run this, which is a count of the number of missense records that we have, and then finally a count of the number of missense records that actually pass the filters that we had. And so what you'll see is that we start out with about 124 samples in this particular region. It goes down to 21 when we're just looking at the missense records that we have identified here. And then if we're looking at the ones that only pass our filters, then it goes down to 10. So we start with 124 in that region and it goes down to 10. And so as you can see, these 10 are the ones that we should be most confident in, but they can definitely, and so if you're going to be looking at these variants in detail, it's a lot better to just have 10 that you can look at in detail as opposed to 124 that you might need to have a little bit more confidence in, making sure that they're not, false positives before moving forward. So there's a little bit more information here if you want, but that's it for the demo. So are there any questions? We've been taking questions as we come, but are there any questions before we move on? Fantastic. Okay, I think that we'll take a 10-minute break right now, and then we'll rejoin here at 3:05 so that Mehrtash can give his talk. You need to? Oh, sure. Actually, can you do it now and then? Okay, so let's not go on break right now. Mehrtash is gonna, yes. Hold it in. If you need to use the bathroom, hold it in for just a few moments, and then silently express your anger with your eyes at Mehrtash. Oh, and I will give you.
Can you hear me? Oh, yeah. All right. So, oh, that's my little friend. Okay, so this is something we're going to need for the exercise later, but it's going to take some time. So let's just run it in the background. So the idea is to go to the workspace that you should have a copy of because you've made it before. It's oh, not this one. Oh, yeah. All right. So it should start with like 2023 Cambridge workshop, GATK4 blah, 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 copy. Just go and find it in your workspaces. Like here's like how you would do it. You would need to go and here. Well, I mean, you can first like search by keyword. Just maybe just put 2023 Cambridge, and then just make sure you find your own email. I don't know if you see all of that. I do. Yeah. Yeah, make sure you're working with your own copy. Okay, once you located that, go to analyses and select 08 GATK Somatic CNV tutorial. And you should see this guy. Is everybody with me? Anybody who's okay? Cool. Now click on open. If you've been like, it might take a couple of minutes for you. Okay. Whose VM was already running and who's getting the message they need to wait for 2 minutes before it runs? 2 minutes, 2 minutes. Okay, yeah. All right. So let's just give it to it. But I'll tell you what's happening here. So we have a Docker image for GATK, and then there's another Docker image for running GATK in a notebook mode. Something broke sometime, and we forgot to fix it. So it made some of the GATK tools that uses Python not to work properly with the current Docker image in the notebook environment, but it's a very easy fix. What we're gonna do is that we're just gonna, once your VM is up and running, we're gonna copy this set of bash commands and run it in a terminal. What it would do is that it would create a conda environment with the GATK Python packages already installed in it. And then that will allow you to use GATK, certain tools in the GATK that uses Python properly in the notebook mode. Are you guys familiar with conda and Python and those types of things? If you're not, here's a 30-second version of it. So Python is, there's this thing called Anaconda, which is a package management system for Python and allows you to make environments, and each environment has a set number of Python packages installed in it. GATK requires certain Python packages to be installed for certain GATK tools to work. So what we're going by, what will happen by running this set of commands is that we will create that Anaconda environment inside the virtual machine that runs your notebook. Okay. All right, can you give me a show of hands if your VM is still trying to restart? So your VM is already running? All right. So if you're all good, if your notebook has already opened, then what you need to do is that you need to copy the contents of this cell 1.2. Just copy the whole thing and click on this terminal icon on the right side panel, and it will open a Unix terminal inside your virtual machine, and then paste all of that. Okay. So this is the part that takes about 7 or 8 minutes to complete. If you're there, I guess just feel free to take a break and come back to it. All right, we're gonna start the presentation in 7 minutes, 3:10. Okay, take a break. All right, how are we doing? I'm now at the point that it's installing the IPy kernel. Hopefully you're around the same place. All right, so this is another important step. Make sure you press enter because the very last command just for some reason just doesn't run automatically. So return, and we should be done. If everything has proceeded successfully, you should get something like that. It should say GATK. If you type conda-env list, you should now see this new GATK conda environment. Who hasn't reached that point yet? But it's going, yeah. And yours is lovely. Yeah. All right. Once all that is done, let's just click on this cloud thunderstorm thingy, and then click on settings on Jupyter, and increase that auto pause minutes of inactivity to 120 minutes because we don't want this thing to get paused. Otherwise, it would just take another couple of minutes. And for some reason, you lose all that configuration. You have to just do all that again. So just increase it to like 120 minutes so that you're good for today. Once you increase that, just go all the way down and press update. Okay. Final thing, go back to your Jupyter notebook and refresh it. Just command refresh if you're on a Mac. For Windows, Control+R, just refresh it. And something magical should have happened now in your Jupyter notebook. If you go to kernel and change kernel, you should now have this additional conda-env:/GATK. Did you get it? Cool. Make sure you select conda-env:/GATK, but not GATK, not the very last thing at the bottom. This guy. Select it, and then go to kernel, and then restart. And now you should have a GATK conda kernel active. Perfect. So I'll check back on all the steps before we start doing the exercise. So let's just go back to the presentation mode. Beautiful. So today we wanted to talk about somatic copy number alterations, but we decided we'd do something much cooler because somatic copy number variation is boring. So I'm very excited to tell you about something that we've been working on for a couple of years now. It's called ChatGTK. It's the first AI-powered conversational variant caller. So why? What is ChatGTK? And why now? Well, as you know, there are just too many bioinformatics tools, right? So we have like so many genome aligners and so many variant callers, and it's just difficult to use them, right? So the other problem, like a more practical aspect of this, is that the command line arguments are just doubling approximately every 8 months. So they're just like Moore's law. Like GATK1 was here, GATK2 is here, and now with GATK4, we have like super long command lines. It just gets more and more difficult to memorize. So here's what we did with ChatGTK. We trained a large language model on about 2,500 different bioinformatics tools and 5 whole million human genomes. It's a gargantuan model. It has about 800 trillion model parameters. We trained it for 6 months on 8,000 GPUs, and it cost us about $8 million to train. And then at the data sciences platform, we fine-tuned it using this technique known as supervised fine-tuning. So the way it works, if you're not familiar with it, is that the user inputs a prompt to ChatGTK, And then ChatGTK produces several plausible variant calls. And then ChatGTK gets a reward if it produces high quality variant calls. And this is how we basically just train this model. So this is still a closed beta. We expect a public release in Q3 2023 as a Terra featured workspace. So here's how it works. So it has a very simple command line interface. So ChatGTK, you give it a prompt, like, hi, ChatGTK, could you please something? And then your sample BAM file, and you get your variant calls. So the cool thing about this is that it's such a powerful model, and the variant calls are so high quality, you don't even need to perform any variant filtering. All right. So you can specifically ask for that. You can say, hi, ChatGTK, could you please produce the best possible variant calls for my sample? Aim for genotyping quality greater than 5,000 and beyond. So, you just don't need to do any filtering. It also supports different file formats, including images. Like say, hey, bro, we did generate an image of the person based on the provided BAM file. And here I got like Jonn Smith's genome. And it's very easy. You get this? Oh, something went wrong. It says that, oh, did you just call me bro? How about you try again? Ask nicely this time. So, all right. So we asked nicely, hi, ChatGTK, could you please generate an image of the person based on their provided BAM file? Thank you, you're the best conversational variant caller, heart, heart. And, oh, I'm not sure what happened. This is John Bon Jovi, not Jonn Smith, but I'm pretty sure this morning it worked. All right, so maybe it's actually not ready for prime time. So we'll go back to the original topic, which is somatic copy number alterations. Sometime soon, not today. All right. So let's just talk about CNVs instead. So this is like a historical perspective of copy number variation tool development. I'm going to tell you more about what is a copy number variation, but I think at this point, you probably have a good idea of what they are from all the introductory slides. So here's a very GATK-centric history of that. So we started with some ancestral tools. These are tools that were also developed at the Broad. XHMM was the germline copy number variant caller. CAPSEC and ReCAPSEC and allelic CAPSEC and absolutes. These were like somatic copy number variant callers. In GATK4, we basically ported them, some of them verbatim, some of them slightly improved version of them. And then in GATK4 final release, here's where we are. We have two different workflows. One of them is called gCNV in short, which is for calling germline copy number variant events, both in whole exome and whole genome sequencing data. In principle, it works for both like common variation and rare variation, but the default parameters that we've provided, they're tuned for rare events. For somatic copy number variation, there's another workflow which is called model segments, and it also works for both whole exome sequencing and whole genome sequencing data. And today, we're going to be talking about model segments workflow. Tomorrow, we'll cover gCNV workflow. Okay. So what is somatic copy number alteration or copy number variation? These are completely synonymous, by the way. Sometimes you hear CNV, sometimes you hear CNA, copy number alteration, copy number variation, same thing. So as the name suggests, these events involve some type of a structural variation of the genome that involves loss or gain of large chunks of the genome. Yeah. And here's a very cool assay, experimental assay that you can actually visualize copy number variation. Here on the left, you see normal cells. On the right, you see ACC 1940-54, which is a cancer cell line. This technique is called spectral karyotyping. The way it works is that you have probes that just go and hybridize with different chromosomes. And then you attach fluorophores to each of those probes so that when you do fluorescence microscopy, you see different chromosomes lighting up in unique colors. So you should basically get 24 different unique colors that allows you to visualize your chromosomes. Here's how it looks for a normal sample. On a cancer sample, it looks crazy. Like you can see that, for example, chromosome 1, it has like an extra half an arm here, like half an arm here, some chunk of chromosome probably 19 or X, or maybe actually 11. I'm a little bit colorblind. Is attached there. You can see all types of like, structural rearrangements of the genetic material between the chromosomes. But the most important thing for us here is that you see like loss or gain of big chunks of the DNA. So this is called somatic copy number alteration. And it can be very, very dramatic, involve like, half of a chromosome, something like that. And often they are like hallmarks of cancer. And different cancers actually have different copy number variation signatures, for reasons that I will tell you in a second. So if you look at what kind of genomic regions tend to be amplified in cancer and what kind of genomic regions tend to be deleted in cancer, you see this type of pattern emerging that many of the genes involved in cell proliferation, in cell growth, are both receptors and ligands. Those are the genomic regions that tend to be amplified in cancer, because that gives like a gain, like an evolutionary gain basically for the cancer cells, because it can just like replicate and grow faster, right? And then the regions that tend to get deleted, these are basically regulatory genes, mostly like tumor suppressor genes, like BRCA1, BRCA2, p53, NF1, NF2. These are like well-known regulatory and tumor suppressive genes that are involved in like cell apoptosis, things like that, right? So it basically tends to, what copy number alteration does for cancer cells is that it basically makes them very aggressive. Just grow as fast as you can and just make sure you don't die. So now that's kind of like why we care about them in the context of cancer. But we also care about copy number variation in the context of germline, like germ cells, like which all of us are endowed with many copy number variation events when we're born. And those are also important, and those are also linked with diseases for the reason that many of the genes are either haploinsufficient or triplet sensitive, which means that one copy is not enough and 3 copies is too much, because those genes are like so, acutely need to be regulated that you just cannot tolerate a gain or loss of a copy. And often copy number variation at the level of germline, if the fetus survives, and what we often see is that leads to rare diseases. Like here we have like 2 examples. Deletion of a locus in chromosome 22 causes this DeGeorge syndrome, developmental delay, heart defect, and other things. And a duplication of a nearby locus, leads to Schmidt-Fraccaro syndrome, which is a hallmark of which is like down-slanted eyes, and again, like a number of other things. So, so basically, we care about copy number variation in both disease and also just the germline contexts. Okay, so how do we identify copy number alteration? So let's make this sort of working hypothesis for now. Let's imagine that you have your genome, and if you just review how next generation sequencing works, is that you chop your genome into many, many small pieces, and then you just randomly sample them and sequence them, right? Let's imagine that there is no bias. Like basically every genomic fragment has equal chance of being sequenced, yeah? So let's say that you just collect all of these genomic fragments and align them to the reference genome. So what will happen is that a region that is unaltered, let's say you just have the same usual 2 copies deployed. Let's say you get 2 units of coverage, but the region that let's say has gained an extra copy, you might get more coverage, right? Because more initial genomic material would imply more number of genomic fragments sampled from those regions. And once you basically align them to the reference genome, you just see like more reads aligned to that reference genome. So that's the main idea. Like, copy number variants cause coverage imbalance. But I made a very terrible assumption. The assumption that I made was that genome sequencing is unbiased. It's not unbiased. In fact, it's a very, very biased procedure. You get very little reads from telomeric and centromeric regions. You get very little reads from regions that are rich in Gs and Cs. And in the case of whole exome sequencing, by design, you get only reads from the regions that you have selectively enriched for sequencing, and those are the exons, or the coding regions. And here's like an example. These are like IGV screenshots. You can see this coverage tracks, and you can see that basically, these are two different whole exome capture kits, and you can see this basically no coverage outside of these two exons, basically here and here, and then a bunch of coverage when you do have an exon. But interestingly, if you just compare kit 1 to kit 2, you see that this kit 2 actually just has a tendency to get more reads from this exon. Yeah. So if you're treating this in a naive way without correcting for these types of biases, you might erroneously conclude that this exon is amplified in relation to this exon, or the other way around. Maybe you conclude that the left exon is actually has lost one copy. So it's very, very important that we take into account these biases that might exist in sequencing. Whole genome sequencing is a little bit better, but it's still not an unbiased protocol. Yeah, so you can see like a nice-ish even coverage, but again, you do see generally loss of coverage in certain parts of the genome. So the assumption of unbiased sampling of the genome is never a valid assumption. And that's why, we've been doing like all these panel of normals and just looking at multiple samples because we want to learn about these biases. Okay, so when we talk about like copy number variation, there are two concepts that we need to differentiate. One of them is called copy number, and the other one is called copy ratio. The difference is very simple. Copy number is an integer, like 2 copies, 3 copies, 1 copy, 0 copies. Whereas copy ratio is basically the ratio of the number of copies that you observe of any genomic region with respect to the average. Yeah. So it becomes extremely important in the context of somatic samples for a very important reason. And that reason is that tumor is never a single cell type. Tumor is very heterogeneous. You have like multiple clones that they're competing with one another. One of them might gain some evolutionary advantage and it will just take over. The other one will take over. And you always have like an interplay between like multiple different sort of lines, or subclones within the same tumor. And let's say one of those subclones gains a copy of some region in chromosome 1, right? But the other subclone doesn't gain that. So if you just take that piece of tumor and then just put it in the blender and just do like genome sequencing of that, and if you just focus on chromosome 1, you will never see like a nice integral copy number for that region that is amplified. Because let's say your tumor might be like only 30% from that subclone 1 and like 66% from subclone 2. One subclone has no copy gain or loss, the other one has a copy gain or loss. So you get something fractional, you get something in between, right? So depending on how many subclones you have and what is the tumor purity, how much of your sample is actually tumor, how much of it is actually normal, like diploid, you get all types of like continuous, copy ratios. And that is what we're going to focus in today's presentation, to basically estimate copy ratios of different genomic regions and not absolute copy number. It's a very, very hard problem to infer absolute copy numbers of tumors because we don't have prior knowledge of how many subclones we have. And typically this deconvolution problem, namely taking one tumor sample, inferring how many different subclones we have in them, and what is the absolute copy number of each of the genes in those subclones, and what is the basically fractional presence of each subclone in the tumor sample. This is a very, very hard problem, and it's an ill-defined problem. Many people have tried to solve this. There are many tools out there for it. None of them work well. So my answer for that type of question is that we should wait for better technologies to emerge. Right now, short, like next generation sequencing, just not a good technology for inferring absolute copy numbers from tumors. Maybe something like single cell DNA sequencing is something better, but we don't have that quite yet commercialized. So let's just focus on the problem that we can solve, and that's the problem of inferring copy ratios from our samples. And we can already do quite some stuff with it. Okay. Now here's you're going to see a lot of these plots in this presentation, and these are naive. And I'll tell you what I mean by naive. Naive copy ratio estimates on the y-axis, and on the x-axis, you have different genomic positions. Let's say you have lined up all the chromosomes. These are all the regions on chromosome 1, chromosome 2, chromosome 3, right? And every point that you see in this plot is just one tiny genomic region, let's say one exon. You know, we've specified them at a time, and we've looked at how many reads we see over that, you know, genomic region, and we sort of normalize it with respect to all the reads that we see. Yeah. So as we expect, they all like hover around 1, right? Because on average, every you know, once you normalize your total, you know, the coverage over any genomic region, you know, there's this regression towards like mean if you just normalize it by the average, right? So, but you see like lots of genomic regions, they seem to have higher coverage, and many regions that have lower coverage. So this is for a normal sample. If you do this for a tumor sample, you see pretty much the same thing, no difference between them, right? And if you know you show something like this to me, I don't believe it for a second, because I just don't believe that a normal tissue would be so crazily, you know, infested with copy number variation, because we know that copy number variation is actually a rare event, right? So we would expect, you know, this, you know, for a normal sample, the copy ratio to be like mostly 1, maybe once in a while you have a copy gain here, copy loss somewhere, right? So this is telling us that this data is very, very noisy. And this is, in fact, the main source of noise in this data is what I mentioned earlier. That's the bias. That different genomic regions, they actually have systematic biases that are going to be like either undersampled or oversampled. And just correcting for these like systematic biases goes a long way in just denoising these sort of copy number profiles, and then allowing it, the actual underlying copy number to be revealed. So here's kind of like a fast forward of how that would look, and you know how the whole process looks. So first we collect some data from our BAM file, and that data comes in the form of 2 things. One is read count, like just the coverage, how many like units of coverage do we get for each of the, you know, our favorite like genomic intervals. And the other thing that we care about is the thing which is called like allele fraction. I'll return back to it later. And then we denoise them, and you can see that once we have sort of denoised them, you already see like some patterns emerging. Like most of these segments are now like copy ratio 1. You see some copy gain here, some copy gain here, neutral, loss, a bit of gain, more gain, neutral, right? So you can actually see something there that you just can't see here. So that's the magic of like denoising. And once you know we've reached this point that we've sort of denoised our data, then we can try to segment it into like constant pieces. You can say, okay, this region, this is all like copy neutral. Maybe, you know, copy neutral, a bunch of copy gain here, you know, so determining like these sort of change points that as you move along the genome, you gain a copy or lose a copy. That's the last step of the pipeline, which is called the segmentation. So we're gonna like now go through all the steps one by one. Any questions so far? Yeah. Okay. Make sure you don't forget. Okay. Yeah. Yeah, we are so what we're coming up here with is just a bulk estimate, which includes basically just a mixture of like ploidy and purity, so that inferring purity from this data and then then just focusing only on the cancer proportion portion of your data and then inferring the ploidy there. That's something that is not part of this pipeline. Yeah, so that's considered like an extra like step that you got to do after you get this result. Yeah. Okay. Here's the workflow. Here's the simplest version of the workflow, which we don't talk about like these allelic counts, and I'll return back to those at the end. So, for the bird's eye view is this. So you have your reference genome, you specify your intervals that you care about. This could be like, equi-length intervals along the genome, like, 1 kilobase, 1 kilobase, 1 kilobase intervals on the genome. Or these could be like regions of interest, like exons, or maybe just genes, just, whatever interval you care about. You specify them as input to the tool. And then you pre-process those intervals, making, just doing some QC over your intervals, making sure that you don't have like too big of intervals or like too short of intervals. And then once you have like your QC'd intervals, you give them to this tool called collect read counts. And as the name suggests, what this tool does is that it takes a BAM file and then just calculates coverage over each of your intervals to give you these read counts. And then these read counts will be denoised. And then you get sort of these denoised copy ratios. And once you get your denoised copy ratios, you give it to this tool called model segments that then this tool, what it does is that it identifies segments of constant copy ratio sort of segments along your genome. And then the final tool here is called call copy ratio segments. It's basically assigns like a gain or loss to each of your segments. Basically, this tool, what it does is just does some simple statistical tests to identify whether each of the segments, it is copy neutral, it has a gain, or has a loss. Hugo, can we return to your question now? It's better. Yeah, so this is for a whole exome sequencing data. For whole genome sequencing data, it's generally much less noisy, much cleaner. And it's because whole exome sequencing has just so much like noise and bias that no matter how well you try to denoise it, you still end up with a bunch of residual noise that you just can't get rid of. Yeah, and it's just because of the step, that particular step in whole exome sequencing, which is this enrichment for the targets of interest. That's a very biased step. It involves a bunch of like additional PCR reactions. And whenever you're doing more PCRs, you're introducing more bias. Okay. Oh, there are more steps here. So important step here is that once you do, but you reach this point of collecting read counts, you do not collect read counts over just one sample. You collect read counts over many samples. Yeah. And that's the usual, like the name of the game that you've been playing in different tools. And that is that once you have many, many different normal samples, that allows you to estimate systematic biases of your sequencing platform. The key idea is that if you just give me 50 normal samples, and I just look at one genomic region, same genomic region over all those normal samples, and I see that systematically, I get like more coverage over that genomic interval compared to the rest of the genomic interval, it's highly, highly unlikely that all of the samples have a copy number event over that interval, so that I can call that as a bias. So that's the idea here. So once we have the coverage profile over many normal samples, we turn that into something which is called a panel of normal. This is a different panel of normal than the MUTEC panel of normal. We have many different panels of normals in GATK. This is a copy ratio panel of normal. This is a very CNV specific type of panel of normal. And then this panel of normal is used in the denoised read counts tool to do the denoising step. Okay, so let's quickly go through the command lines, but I'm not going to spend too much time because we're going to do this exercise together next. But basically, we have this pre-process intervals that just give it a bunch of interesting intervals. These could be, again, either like equi-length bins along the genome or exon or whatever thing that you care about. And then you pad them, you make sure that they're not like too short, because if an interval is shorter than the length of a fragment in next generation sequencing, probably not going to be able to use that information at all, right? The resolution that the highest resolution that you could ever achieve with next generation sequencing and calling copy number events is basically at the level of the length of a fragment. If you have some events that involves like just 100 base pair or something like that, you're not going to be able to detect it with this protocol. So that's why we pad our intervals, make sure that they're big. Yeah, and so that's typically what we do for exomes. For genomes, since we haven't enriched the sample for any specific genomic region, we tend to just use some to bin the genome into like equal bins of, let's say, 1,000 nucleotides. Okay, so this tool ultimately gives us this pre-processed interval list. So as somebody asked the question, it's just a list of intervals: start position, chromosome start position, end position. That's it. Okay, step 2, it's very simple. You get your BAM file, and you have your interval list. And then you just iterate over each of your intervals. And then let's say this is one of your intervals. And then you just count how many reads overlap with that interval. And just tabulate it. And then you get this guy. Yeah. And then this is how we do it in GTK. There's this like collect read counts command. Takes a BAM file, takes the interval list, and also takes some like rules for like how to deal with intervals that could be potentially overlapping, like merge them or like split them, things like that. And then ultimately like where to write that counts data. Now here's a fun part and a little bit of a tricky part, the denoising part. Okay. Let's say we've collected coverage over a bunch of normal samples, ideally from the same sequencing center, ideally from the same person who has done all the sequencing. Like the more you can keep things uniform, the better you can estimate the systematic biases, right? Because different like wet lab people tend to have like different habits, and that actually leads to different biases, believe it or not. So even though the lab protocol is like set in stone, just do this for 30 minutes and then do that for that. People just just 1 minute lunch break here and there can just lead to different biases. Okay, so tend to keep things as uniform as possible. Just take as homogeneous and matched normal samples to the tumor sample that we ultimately care to analyze as possible. And now we get like hundreds of these copy number profiles. So the very first thing that we do with them is that we just look at the average coverage of each of those intervals, right? Because like I said before, if you see that on average, like this particular target or genomic interval is just has like more reads to it systematically in all of the samples, it means that that's something that you probably want to correct for, because it cannot be a consistent event. It cannot be like a biological thing. It needs to be something technical. So that's the most important thing. It's called standardization. Estimating this per target or per genomic interval systematic offset in multiplicative sort of rates of coverage and sort of correcting for that. The second thing it's called PCA denoising. Who in this audience here is familiar with principal component analysis? Just show of hands. Okay, cool. So just to refresh your memory for those of us that are not familiar with it, principal component analysis is a very useful unsupervised machine learning technique basically for identifying directions that explain highest amounts of variation in the data. So in the context of what we're talking about here, imagine that you have 100 normal samples, and each of your normal samples has coverage over 100,000 genomic intervals. You can think of this as 100 observations of 100,000 dimensional points in the space of coverage, in the space of targets, right? You have 100,000 different dimensions, and then you have 100 samples. Now you can ask yourselves that what are directions in this target space that explain most amounts of variation in this normal data. So here is like why we might think that this is a good idea. So this is actually something real. Genomic intervals that have a lot of G's and C's tend to be undersampled. And the reason is that the GC bonds are very strong compared to AT bonds, and they're just more difficult to break. So during like PCR amplification, you know, the denaturation step and the amplification step and hybridization step. So these are all like less efficient for regions that have like more G's and C's. More importantly, the amount of sampling bias that is induced by this, you know, GC content of each interval is a very sample specific thing. It depends on the enzymes, it depends on the temperature, it depends on the pressure, right? So even though the same target, which is like GC rich or like AT rich, systematically will be either oversampled or undersampled. The amount that it will be oversampled or undersampled is a sample specific thing. You can imagine that there is this underlying knob that each sample has that we don't know that controls like how much that bias ultimately contributes to each of these intervals. Principal component analysis is actually it allows us to identify these things. Like it basically looks at the correlation between coverages across different targets, and then it identifies these sort of correlations, and then it sort of gives us some low dimensional space in this 100,000 dimensional space of all the targets that explains sort of this sort of covariation between you know, undersampling or oversampling of different targets. So that's the second trick in denoising. And the first one is the most straightforward one, is just standardization by the average coverage. And the second one is to identify these directions that explain a lot of variation in the coverage of normal samples, and then subtracting out those dimensions from our tumor data. Yeah, you can think about that. It's if you just have a geometric intuition, you can imagine that this like high dimensional space of coverage, there are some directions that you learn using this principal component analysis. These are like technical directions that explain technical variation. So you don't care about them. So what you do is that you just imagine like a hyperplane, like it's basically just a high dimensional plane that is orthogonal to each of those directions. You basically just project your sample into that hyperplane. So you get rid of those extra dimensions that explain only technical variation. Okay, and here's what happens. So when we do that, if this is a case sample, namely like a tumor sample, the raw coverage might look like this. But once you denoise it by standardization and PCA denoising, you get this. And now all of a sudden, we can see that, there is like a loss of ploidy here. There's a gain of ploidy here. And the rest of the genome is copy neutral. Okay. Cool. So the way we do it in GTK is that once we collect read counts over a whole bunch of normal samples, there's this tool called create read count panel of normals. It takes as input the read count of each of your samples and produces this thing, the panel of normal file. And then either you can basically rely on this sort of mathematical trick to learn about GC bias, or if you specifically want to model GC bias that I explained before manually, we can also do that. in practice, it doesn't really matter all that too much because GC bias is one of the first types of biases that you learn by making a panel of normal. So you just don't need to specifically do that. I mean, unless you have a very, very small panel of normal, like maybe only 2 normal samples, in which case you probably do want to correct specifically for GC bias, because this is a very, very important bias. Okay. And then once we're there, we have our counts from our case sample. We have our panel of normal. Then there's a tool called denoise read counts that basically takes the counts, takes a panel of normal, and then it just gives us a denoised copy ratio profile. Again, since we're going to go through this command lines in detail in the exercise, I'm not talking too much about them now. All right. So again, here's how our raw read counts might look like for a specific sample. Here's how much denoising we get by just standardizing with respect to the average coverage of the samples in the panel of normal. And here's how much additional denoising we get by applying the PCA trick, by learning those technical covariates from many samples and then also correcting for them. And you can see that there's a big difference. Like even like visually, you can see that PCA denoising does quite a important thing in denoising our samples. So, but if you just look at that, you might say that, oh well, I mean, I can already see the events here. There's an event here. There's an event here. I didn't need to do the PCA denoising. I was already done there. But then there are samples that you actually only see smaller events when you do the PCA denoising. Like here's an example one. Here's like another sample that just by standardizing by the median coverage profile, you get something like that. It's not too easy to call, maybe here's something going on here, maybe something going on here, here, and here. But once you actually denoise it, like here is in particular, you just can't tell what's going on. Is it a gain? Is it a loss? Something is definitely happening here, but you just can't tell. But by doing PCA denoising, you can say that, so there is like a combination of gains and losses. Lots of intervals. Some of them have gained a copy, some of them have lost a copy. And so that to be able to make these sort of finer resolution calls, you need to step up your denoising gain as much as you can. All right. This is just like a more quantitative way of saying the same thing that I've been saying. So what we have here is called a SMAD score, or median absolute deviation. That's how much copy ratio changes between 2 adjacent intervals. Typically, you want this number to be small, because if you have like a really noisy data, there's a lot of variation from copy ratio from like interval to interval. So the more denoised your sample is, the smaller this number becomes. And you can already see that you initially start with a median absolute deviation of like 0.3 almost. By doing PCA denoising, you just reduce it down to like 0.04, 0.04, so almost like 6 times, 7 times like less noisy. Okay, so it's also important to use a good panel of normal. Like here on the left, you can see what we get if we use the good panel of normal. You get a lot of denoising. If you use a bad panel of normal, you get something like that. I mean, it's still okay, but it's not as good as that. So that's basically just reiteration of what I said earlier. The more closely matched a panel of normal is to the sample that you're analyzing, the better you can just do denoising. But sometimes it's unavoidable. So you gotta basically just use what you can get. Okay. I think I covered all this. Make a good pawn. And use samples that were sequenced in the exact same way. Younger individuals are less likely to have copy number variation from clonal hematopoiesis. So using blood normal for older patients is kind of tricky, because you do see copy number variation in actually in blood and as we age. Definitely normals from patients with hematological cancers, they cannot be considered as like normals because those are cancer samples. And finally, like how many samples do you need to make a good panel of normal? You can get away with 10. Anything above like 30, 40 is sufficient. After like 100, there's nothing much to be gained. So a good panel of normal has about like 40, 50 samples. For whole genome sequencing, you can get away with even like 5 or 6. So this is really like relevant to whole exome sequencing. All right. So I covered basically that part of the workflow, but there is one part that I totally skipped. See how I'm doing on time. I tend to talk too much, so I'll try to finish this in like 10 minutes. So there's this other important step. It's called like collect allelic counts, and that's another source of information that we can leverage in calling copy number events in case samples. So and here's how it goes. So imagine that you have in your normal sample a ref alt genotype, right? So that you have like a point mutation, common variant or something, right? That you have 2 different alleles on your paternal and maternal chromosome. Okay, so the allele fraction here is going to be half. This is a canonical like heterozygote. But now let's say one of your chromosomes has gained a copy, right? So the allele that was basically made its way into the extra copy changes this allelic balance. So a SNP site, which was previously the allele fraction was half, the allele fraction will now be 1/3. Or depending if it was like home ref or home alt and something like that. So ref alt, depending on whether the alt was duplicated, the ref was duplicated, you could either get the allele fraction of 1/3 or 2/3, right? So, but nevertheless, what I'm trying to say here is that your allele fraction will deviate from 50%. Like if you choose a site which was heterozygous in the normal sample, it will not be heterozygous in the tumor sample if that SNP is in a genomic interval that has undergone some type of copy number variation. And that's sort of the other interesting signal that we can use to detect potential ploidy loss or ploidy gain, right? Here's a typical sort of allelic count track, how it would look like. It's very similar to your, to the copy ratio or plots that I was showing before, except for that on the y-axis, now I'm showing the alternate allele fraction, the fraction of reads that support the alternative allele at some sites that we have determined to be heterozygous, right? So if we make the same plot for the normal sample, it should be like straight 50%, maybe with a bit of noise. Yeah. So anytime that we see like allelic fraction going above 50% or going down 50%, we can definitely tell that because this something is happening here. So either the maternal copy is getting amplified or the paternal copy is getting amplified. And that's why you get basically this sort of bifurcations, right? You go both up and down simultaneously. Okay, so the tool that does that is called collect allelic counts. Just takes a bunch of SNP sites, common sites from dbSNP or Nomad or your favorite source of common variants. It takes your tumor sample and it collects the allelic ratios over those sites. It does the same thing for your normal sample, but ultimately it focuses on the sites that are heterozygotes on the normal sample, because those are the ones that are informative. All right, so now we have 2 streams of information. We have our copy ratios, and then we have our allelic counts, and both of them are informative about the change points. So, and that's what this tool, model segments, does. It takes both of these information and then it identifies change points and segments. All right, so this is getting into the technical land, but the tool that the approach that the predecessor tools we're using for determining these change points were based on an algorithm called the circular binary segmentation, that it would basically look at, it would try to identify change points that would have like different mean before and after. This is something that is definitely relevant to the coverage, because whenever you have like gain or loss of ploidy, before that breakpoint, to the left of it, to the right of it, you would have like a different copy ratio. So the mean coverage would be different. So that's the natural way that you want to define the change points. But when we're looking at something like this, allelic ratios, the mean is always 50%, right? So it's because the alternative allele is completely, it's not well-defined. We just take something that is not referenced, we call it alternative, but it's not the reference for that sample in particular. So that's why that you have like these, the mean of allelic counts remain at 50%, but it's really the variance that is changing. Yeah. So we need to have like a segmentation algorithm that is not only sensitive to changes in mean of a distribution, but it's also sensitive to variance and potentially higher moments of the distribution. And that is the algorithm that we've implemented in GATK. It's called a kernel segmentation. And kernel segmentation, a kernel change point detection, they belong to certain class of change point detection algorithms that can very sensitively identify change points in the trends of your data, depending on whatever change that they might have, either like in mean or variance or like higher moments. And that's how we can get away doing like segmentation of both allelic counts and copy ratios simultaneously. Okay. And it's very easy to use. You give it your copy ratios, your allelic counts, and also your normal allelic counts, and then it just gives you segments. Now, there's this very important parameter here is called number of change points penalty factor. And that's because in principle, any point can be a change point. So you gotta have to determine the sensitivity of like how many change points you want to allow to be detected, right? So too many change points probably is not a good thing. Too few change points is not a good thing, but there's a single knob that determines how many change points we detect in the sample, and that's its penalty factor. If you're interested in the math behind all this, just let me know and I'll send you a paper. But this is the single parameter that if you increase it or decrease it, it changes the number of change points it detects. Here's visually how it looks. If you change that penalty factor to a very, very small number, you identify a ton of change points, both on the copy ratio segments and on the allelic fraction segments. And as you increase that penalty factor, you just identify fewer and fewer segments. So if you want to look at like really specific events and like specific chromosomes that you care about, like specific locus, you probably want to increase this parameter so long as you can basically see that focal event that you're looking for. All right. So I covered now that part of the workflow, and the final part of the workflow is a very, very simple part. Once you do the segmentation, it's you just want to like refer to the segments that either like gain of ploidy or loss of ploidy. It's a tool called for that. It's called call copy ratio segments. It takes a segmented copy ratio and it gives you like gain or loss determination to every segment. There are also tools for plotting them. Like all the plots that you saw in this presentation, they were made using this tool called plot denoise copy ratios. It just takes the same files that you've generated using previous tools and just makes these nice plots for us. And here's the entire pipeline. To recap, you pre-process your intervals, you collect read counts, you make a panel of normal, If you have a panel of normal, you can step that step. You denoise, you collect allelic counts, and then you take the denoise profile and the allelic counts, you give them to model segments, you get the segments, and then you call gain or plotty gain or plotty loss in each of the segments, and optionally you make some plots. That's the entire part. All right, let's take a break. Yeah, please. You mean cancer-specific signatures of copy? Yeah, there's a bunch of papers on that. Yeah, yeah. You're right. Yeah. So if you just take the same segments that you get here on like many different cancer cell lines, and then you cluster them, you actually do see some recurrent ploidy gains or ploidy losses that are hallmarks of specific cancers. They're like BRCA1, BRCA2, breast cancer and things like that. So you do see them. Yeah. It's again very varied. So the copy number profile of cancer is not like not all the lung cancers, for example, have the same copy number profile because there are lots of passenger copy number, copy gains and copy losses. It's a usual game that we have in cancer. You have a bunch of like driver mutations. In this case, you have a bunch of like T amplification or like deletion events. But then since the genome has become unstable in cancer, you keep gaining a bunch of additional gain and losses. So it's kind of difficult to establish a very a cancer-specific copy number profile. Yeah, so we don't have that in GATK. That's actually, so there are actually tools that even take those as priors, specifically tools that are developed for like determining absolute copy numbers. Yeah, but we don't have that in GATK. All right. Derek, do you want to continue today for the exercise, or do you think you can? How's everybody here feeling? Do you have the energy for another like 40 minutes of exercise? Sticky note, red sticky note is like, green sticky note is let's do it tomorrow. Well, all right, let's just take 5 minutes break then. And then we'll do it today. Oh, that was green? Sorry, I missed it. All right. So let's get started. And let's try to finish as quickly as we can. All right, so open up your Jupyter notebooks from the earlier step. It should be in the workspace like 2023 Cambridge workshop, the same workspace that we did that terminal magic on. And then yeah, open, if you have not already opened, open like 08 GATK Somatic CNV tutorial. Let's go to the kernel. Make sure you do have the conda and GATK kernel there, and make sure that it's selected. Show of hands if you don't have that. Cool. Great, you're on the same page then. Okay. So let's just go through the steps. We've already done this. We've already done 1.2, which was to set up the GATK conda environment. We've already checked the kernel type. We made sure that it's conda env colon GATK. And now this is like very similar to some of the other notebooks that we've seen before. Let's just go through them. Here we are defining the bucket. That's the Google storage bucket that is associated to this workspace. We are using data from one of the previous workshops. That workshop was in like February 20. That's the identifier for that workshop. Let's now make some local files, some local directories inside this virtual machine. And then Let's just import a bunch of Python packages for displaying images. And then let's just list that GATK tutorials Google bucket. You should be able to see something like that. If you do see it, it means that you have access to the files that we're going to use for this tutorial. Red sticker, red sticker.
It says may not exchange. Is that?
Yeah, but we probably have 2 copies of it open, so you can override this, but you can kill the other copy.
Okay. No, it's okay.
Now, let's move along. In 1.6, we're going to download the data to this notebook. Cool. Just about half a gigabyte of data. It should be pretty quick. Okay, at this point, everything is set. So let's just go through the first step of the pipeline, which is to process intervals that we care about. So here in this tutorial, we're going to use a bunch of targets on chromosome 17. We are gonna process them with the process interval tools. We're gonna pad them by 250 base pairs on either side to make sure that they're long enough so that they can actually get some coverage over them. If they're too short, they don't get much coverage, and then that interval might be filtered in downstream steps. So let's run this. Now, it's probably going to take about a minute. Yeah, and once we're done with that step, we should get our interval list file. Should contain about like 11,000 targets, which we will then use in the subsequent steps for like coverage collection and calling up numbers and whatnot. All right, it's done for me. So let's just look at it. Here, all I'm doing is that I'm taking the original interval list that we gave to the tool, to the pre-process intervals tool, and I'm skipping the headers, and then I'm just counting the number of lines, and I'm getting 11,307. That's how many targets we gave to this pre-process interval tool. Now, let's see how many targets we get out of this tool. And we get about as many targets out of this tool, which is reassuring. And here is just like print bunch of, first targets. So here you have the chromosomes, here you have the start position, the end positions. These are ignored, so don't worry about them. And yeah,
the problem that the pipeline is broken.
That is a Jupyter notebook thing. Yeah, don't worry about that. Okay. And this is this is the original interval list that we provided to the tool, and this is the pre-process interval list that we get out of the tool. If you compare them, you will see that the interval lists are slightly different. So here's like the length of the initial interval that we gave it to the tool. So this interval, the specific interval, was 75 nucleotides long, which is definitely too short. And that padding command that we just basically added by 250 base pairs symmetrically should increase the length of this interval to 575. And indeed, you can see that this interval is now the length, which is written on the last column of the interval list file, is like 575. In fact, every single interval that you get out of this pre-process interval tool should have at least 500 base pairs in length, because we pad them, we pad anything that goes into this tool by 250 base pairs from either side. Okay. Great. So now what we want to do is that we're going to take our tumor sample here, I think the tumor that we're going to be looking at here is a cell line. It's HCC 11 something. It's a ductal carcinoma, breast cancer sort of cell line. It's a very old cell line. It has been passaged a zillion times, I guess, at this point. Yeah, so what you're doing here is that you're just giving it the input tumor BAM file, our pre-processed interval list, the reference, the usual HG38 reference. We're gonna ask it to give us the output in the TSV format. Interval merging rule is that do not merge intervals that are like basically abutting one another, because otherwise, if you have like many intervals that are just like, you know, head to tail with one another, they would be merged into one big interval, and we don't want that. You know, we want to have like distinct intervals. So that's what that command does. And ultimately we specify like where we want the output to be, which is going to be in the sandbox directory that we made earlier in this notebook environment under the name tumor counts TSV. So let's run that. And we run the exact same command on the normal BAM file. It's a matched normal for that sample. This should finish pretty quickly. So on my end, I'm already done with the tumor read counts, and I'm done. I'm on to doing the normal sample read count collection step. And okay. And if you take those TSV files, and then if you just make a plot of them, you would see something like that. So this is the raw coverage of our normal sample. This is the raw coverage of our tumor sample over those 11,000-ish intervals that we provide. Okay. Now, the tutorial is asking us that, can you tell if either sample has copy number variance? And then you should answer no, because they're both very noisy. So what we're going to do now is we're going to go ahead and make a panel of normal. So here we're going to make a very bad panel of normal because it's going to contain only one sample that matched normal for this tumor, right? That's not good practice. This is just, you know, something we're going to do for this tutorial. If you don't have any other way of making a bigger panel of normal from other samples from the same sequencer, same technology, then maybe this is the best thing that you could do, but you should avoid doing this in general. But let's do it for now. We're going to take that single normal sample that we had, take the read counts that we collected over that sample. I'm going to give it to the create read count panel of normals tool, and then this is going to give us like an HDF5 PON panel of normal file. If you have multiple samples that you want to use to make a panel of normal, you just input each one of them separately with a -i command. We have like 40 samples, just have like 40 of these -i. Okay. So the tutorial is asking us to study the standard output from this tool. Let's just take a look at the standard output. So this is the usual GATK output. And then we're going down. You have 11,307 intervals, so far so good. Pre-processing and standardizing read counts, doing a bunch of things. After filtering, 10,169 out of 11,307 intervals remain. So what happened here is that we lost a bunch of intervals. So you might ask that, why did we lose some intervals here? The reason is that this create panel of normal tools, by default, it applies a bunch of filtering on the intervals. So if you have an interval that doesn't have any read count over it, it just doesn't have any coverage, that's not a useful interval, right? Because the idea is that you want to use that interval to determine like average coverage so that you can then normalize your tumor's coverage to that average coverage. So if the average coverage is 0, zero, anything divided by 0 would be infinity. So in general, whenever you have any interval that doesn't have any counts, it's very likely that that interval was not even enriched for in your whole exome sample. So it's apt if you just removed it. So by default, what happens is that this tool just looks at the distribution of coverage across all of your intervals, and it just looks at the tail of this distribution, and just removes like the bottom 10 percentile of counts. But then you can modify that behavior if you want by changing the default arguments. All right, so we can repeat that step again, but now we can change the minimum interval median percentile to 5% from its default value. I think the default value is 10%. So by changing it to 5%, now we're probably going to retain a few more intervals because we're not going to be as aggressive in filtering the intervals. So let's just run that, and then let's take a look at the output. Now, what we see in the output is that oh, well, now we're actually using a whole not only on chromosome 17, but this read count. This is something that we have collected before. This has like coverage over the entire genome. So that's why we had here 188,000-ish intervals to begin with, but we ended up with like 179,000 intervals. So, but a good exercise for you would be to just take this single normal sample, just the coverage that we collected over chromosome 17, and mess around with that minimum interval median percentile parameter to see the effect that it has on the filtering of the intervals. Okay, so yeah, so we're actually going to use that the entire genome-wide coverage for the rest of this tutorial. So just focus on chromosome 17 and just the first step, because otherwise it would take a couple of minutes. Okay, so now here's the fun part of it. If you're going to the fourth step of the protocol, the workflow, which is to denoise our tumor sample using the panel of normal that we made. Okay, so the command is denoise read counts. It takes as input here we're basically denoising both our normal sample and our tumor sample separately. And we're using actually a big panel of normal that we also supplied for this tutorial, not that single sample panel of normal, which I said is not a good practice. So basically what's happening here is that we made a panel of normal ahead of time using 40 samples. And now we have these two samples, a tumor sample and a normal sample, and we're denoising both of them using that panel of normal. So the first command denoises the tumor sample, second command denoises the normal sample. Just run both of them. It's pretty quick. Okay, so if you just skim through the steps of the denoising, that gives you a good idea of what kind of denoising transformation is happening on your sample. So it says here that pre-processing and standardizing sample read count. That's basically just taking your raw coverage, dividing it by the average coverage for that sample, so that you can just standardize coverage for your sample to begin with. And then that's the step that it says transforming read counts to fractional coverages. Does that. Subsetting sample intervals to post-filter panel intervals. So, some of the intervals that we didn't have in the panel are going to be removed here. Dividing by interval medians from the panel of normal. That's basically just correcting for the average systematic capture bias. Standardizing read counts again. Dividing by sample medians and transforming into log space. So before we do the PCA, we take a log. It's because PCA is really applicable to normal data, whereas coverage coverage by definition is a positive number, is not normally distributed. So the trick is that whenever you want to apply a statistical method to any data that is not normally distributed, just take a log and hope that it becomes normally distributed. It's a heuristic. That's what you're doing here. It often works very well. Yeah, so then you're subtracting sample median in the log space is equivalent to dividing in the real space. So that step is again standardizing the counts of our sample with respect to the panel of normal, average per interval coverage, and then it is using 20 eigensamples to denoise. Basically, what it's doing is that it is doing a principal component analysis of the panel of normal samples, the coverage of those guys. It takes the top 20 principal components, and it's assuming that those top 20 principal components are technical variation and sort of projecting those out from the sample that you're trying to denoise here. All right, that's pretty much it. Then it does a bunch of additional standardization and then returns back from the normal from the log space to the normal space. Okay, so at this point we can plot our denoise copy ratios. It should look much better than what we had above. Remember that we started with something like this for our normal sample, super noisy. Now let's just go and plot. Yeah, we were here. Just plot this guy. It is going to produce a PNG file somewhere, which we're going to display in our notebook environment. Oh, yeah, that's for our tumor sample. This time around, this is for a normal sample. Anybody who got potentially an error message at this step, you shouldn't if you had already set up your Python environment properly. Excellent. And now let's display it. Excellent. Now we get these two denoised copy ratio profiles for our normal sample. And so this is the first one is denoised just by standardizing, by just getting rid of that systematic per target capture bias. And here it is doing additional denoising using PCA denoising, which I can see that it's basically just a little bit cleaner than here. But something funny is going on. This is supposed to be a normal sample, but it looks like that the copy ratio over chromosome 2 is not 1. It's actually slightly elevated. What do you think is happening here? I'll give you a hint. This is a cell line.
Will be a Germline CNV or tumor and normal health.
Yeah, that's exactly what's going on. So whenever you're dealing with cell lines, and you know it very well, that you make two different cell lines, one from, in this case, from tumor, one from your normal sample. And to make a stable cell line, you need to passage these cells like 100 times to stabilize them. And during this step, every time that you're passaging your cells, you're passing them through some bottleneck. And then if some type of a copy number gain leads to some proliferative advantage in some of your cells, they will take over. And if once you passage this 100 times, it's basically running evolution in real time, and you're just selecting for cells that are like fit for like proliferation. And that's why you basically have copy number alteration. So this is basically formation of cancer in a petri dish, in a cell culture, basically. So that's what's happening here. So this basically tells us that this is not a good control, right? So if you use this normal sample as a matched normal to denoise the tumor sample, you would be doing actually very poorly, because this is not actually a good normal for that tumor. They have diverged. Luckily, this is not what we've done. We basically took another like 40 samples externally, and we used those 40 samples to make a panel of normal, and we denoised both the tumor sample and the normal sample separately with respect to that panel of normal, which hopefully only contains technical variation and not this type of like biological variation we care about. Yeah.
And where you have GMAP. Could you use those and use that
Absolutely, you can. So the question is that if you have like 10 samples from patients from your normal, can you make a panel of normal from those 10 samples to call other samples, but also call the same samples with? And the answer is yes. So if for example you care about looking at some aneuploidy events in those normal potentially, like if there's like blood samples, you might lose some sensitivity there if you use the same panel of normal for the same samples, to denoise the same samples. But if you use that panel of normal to denoise tumor samples from those same patients, you're good. Okay. Cool. So we got basically two sets of plots. Now here in the notebook, we're showing them side by side. This is our tumor. This is our normal. On the top row, we have the most straightforward denoising, and on the bottom, we have our best practices denoising, which is a combination of standardization and PCA denoising. And the point is to say that the median absolute deviation is slightly lower once you do like PCA denoising. So it's good. Generally, you can also see that your tumor sample has a lot more ploidy alterations compared to your normal samples. Okay, now this is the step that we can take these denoised read count profiles and give it to model segments and ask model segments to produce constant copy ratio segments for us. Just run it. I'm at cell 261. The first one is for the tumor. The second one is for the normal. Should be pretty quick. As you can see, it's identifying change points, then it's applying penalty, it's refining change points. It's an iterative algorithm that tries to identify the best set of change points along the genome that segments your sample. Okay. Try. Okay, I'm done with the tumor sample. I'm down to segmenting the normal sample now.
Yeah. Is it possible to have the same plot, the last one, so with the cell line?
Yeah.
No, if I lower the tumor sample time, that the tumor sample is the tumor, and in the place of the normal, I use the tumor. Will be also this human error.
Oh,
Give us.
yeah, I see what you're saying. So in this tutorial, you're running everything on the normal sample and on the tumor sample side by side. You could just run this tutorial on the normal sample or on the tumor sample or both. There's no place in this workflow that we're using the normal as a match for the tumor or anything like that.
In case I make dot 2 times the like tumor normal. I know that
it's user error, yeah. So that can happen.
User error. Well, do I have the same plot?
Mm-hmm.
I mean, I will have the same situation.
Yeah, so I mean, if you, for example, here, well, here we actually on the sample names we put like a tumor under it so that we can hopefully like catch these errors. But I guess maybe the hallmark of using a normal sample in place of a tumor sample is just seeing a lot more copy number activity. Yeah, so there's nothing in the workflow that allows you to catch that, and you have to visually inspect it. That's what we have this plotting. Yeah, but this pipeline in particular is that doesn't leave too much room for mistake because you're not using any sample as a matched for any other sample. Just a panel of normal and a case sample. That case sample could be normal tissue, could be tumor tissue. If it's a normal tissue, it's going to be sort of silent, not much copy number activity. If it's a tumor tissue, it's going to be like that. Okay, so our segmentation is finished, so we can use the same okay, so now we have another tool called like plot model segments that it takes denoise copy ratios, the segments that we got from the previous step, and a bunch of additional things for like making sure that we can visualize things properly, and it just produces some additional images that we can inspect of the segments. So I just run that for the tumor sample. I'm like just showing it, and voila, here's what we get. So it's basically the same sort of copy ratio plot that we had before, but now we have ascribed sort of segments to it. And just as a visual aid, every other segment is colored red and orange, orange and blue, sorry. So for visual clarity, so it's like blue, orange, blue, orange, blue, orange. So that's what the color scheme is. And you can see that we get a lot of samples here. We get a lot of segments here, right? Okay, so if we do that for the normal, we probably get fewer segments. Plot. All right. So here's what I get on my normal. As you can see, I get far fewer segments. Basically, I get one segment here around the region that we discussed before. That is probably some type of ploidy gain in our normal sample, and a bunch of ploidy loss here in chromosome 6. Yeah. And optionally, you can take your segments that you obtained from the segmentation step, and you can sort of call them, which, like I said before, it ascribes to each segment either plus, minus, or 0. 0 being that segment is copy neutral, plus gain, minus loss. You can inspect the output of this tool on your own time. Okay, let's move on to the last step of the pipeline, and that's incorporation of allelic counts information. So right now, we've only looked at copy ratios and coverage, right? We collected coverage data, we denoised coverage, we segmented coverage, we called coverage, right? But like I said before, there is another very useful piece of information that we can use for identifying these copy number gain or loss events, and that's to look at changes in allelic bias of heterozygote sites. And for this, you're definitely going to use the match normal. And you're using the match normal to actually identify het sites. Okay. So the tool is, the very first step is that we're going to like collect allelic counts over our tumor BAM file and normal BAM file. We're going to provide a list of putative SNPs so that we can then later on look at those and identify hets. Het is heterozygote. And because it takes a while, we're only doing this just over a piece of chromosome 17. but later on, we will just use the allelic counts that we collected ahead of time over the entire genome for the subsequent steps of the workflow. Yeah, this should finish in like a minute or two. Any questions so far? Okay, while this is doing its thing, let me also mention something that I might have forgotten to tell you, and that's another very important thing that we can actually see by collecting allelic counts that we don't see necessarily in coverage. And that's the type of thing that happens a lot in cancer. Is called copy-neutral loss of heterozygosity. So imagine some events that takes like an entire paternal chromosome, it gets deleted, and then the cell tries to fix itself by sort of using, some of this like homologous, repair mechanisms. It's probably gonna grab the other copy that has remained and just make an extra copy out of that, right? So what's gonna happen is that you're gonna get you're gonna get the second chromosome back, so you're back to diploid. So if you just look at changes in copy ratios, you're not going to see anything, right? Because in terms of like copy ratio, nothing has happened. You just lost your paternal, let's say, chromosome, but your maternal chromosome got basically duplicated. Nothing, no ploidy lost or no ploidy gained. But if you look at SNP sites that were heterozygotes in your normal tissue, they're now all going to be either home ref or home alt. They're going to be homozygous now because you get the same alleles over the two either paternal or maternal chromosomes that you now have, right? So in terms of looking at your allelic counts, let's say what's going to happen is that all the SNPs are going to be like the minor allele fraction is going to be half. And once you enter the region that the genome has undergone copy neutral loss of heterozygosity, it's going to be either like 0, 1, 0, 1, 0, 1, 0, 1. Because it's either going to be like home ref or home alt, right? So that's the type of thing that is very, very important for us. If you want to like, if you care about that type of events, it's very important to use allelic information. And we're going to see that in a second. All right, so just take a look at our allelic counts. It's going to be like a table like that. It's going to look at like every SNP positions that we have provided. It's going to tell us like how many ref counts do we have, how many alt counts do we have, and what is the ref nucleotide and what is the alt nucleotide. So if there is a SNP position that we don't have any alt counts, we're going to get an N nucleotide here, but we just don't know. And those are going to be filtered out anyway. Now, what we're going to do is that we're going to like go back to the model segment step and not only provide the tool, the denoise copy ratios, but also give it the allelic counts for the sample that we care about and the normal allelic counts. Yeah. So now we're going to just run this on the tumor because now we're going to use that normal as a match for this tumor. And you got to remember that, because of those weirdness that was happening on chromosome 2 and chromosome 6, maybe you're going to get some like loss of sensitivity there. All right, let's just run model segments. So in general, do you expect to get more segments or fewer segments once you include allelic information compared to the copy ratio alone? More? I think you're going to get more. And the reason is that exactly because of those events that you're going to miss in copy ratio, and the CNAs, copy neutral loss of heterozygosity. Those are the things that we just can't detect by looking at copy ratio alone. But the allelic information, now we can detect those additional change points. So make sure you check that on your own. So under the hood, what's happening is that this tool separately goes through all the targets in each of the contexts separately, does sort of change point detection over each context separately. So that's why you get like so much output from the tool here. It's like as many of these as the number of contexts that you have. While it's running, let's just sort of look at some of the output if just to see if we see anything interesting. Yeah. So basically, we provided the tool about 9 million putative SNP sites, but out of all those, it only kept 17,000 of them. So what happened is that the tool looked at our normal sample and identified that out of all these like 9 million het sites, about 17,000 of them were heterozygous in the normal sample, and they had enough coverage to be reliably called heterozygous and stuff like that. So that's why we go from 9 million to 17,000. So ultimately, you can imagine that we're down to an allelic count, which only contains counts over 17,000. That's like how many of them that the model is using. Okay. This is done. All we got to do now is to make plots. Same command line, plot model segments. Again, the only difference is that we're giving it the allelic counts, and we're only doing it for the tumor because now we don't care about the normal. The normal is used as a control for heads. And let's just display it. And here's what you get. And indeed, you get a lot more segments. You get about like 440 segments, whereas before you should have gotten something about like 240 segments. So you get almost like twice as many segments. Yeah, if you inspect this a little bit closely, you can see some very interesting stuff going on here. For example, look at like chromosome 4. Pretty much all the allelic ratios, like my alternative allele fractions in the tumor sample is either 0 or 1. But the same sites, by definition, in the normal sample should have had an alternative allele fraction of half. So definitely this chromosome has undergone copy neutral loss of heterozygosity, or not necessarily copy neutral, but definitely loss of heterozygosity. Maybe one of the chromosomes got deleted and the other chromosome got duplicated 5 times. So now you're going to look at the copy ratio to basically infer what's the copy number. So it's really the combination of these two signals that tell us a whole lot of information about what might have happened in this sample. Okay. Now, you can also just do segmentation just on the allelic counts alone. Forgot the fact that you also had like copy ratios. And you can do that. And I spare you the wait time to see that result because it's getting a little bit late. But this is what you get if you only segment allelic counts. You get very, very few segments. And the reason is that allelic counts is not a very informative source of information on its own. You get only about like 16,000, SNPs that we could use. And yeah, so it's really the combination of the two streams of of signals that allows us to get like really good segmentation of the copy ratios. Yeah, so that's basically about it. Great. Any burning questions? Awesome. So feel free to write more questions on that document if you had any. All right, we're done for today. We just finished like an hour and a half later than what we expected. So sorry about that. We'll see you tomorrow morning. Have a great evening.
To our standard calling pipelines. And I'm going to start out this morning by giving you a bit of a tutorial on how to use and what concerns you might have if you want to do mitochondrial calling using the GATK. So like everything in this workshop, it's all easily available in a featured workspace on Terra so that you can see, with some easy to run demos, so you can see exactly how you would go about doing mitochondrial calling with our GATK tools, and you can see our best practices pipeline for calling the mitochondria. So once again, you go to Terra. If you are just going to the main page, terra.bio, you want to log in up here, and that'll get you here. Go to view workspaces, and you should see all of these featured workspaces available, and you can just type in mitochondria. And here we go. Mitochondria, SNPs, and indels, HD38. So like everything, this is a reasonably well-documented featured workspace that everyone should be able to see. So this is our best practices workflow for actually running mitochondria calling. You'll notice in our, standard best practices calling intervals for haplotype caller and mutex, generally we exclude this. That's because mitochondria is different in a number of ways and makes it actually really difficult to apply the same tools to do calling on the mitochondria versus not. But we have this available for anybody to use. It's read-only for me, it's read-only for all of you, but as usual, you can just clone it, and you should be able to just run whatever you want in your project if you want to deal with mitochondrial data. Let's go over to my clone, and let's look at what's actually in this workspace. So you can see here, we have a few things. We have this participant and sample. Again, there's a couple of ways that we can sort of put our data in, but you can see this participant field isn't particularly exciting in this sample. If we go down to the data, you can see we have one sample in here. It's again our favorite sample, NA12878. And this is just a reminder that most of the time when you're doing whole genome sequencing on a human, you are also getting a lot of mitochondria data that sort of carries on for the ride. And so you can just take your whole genomes and put them into the mitochondria pipeline. In fact, you might even be able to take whole exomes because there's so much mitochondria contamination that comes from your calling. So what's in here? We can sort of look around. Reminder, these are long, wide arrays. In Terra, if you want to find what's actually there without dealing with the UI, you can click here and you can sort of see what's actually in here. You can see this has actually already been run for demonstration purposes. So it already has the output VCFs present, but you can see all the stuff, all the inputs that are actually present for running, including the subset BAMs, some inputs. So let's go over to the workflow. We have an easy to run workflow that is just our full mitochondrial pipeline. So let's click on that. You can see it's version 4.1.8, and it's actually just going directly to the GATK Broad Institute GitHub. So this is actually happens to be hosted in the GATK. We host our WDLs in all kinds of places. It shouldn't be too difficult to find any WDL that you care about. But if we want to actually run this pipeline, you can see there's all kinds of inputs. This workspace has been curated such that all of these inputs are available and should be easily runnable if you clone it. Most of these site inputs are actually just direct information that is relevant for calling. I'll get into why we need so many of these mt.anb and ANN and BWT. These are all just inputs for the aligner. You know, in some of the other pipelines I showed you, those were side inputs that sat with the reference as well. We need the same thing here. We actually provided just a mitochondria reference, but we should be able to actually run this data from here. So if I go select data, I go NA12878, and I hit go, and hit run analysis. It's as easy as that. And actually, on top of that, since in this workspace I've run the mitochondria analysis recently, this should use call caching, and so this should actually be done hopefully by the time I'm done talking. Just because it's pulling up in the data that's already been run from workflows that have already been run. But if we go back to that workflow and we want to interrogate what's actually going on here, as always, there's a couple of ways to view the script. I can click here into the source and it'll take me to doc store, and then I can go interrogate what the actual files and tools are, and I can go read the WDL, and I can see down here it's doing all kinds of things like splitting multi-allelic sites. It's using some GATK print read commands. It's doing a bunch of stuff. You know, if I wanted to write my own tools based on these pipelines, going and reading the WDLs and finding out what, my own pipelines based on these pipelines rather. And I don't want to use WDL for whatever reason, I can always go and find all of our WDLs are publicly available, and you can find exactly the scripts we want run. It's just bash inside a Docker image somewhere. And you can also view the script here if you'd like. But let's do a little bit of talking about why the mitochondria warrants this extra pipeline and why we went through the trouble of making this sort of a first-class pipeline within the GATK. So what's different? Well, mitochondria DNA is actually kind of difficult in a lot of ways. As you might have remembered from your biology, it's a circular chromosome. It's present in almost every cell in your body. In most tissues, it's present. And you tend to have somewhere in the order of thousands of them per cell. And what that means is when you're reading mitochondria in just a whole genome sample, you're going to end up getting many, many thousands of copies of mitochondria DNA over your autosomal DNA. It's just so overrepresented in your tissue. And this is tissue dependent, right? If you're doing a blood sequence, actually, you're unlikely to get as much as if you were doing, say, muscle sequencing. But that means that even if you have nominally a 30x whole genome that is really easy to call in haplotype caller, you end up with so much extra data that you have to do something with. And that could actually cause problems for calling for a lot of our tools. There's also some interesting differences. You wouldn't want to run haplotype caller necessarily for mitochondria DNA because it is haploid, right? So you get one copy, or at least one cell's worth of copies of the mitochondria from your mother. But oftentimes the interesting thing you might want to study in mitochondria is to start looking at somatic variations that you've accumulated over the course of your life. So for that, we might want to use mutect for calling the mitochondria. And then importantly, if you do start worrying about those really low allelic depth calls, so stuff that you only see like 10 or 20% in your mitochondria DNA, well, you actually have to start concerning yourself with a big source of artifacts, which actually come from these things called numites, or nuclear mitochondrial DNA. Basically, over the course of evolutionary time, all of us, and dependent on different populations, there are different numbers of these that are present as SVs in our genome, have copies of the mitochondria DNA that have been absorbed into the autosome. So at some point, something went wrong. A chunk of 1,000 bases, or in some places upwards of all 16,000 bases of the mitochondria, get put into your chromosome somewhere. And then that sort of gets decoupled from the mitochondria DNA that you see that we actually want to sequence, that is actually present. And so that nuclear DNA can pick up variants, just like any other nuclear DNA. And it's usually not under much selective pressure. So oftentimes you get these these chunks of the mitochondria that are just sort of in the nucleus or in your autosomal DNA. And those variants will then, if it's not present in the reference, or even if it is present in the reference, those reads and those variants that come from that nuclear DNA get aligned to the mitochondria. And they have other variants on them that aren't real, that aren't actually present in your mitochondria sample. And so they can create lots of artifacts and misalignments and errors when you're doing mitochondrial calling. So you want to be very careful if you start calling like suballele fraction or subclonal fractions of mitochondria variants, you want to be very careful that you aren't actually calling a new mite that really did exist in the nucleus somewhere. So what does the pipeline actually look like? We have a couple, like, this is basically a representation of the pipeline on the right here. You'll notice at the highest level, it's just subsetting the BAM to just the CHRM. So we're subsetting to just the reads that are mitochondria or aligned to, or look like they might possibly be mitochondrial. We revert the BAM, so we actually end up doing a full realignment of our reads. And then we do this extra special step called shift FASTA, or aligned to shifted MT. So we do this aligned to MT and aligned to shifted MT. So our solution in haplotype caller, or well, at least in this mutex pipeline that we use for calling to deal with circular DNA, is to take our linear reference of the mitochondria, where, you know, even though it is in reality circular, we, you know, the reference only has 16,000 bases of it starting at some point. So we made a cut point somewhere. If you aren't careful in BWA and in most aligners, that you aren't going to align across that cut point very well at all. You know, if you have reads that read on both sides, it's possible that the aligner is going to throw an error and not do very well, or maybe you're going to get lots of soft clips. So that's actually a really big source of artifacts if you aren't careful handling that. Our solution might sound a little hacky because it sort of is, where when we do mitochondrial calling, we produce two versions of the mitochondrial reference, one that's the basic, you know, NA1 or the basic HG38 version of the mitochondria, and one where we've sort of shifted it or rotated it by, you know, 180 degrees. So the breakpoint is on the opposite half of the chromosome. And so we just do calling and alignment in that region separately. And then we sort of later go back and merge those BAMs together. And we find that actually really helps for resolving some of the errors that you get at those boundary breakpoints. What else does the mitochondria calling help with, and what else is present? We use a special version of mutex 2 that has had a bunch of arguments added to it that sort of help it better handle the depth. Because again, when you're dealing with 25,000 bases and you're looking at a brain graph, right, the chance that any given base has an error in it, just by virtue of how messy the reading is very high. And so you get these like crazy complicated graphs. And so you have to be very careful when you're using any variant calling tool on this to handle those correctly. And also we sort of are doing a somatic call. So we're spitting out a variant allele fraction for your sample. And we're also like have some extra special pneumite filters that are used to sort of filter out anything that looks close to your autosomal coverage. So if you have like 30-ish reads of a particular variant in your sample, it's pretty likely that that actually came from the autosome somewhere, and we just didn't know about that particular pneumite. So that is at a high level what we're doing in the pipeline. Eventually we merge everything, we do some filtering, using filter mutex calls again with our own special set of arguments. And then we're spitting out some coverage information. And that is basically our mitochondrial pipeline. It's relatively fast, it's relatively cheap, and it is convenient, and it fixes a lot of the issues that come up when if you were to just try to run haplotype caller on it. So back to Terra. Did my job finish? The answer is no. And it's thinking. Okay, so it's running. Sure, that's fine. There's a reason, like any good baking show host, it's already been run once before. So we can actually see what the results look like. This is a deterministic tool, so it shouldn't be very hard at all to just look at the outputs. And you can see it's actually very cheap. Again, you're only running mutex over 16,000 bases, so it is a relatively cheap tool to run. And we can look at the job manager, and we can see some information about how this run went. If we were interested, we could view the timing diagram, and we can see how expensive and how long each individual task took. If we wanted to, we can go look at the outputs here, and we can see all the stuff that gets output, but you can see here, we have this output VCF and output VCF index. That is just a set of mitochondrial calls that have been filtered, actually. But if we're interested, there is another thing that I wanted to show you. If you're keeping it home, I believe this is going to be the third mechanism for loading IGV that we will have shown you today, or shown you in this conference. So if we go back here to our data tab and we view our sample and we say, okay, I have these VCFs and I have these BAMs and I actually want to view this data and I don't necessarily want to localize it. Maybe I'm looking at a big whole 300 gigabyte human BAM and it's, you know, messy to try to localize that and use that in IGV. I could just call out to the bucket, but maybe I want to view something really, really quickly. So what do? I go here to select my sample and I say open with IGV. And you need to have be logged in and using your own clone of this workspace to actually use this feature. But now I can just click on actually the inputs I want to use. So let's look at the realigned BAM, this BAM, and this VCF. So let's look at my output VCF and let's launch IGV. And it's going to take a minute for exactly the reason why mitochondrial calling is hard. This is going to be really, really slow. Most of the time, this is faster. And so I don't know if this is going to load immediately. Oh, yes, it is. So let's go to mitochondria. And let's go to a variant that I happen to know is real in this sample. And we can see this looks already at the start a little bit like a standard IGV caller. It is taking it a while to load, however. So let's see what it looks like once it's actually loaded. Again, I can scroll around here. I can do any of the things I normally want to do. I can click on a read. I can get information about that specific read and that specific set of basic qualities. I can go down here. I can click on a call. I can get more information about what it is, about its genotype, about its strand bias table. You can see the depth at this site that Mutect actually used for calling was 10,000. That seems like a lot, but that's actually downsampled from the original depth. If we look at our sample again here, and we click on that position, you can see the actual raw file had 25,000 reads in it. So again, this is like, and this is from a typical 30x whole genome sample. So you get insanely high depth of the mitochondria. And you know, that depth is both a blessing and a curse, right? You could, with that much depth, it becomes relatively easy to do autosomal calling and to look for somatic mutations. Relatively easy to look for somatic mutations just because you have so much data, but you also have to be very careful because, you know, if you're at Q30 for your reads, right, there's a 1 in 1,000 chance that any given read is going to have a base error at any position. Well, if you have 25,000 reads, you expect 25,000 errors at every position. And so this gets really messy really quickly. And again, this is why we have a special version of Mutect that can actually build the assembly graph and be able to actually read data out of it without exploding. So if anyone has any questions, I believe that is more or less all that I have to show you. All right. Is Mehrtash ready? Yeah, so I'm going to hand you off to Mehrtash, and he's going to show you he's going to give you a presentation about the Germline CNV calling.
Awesome. So do you want to hear more about ChatGTK? All right, so let's continue our discussion of copy number variation. Yesterday we talked about somatic copy number variation calling. Today we're going to talk about germline copy number variation calling. Very similar concepts, but slightly different nuances that warrants having two different pipelines. Okay, so the name of this pipeline, in short, we call it gCNV, but it's actually not an official name, so there is no gCNV tool. It's just a shorthand of how we refer to it. There are a number of tools in GATK. The main one is called the Germline copy number variant caller. That's, you know, that's there are a number of tools in this suite of tools, but we just refer to the whole workflow as gCNV. short for Germline CNV. Okay, so just to refresh your memory a little bit, and today I decided that I'll talk a little bit more about the biology of how CNVs are formed, because I realized that yesterday we didn't talk too much about like how we actually get CNVs. Yeah, so just to refresh your memory, copy number variation is loss or gain of copies of any segment of the genome. Like here, you can have one individual, another individual, paternal chromosome, and between the two chromosomes, you can have like different number of copies, right? So here you have in total like 6 copies of that gene. In individual 2 and individual 1, you have like 6 copies. In individual 3, you have 8 copies, right? And these copies could be distributed differently between the paternal and maternal chromosomes. Yeah, for most genes on our autosome, we have 2 copies. So typically, germline copy number variation on the autosome refers to gain of an extra copy or loss of copy or multiple. Okay, so the other thing that is, I guess, important is that CNVs and indels, the boundary between them is kind of arbitrary. So CNVs are just typically larger. Anything that is like less than 50 bases, just call them indels. Anything larger than that, anything between like 50 and 1,000, just can't call them reliably, so we don't have a name for it. But things that are larger than 1 kb, because we can actually detect them with short next generation sequencing data, just call them CNVs. But there's a spectrum of going from like indels to CNVs. So, it's very interesting to know that a big part of the genome, like about more than 10% of our genome, 12% to be precise, they're subject to CNV. It's actually by far more prevalent than SNPs. So the total like dosages of like how many CNVs we have in our genome at germline, not somatic, germline, is actually much higher than the dosage of variation that each of us have due to common SNPs. So many of our like variability in our traits, our hair color, our height, things like that, these are actually controlled by CNVs and not by SNPs. It's very interesting. Yeah, so in fact, it's like 100 times to 10,000 times more prevalent than SNPs. So it's then basically just the motivation that is important to be able to characterize that, because profiling the CNV status of common regions that undergo copy number variation in the genome, it's a good way of it would be a good covariate for, let's say, predisposition of individuals to different clinical traits. Okay, so the other characteristics of germline CNVs is that, and in general, like many structural changes of the genome, not necessarily just CNVs, is that they are recurrent. They're not like completely random in the genome. They're like some hotspots for them on the genome that they tend to occur, and I'll tell you in a second why that might be. Sort of many of the recurrent CNVs, they're found around the regions in the genome which are called LCRs, or low copy repeats. Also, some people call them segmental duplications. But so LCRs, or segmental duplications, these are like stretches of the genome. They're pretty long, but like 1 kilobase. We have many copies of them on our genome. And they're very similar to each other. They're not completely homologous. They're like not like more than 99% identical to one another, but they're like more than 95% identical to one another, right? So you have like these regions in the genome that are so close to one another, and I'll tell you in a second how the presence of those things in our genome is actually a mechanism for generation of copy number variation. So just to take away from the slide is that CNVs are recurrent, they're not random, and they tend to colocalize near certain parts of the genome that are called like segmental duplications or low copy repeats. So there are other places that you get to see like CNVs in addition to the hotspots near LCRs, and sort of those are regions that have microhomology to one another, short stretches of the genome, like maybe like just 10 bases or like 20 bases that are just identical, or like very between like one genomic region and another genomic region. And what happens is the presence of these like microhomologies in the genome messes around with an important mechanism by which the cell repairs damage to DNA. That mechanism is called homologous recombination. And so the presence of these microhomologies and also LCRs just confuses homologous recombination, which is what I'm trying to explain next. Okay, so what is homologous recombination? So homologous recombination, just a refresher, is a mechanism by which our cells repair damage to DNA. So damage to DNA could come in many different forms. One of them is this sort of two-ended double-strand break. So let's just have like a break on your DNA. Okay, so what happens here is that there's an enzymatic resection of the 5' end of the two 5' ends near this breakpoint. And by the way, when you get a double-stranded break on your genome, there are like a host of enzymes that come around this DSP region and make sure that they're held near each other. It's pretty cool, actually. Everything is regulated in our cells. So it's not that, once you have a break, they just, float around. They're like a bunch of enzymes that just come there and say, oh, I'm going to hold you guys together so can repair it. So, okay, so yeah, so you have like a resection of the 5' ends, and then this sort of 3' end that is not exposed. What happens is that it invades the homologous sort of region of the other chromosome. So basically it goes in between the two strands of the other chromosome. Yeah, and the same things happens on the other end. And then what you form is this thing, which is called a sort of a holiday junction. So, and as you can see, basically through a bunch of like polymerase extensions and second end incorporation and ligation, ultimately what you have is that part of the green homolog sort of gets copied to the red, and this is the part that was sort of resected here, and then again you get the other part of the green copied through polymerase extension to the red. So effectively, what you're doing is that by a combination of polymerase extension and ligation, you ultimately copy some of the genomic material from the other chromosome to the chromosome that needs repair, and ultimately you end up in this situation. Now you have to resolve it because now the two chromosomes are like entangled with one another. So either you want to do a crossover, or you want to do some type of a topoisomerase and helicase mediated resolution of a non-crossover type solution, in which case you get either end up with this scenario or the other scenario. Okay, so that's homologous recombination. If the green and the red DNA, if they're identical to one another, this is a perfect strategy for fixing that break, right? Because if they're literally just like two identical copies of the same sequence, we are sort of copying, some missing stuff from the green to the red, and it's just, it's perfect. So we basically, the net effect of what we have done here is that we just sort of fix the break on the red chromosome. So this is another way that DNA, so cells sort of repair damage to the DNA. Just as a quick refresher, this is how DNA actually gets copied. So you basically form this fork. It's called the replication fork, and then the DNA gets unzipped, And as it gets unzipped, then there's a polymerase that sort of synthesizes the second strand over this sort of, this exposed single strand DNA. And then you have this lagging strand, and then there's like Kozaki fragments, just a bunch of crazy enzymatics going on there. But the net effect is that you get this sort of extra copy of the double-stranded DNA is getting copied. This region is called the replication fork, right? So sometimes what can happen is that during DNA replication, that fork can collapse, which means that, for example, you can have like a missing fragment here, or somehow these enzymes might get dislodged. In which case, what will happen is that this replication stops, but it needs to somehow be rescued and needs to continue. So the way that happens is kind of very similar to homologous recombination, but again, the idea is that once you have like this red double-stranded DNA that is sort of getting generated, it's being copied from the green. If the replication stops, now you're ending up with something like this, that you only have like just the one end of the DNA, of the double-stranded DNA. So you do something very similar to what we did before the repair. So you sort of resect the 5' end. Now you have this sort of 3' end now exposed. So this guy goes here and invades the homologous sequence, and then you just generate a new replication fork, and then you continue. And then this process can continue again and again and again. And if every sequence on the genome is unique, sort of this invasion of the exposed 3' end is also kind of very uniquely going to go find its homologue on the green sequence. The problem is that our genome is not like completely like identifiable. There are like many genomic regions that are highly similar to one another. And those are like the ones that I mentioned before, this low copy repeats. Those are basically long stretches of the genome that are nearly identical to one another. So when the cell wants to repair itself, sometimes instead of like finding the exact same position on the correct copy of the DNA, it just goes and finds some other position that is good enough. And that will lead to copy number variation. And here's how it happens. So let's say that this is our red sequence and this is our green sequence. And let's say in the red sequence, we have on the red DNA, we have just named these subsequences like W, X, Y, X, Z. And as you can see, I have 2 X's. And let's say these 2 X's are the genomic regions that are like very, very similar to one another, right? So let's say that I have some error near this X, and now I want to fix that. So what I would like to do ideally is to identify that, okay, I'm like WXYXZ. I need to find another chromosome that is like WXYXZ. And then, all right, I need to align them. And then this is how, this is the correct copy that I'm going to use to fix this guy. But I mean, cells are not that intelligent, right? They kind of like act a little bit more locally. So during homologous recombination, if the error is near this X, the 3' end that is now exposed and it's like trying to go and find its mate to fix itself, it will find anything that is like good enough. And in this case, it might erroneously find this guy, this first X. And now you do form that holiday junction and crossover business, blah, blah, blah. And the net effect is that you get this sequence. Right. So what happened here is that we have like WXYX, right? And now we go to again to WXZ. So what effectively has happened is that now we get 2 copies of Y and we get an extra copy of X. Yeah. So just by finding the right sort of stretch of the genome to use as sort of a template copy to fix an error in the genome, we can gain or lose copy. And the same thing can happen during, when you have a break in the replication fork. It's a very, very similar mechanism. So it's basically all about like invasion of the 3' end of the DNA into the wrong spot in the homologous sequence. All right, any question? I think it's very important to know kind of the biology of how our data is being generated, because then it also like allows us to design better methods for detecting them. Yeah.
They actually identify the cells
Yeah. So what I described here, this is something that is happening every day in our cells, you know. What happens in like more pathological conditions is that these types of like errors that I just mentioned that could happen, just the rate of them might just increase by orders of magnitude. For example, this very important thing that I just alluded earlier is that when you actually do form like these like double-stranded breaks in your DNA, there are cellular machineries that makes sure to detect the presence of these types of error in the DNA. So it makes sure that these two the two ends of the double-stranded DNA are associated to one another, right? So that when homologous recombination leads to a fix of that error, most likely it fixes it in the correct way. During cancer, for example, what will happen is that those cellular machineries might be disrupted, so that when there is a cut on the DNA, then they can be sort of dissociated with one another, and then whenever it gets fixed, it might fix it a very, very terrible way. This is called chromatopsis, for example. You get basically all types of like crazy structural variation happening in the genome, right? So, so this mechanisms that I described here, these mechanisms are part of just natural like cell cycle and life, right? They lead to copy number variation at a very, very small rate, but sometime during more pathological cell states, the same type of mechanisms can get disrupted a little bit further, lead to generation of lots of copy number variation, which are kind of like the scenario we discussed yesterday.
But then additional are actually consequent these damage was very
That's a great question. It's a nice research problem. So yeah. This is not something that we use in modeling, but that's definitely like the interesting type of question to ask. Okay. Any other questions? Okay, cool. So okay. So with that, a little bit of a biology intro. Let's just just frame today's conversations in relation to yesterday's conversation. So let's refresh our memory about what is germline, what is somatic. So germline copy number variation is a type of copy number variation that originates in germ cells. So these are like sort of the type of CNVs that we inherit when we're born. The way we detect them is by doing genome sequencing on small and healthy tissue samples. Let's see, just a tiny piece of the lung. We just take that biopsy and then we do whole genome sequencing on it, or just some blood draw or something like that, right? Germline copy number variations are very rare and very small in relation to somatic copy number variation. So if you remember these sort of copy ratio profiles that we were looking at yesterday, this is another type of thing that you expect to see for a germline sample. Like most positions are sort of copy neutral, and there might be just a few small genomic targets that might have like some gain, just some loss, right? And this must be contrasted to somatic copy number variation, that you have like a lot of copy number activity. Yeah. The other major difference between germline and somatic is that in germline, the main assumption is that your cells are isogenic, right? Which means that they all have the same genome. if you just take a small specimen of some of any tissue from a healthy individual, odds are that basically all of those cells, they're like almost like high fidelity copies of one another. So they all have the same genome, which means that sort of if you take any genomic regions, you have like an integer number of copies of that gene in the whole sample, because every cell has an integer copy number, and if all the cells in your sample they have the same genome, then in total you also get like an integer like duplicate of the same copy. Yeah, and then this must be contrasted to the somatic case, something like cancer, that you're dealing with a very heterogeneous cell population, right? You're dealing with like multiple subclones. They're competing with one another, and each of them they have their own genome. And one of the subclones might have a certain copy number profile, the other subclone might have a different copy number profile, and then in your whole genome sequencing data, sort of you're getting a mixture of DNA from all of those cells. So that's why you get sort of this very complicated copy ratio profiles that are not necessarily integral. Yeah. So that's the other thing that we're going to use heavily in our germline copy number calling pipeline. The assumption that basically the events that you're looking for, they have integer copy ratios 0, 1, 2, 3, 4. All right, so Germline CNVs are also of major clinical significance. Whatever doesn't kill you doesn't necessarily make you stronger. So they're linked with cancer risk, and just to see how clinically significant they can be, these 10 papers, each of them discusses a rare disease. They all came out during 2021, 2022 by a single publisher. So you can just pick your favorite publisher and just search for like germline copy number variation in rare disease, and then you find 10-ish papers. So every year, 10 rare diseases, from any one of the major scientific publishers, you will find like evidence of association between germline CNVs and rare disease. And there's a reason for that, right? Because if an individual survives loss or gain of a copy of a gene at birth, something should go wrong, right? Most likely, because most of our genes, they just cannot tolerate loss or gain of a copy, because things are generally very heavily regulated in eukaryotes. Yeah, and this is looking at germline CNVs is also a very useful way of understanding gene function, because rare diseases, they do not typically affect all of our organs. They typically affect like a few of the bodily functions. So by identifying the association between rare diseases and 1 or 2 genes that might be involved in the rare disease, and the phenotype of the individual that has a rare disease, we can learn something about the function of that gene. So that's the other sort of significance of studying germline copy number variation. It's just, it teaches us about how we function. All right. How do we detect germline CNVs from the lens of short read sequence? This is just another way of viewing the things that we've already seen yesterday, but doesn't hurt to see it again. So let's say that on the top, this is sort of the actual genome, the genome that we have sequenced, and here in the middle we have the reference genome. And let's say that we have a duplication going on here. Instead of 2 copies, we have 3 copies. We have a deletion, homozygous deletion going on here. We have another duplication here. We have the heterozygous deletion here, right? So now if you just sample short reads from this genome, what's going to happen is that here we're going to get a little bit more reads in the region that we have duplication. Here that we had a deletion, we're not going to get any reads. Here that we had like a heterozygous or like hemizygous deletion, we're going to have like half as many reads as usual. So if you take all those reads and align them to the reference genome or haploid reference genome, so what's going to happen is that here that we have like 3 copies, we expect to get sort of 3 times as, well, 3 portions when, in the regions that we have, 2 copies of every gene, you're going to have like 2 portions. And, the region that we have deletions, we're going to get see nothing. And then if we turn that into some type of a, coverage profile, you're going to see something like this. Duplication is going to be up, and then we're going to go back to baseline. Deletion, going to go down. Going to go back to baseline. Duplication up, baseline, deletion down, baseline, something like that. And then once we find something like that, we just segment this. We identify copy number variation events. The problem is that this is how the data looks like in reality, right? It never looks like something like nice and simple, like what we expect. So it's from something like that, I just can't see anything really. And that's like all the issues of like noise and bias that we discussed yesterday. It's present both in somatic copy number variant calling and in germline. It's particularly more important to deal with them properly and with mathematical rigor in the case of germline CNVs, because germline CNVs are very small and very rare compared to somatic CNVs. So detecting them requires extra careful denoising. All right. The key insight here is that we can't do anything but just looking at one sample. If you just look at one sample, and let's say this is sort of the read depth or depth of coverage or read count, these are all like synonymous words. Of a single sample, you're gonna see some, something like that. This is just a color-coded version of the same thing that we had down below. You're not gonna see anything. But if you look at many samples simultaneously, then you might start to see some interesting patterns emerging. For example, if you just look at this genomic region across many samples, here I'm showing samples on rows and genomic regions in columns. You might actually see that there is some type of like an activity going on here. You see, so maybe that's a CNV hotspot. And maybe you can see that, okay, this genomic interval and this genomic interval, they're kind of like extra noisy. So maybe you want to ignore them. And then you might also see that some rows of this sort of matrix, they're just too noisy. So maybe those are just bad samples that you want to entirely ignore, right? So it's really by just putting like many samples together that we can start to learn the structure of our data. By looking at one row of this sort of matrix, you just can't say anything. And that's really at the starting point, our starting point. I'm going to skip this slide because we already covered it. And let's get to this one. So, all right, let's say we have observations from many samples. How do we actually use that to learn the underlying biological process that has been has dictated data that we have collected for each of this data, for each of these samples. The way that I'd like to propose that we look at this this problem is to think of data generation as a physical process, right? Instead of just looking at data as just a bunch of numbers that we get, it's very important to understand how this data is generated and how this BAM file that we have is ultimately connected to a cellular process. Because it's really by following every step of this journey from, let's say, the phenomenon of interest, which is in this case, let's say, copy number variation, to something that we observe, which is that coverage profile. It's really by like going the steps that connects these two. That is that we can take steps back and connect our observation to the phenomenon of interest. So there are many things that happen that connects these two. Remember that we had all these library generation, and during generating library, we have a lot of enzymatic reactions. Then we had this flow cell that we did like clonal amplification. And then we got a bunch of reads, we aligned them. So there are like so many different components at play that ultimately connects our observation to the original biological phenomenon of interest. So we need to model them. We need to sort of have an understanding of the effect of every one of these components of this game. So if you know, here's another like pictorial way of looking at it. So if your data is this guy, this red guy, then so that the phenomenon of interest is this red guy, then you have a bunch of these black guys, and they come together to make this yin yang, type thing. And then in order to go from this sort of convolved like signal and noise to just pure signal, we have to be able to like simultaneously model both signal and noise. Okay. So modeling copy number variation is actually not too difficult. So there are 10 assumptions, and these 10 assumptions they're only natural. So the role of us as like model developers is to sort of figure out these underlying data generative process, the assumptions that are reasonable, that dictate data generation, and turn them into mathematical models. And that's this what I'm trying to do today. So the first assumption is that read count is fundamentally subject to Poisson noise. So what do I mean by that? What is read count? Read count is like how many reads that you basically get from your sequencer and you associate to any genomic interval, right? It's a random sampling process. You have this library. The library has like many fragments in it. You basically just take a bunch of your library, you put it in the flow cell, and you sequence it, right? So it's almost like a random sampling process. Because of that random sampling process, it's subject to Poisson noise. Then some samples and some genomic intervals, they're just noisy. There's no way to deal with it. So some samples, they're just not prepped properly. Some genomic intervals, they're just not very mappable. They just confuse our aligners. So we have to also appreciate that. There's going to be like bias in sampling of the genome, but the key assumption here is that there are a handful of hidden variables, just a handful of them, that dictate the bias of every single genomic interval. So you can think about that, there is like GC content, like what fraction of the nucleotides of any genomic interval is G's and C's. Accessibility, like if it's in the centromeric region or it's like in the non-centromeric region. Mappability, is that sequence like very similar to other sequences or like very distinct, very unique? So for example, let's say these three are the three knobs that sort of control the amount of bias, and each of these factors contribute to different genomic intervals with different weights. So our job is to learn the identity of those unknown latent sort of bias factors, and also to uncover how much each contributes to the bias of every genomic interval. Then I covered that. Yeah, and then of course, the read count of any genomic interval is sort of proportional to its integer copy number. The more copies you have, the more reads you get. Then the typical size of a CNV event is about 10 kilobase. That's an empirical observation. De novo CNVs are extremely rare. We have about like 1 per genome. But common CNVs are very common. We have about 10,000 common CNVs for any given genome. And finally, about 90% of the genome is copy neutral. So about only 10% of our genome undergoes copy number variation. So these are the 10 assumptions. The next steps is just to turn these 10 assumptions into a mathematical model. which we use to then apply to the data and learn stuff from the data. Please, yeah.
One element which from 4 power chemistry.
Yeah. The GC bias is as far as I know, it only entails the PCR. Yeah, so I wouldn't think that it color chemistry has anything. Okay. This is gonna take twice as long as I thought it would take, so I'm gonna speed through. So all right, so before we get into the math, which I don't intend to get too much into the math, but let's just look at the bird's eye view of the workflow. It's very similar to the somatic CNV workflow. The first step, as usual, is just coverage collection. We just have a collection of samples. We look at the read counts. We collect read counts from the BAM files over previously specified genomic intervals of interest. We filter some of these genomic intervals. That's sort of equivalent to the pre-process interval step that we had in the previous pipeline yesterday. We get rid of intervals that involve extremely unmappable targets or segmental duplications or things like that. Then if we have a large cohort of samples, maybe tens of thousands of samples, we cannot process all of them simultaneously. So what we do is that we try to cluster samples and identify batches of samples that we can model simultaneously. So this has 2 effects. One is that it actually allows our pipeline to run, because our pipeline just cannot be run with 10,000 samples simultaneously. And the second thing is that by just grouping samples that are similar to one another in terms of their read depth characteristics, if you have, for example, a cohort of samples that uses 2 different capture exome sequencing kits, you probably want to process samples with one exome kit and the other exome kit sort of separately from one another, because they have very, very different characteristics. You don't want to mix them. So that's the other thing. The other step is to determine sort of a baseline ploidy for each of your chromosomes. So you might have some aneuploidy events that you want to be able to catch, and then you also want to do some sex genotyping. You want to know how many copies of the X chromosome you have, how many copies of the Y chromosomes you have. So basically, you want to determine a baseline copy number for every chromosome, on the top of which then you want to determine sort of focal events. And the final step, this is sort of the main step, and that's where sort of once we get our read depth profile, we do this sort of iterative step of learning some factors of noise and factors of bias, and equipped with factors of noise and bias that we've learned from our data, we do copy number variation calling. And once we've detected copy number variance, then we go back and revise our belief of what is signal and what is noise, and we keep doing it again and again and again until we converge, right? So the key sort of innovation of GATK gCNV is exactly that step. That instead of, let's say that I want to denoise my data, and once I denoise my data, I want to call copy number variant events, we are admitting to the fact that you cannot do this in a 2-pass step. You need to denoise, assuming that some understanding about copy number events, and then once you denoise your data, you call copy number variation, and then now that you know your copy, you have a better idea of what might be your copy number events. You want to go back and revise the way you denoise your samples, and then you want to keep sort of looping back between the two until you reach a self-consistent sort of state of belief. Okay. So the way we do this, so the way we sort of turn this assumptions into a mathematical model is a process called generative modeling. And this is a quote from Richard Feynman. Says here that what I cannot create, I do not understand. That's really the heart of generative modeling. It's an approach to building probabilistic models, converting mechanistic insights that we might have into mathematical models. And it's really, through creating your data from scratch is that you can sort of take your data and step all the way back to the biology. That's really what I'm trying to say here. Okay. So the generative modeling approach in gCNV is as follows. We start with our genomic, with our genome, and we ascribe to every genomic position, a genomic interval class. So it could be either like an active class or it could be a silent class. Active means that it's a hotspot for copy number variation. Silent means that it's not a hotspot for copy number variation, but it can harbor de novo copy number variation every once in a while. Okay, now that we have sort of attributed, active versus silent to every genomic interval class, from this I can generate a matrix of copy number variation events for all of my samples. So let's say that this is my genomic interval, genomic intervals, and these are my samples. So now I can sort of randomly generate copy number variation events. So on the region that I have like active, I just generate a lot of copy number variation. On the regions that I'm like, I'm sort of silent, every once in a while I just generated a small event. Now, the next step is that connecting copy number variation to read count data. So this is the process of like bias and noise and how, read depth is ultimately connected to copy number variation. So this is called the forward process. You start with the top of the hierarchy with the genome. And from the genome, you generate copy numbers. From the copy numbers, you generate read counts. Now we need to learn to inverse this. Sort of go from read counts to copy number, and from copy number to the type of the genomic interval class that we have. And then we just go back and forth between these three steps. And that is called like generative modeling. So so the next three slides, they have a lot of math. So please feel free to, if you can follow all the better. If not, I'd be happy to, meet with you after and, go through them. Or if you just, not interested about this, just do something. Okay, so how do we model this copy number states? So we start with our genome. We have our genomic intervals. We just refer to them like, tau 1, tau 2, tau 3, tau 4. And then so the first genomic interval with a certain probability, P active, we call it like an active region. With 1 minus that probability, we call it a silent region. And then the thing is that since copy number variations, and just things that happen on the genome, they have like a typical characteristic length scale. If one interval harbors a lot of copy number variation, it's very likely that the next interval and the previous interval, they also contain copy number variation. So we want to like sort of model this type of like correlation on the genome. And the way we do it is using this thing is called the Markov process, or sticky Markov process, that there's a characteristic length scale that we specify, and then to sample the class type of the next interval, you do it conditional in the previous class. If they're close to one another genomically, and if this is like active, the next one has also is more likely to be active. So that this is how we sort of induce correlations between different positions in the genome. Now, given our determination of the type of every genomic interval, now we go to our samples. Let's say you just look at our first sample. So our sample, the first sample, This is the copy number in the first genomic interval, copy number in the second genomic interval, third genomic interval, and so on and so forth. And we have like hundreds of thousands of these nodes. We do the same thing here. So first of all, we just look at the genomic interval. If it's an active genomic interval, we allow ourselves to use any copy number event for the sample. If this genomic interval is a silent genomic region, we most likely would want to choose the baseline copy number state, which we determined in a previous in one of the steps of the pipeline, right? So, for example, if it's an autosome and there's no aneuploidy, we probably most likely for this position we want to choose like copy number 2. With a very, very small probability, we can allow ourselves to choose other copy number states. So this process is also sticky because, like I said, copy number events, they have a certain length scale. And if, let's say, these two intervals, they're like within 1 kilobase of one another, and the first interval has, we have determined it to have a copy number event, it's very likely that the second interval also has a copy number event. So again, we model that with another sticky Markov process. And that's how basically can randomly generate copy number events. It sounds a little bit funny because it's saying, why are you randomly generating these things? Don't you want to like infer them from the data? That's how generative models work. First, you just come up with a game of how you can randomly generate data. And then now you feed the actual data to the model and you try to invert the process. Yeah. So now I'm just talking about the first step of how we can randomly generate data. Okay, and the last step is how to go from copy number matrix, for every sample and every genomic interval. We have now a copy number state. Now we want to be able to connect it to the read count matrix. So mathematically, I have my read count matrix, and then I have my copy number matrix. And so I want to learn how they're related to one another. So this is another sort of relatively complicated mathematical model. So let me just tell you that there are 3 pieces to this. One is how we want to model sort of sequence biases, how we want to model extra noise that we just cannot model. Sort of basically how we want to admit that some noise exists that we cannot model. And then also the fact that different samples have different sequencing depth, and then you have also mapping error. So we need to model these 3 aspects of the phenomenology. So, I said initially that the read count is subject to Poisson noise, but since some samples are like noisier than others, we add a bunch of additional noise. We model as a Poisson gamma process, which is also called the negative binomial process. And then the rate of this process, lambda, it's composed of like a couple of different things. So different samples can have different read depth. There's, of course, a copy number state. There's a capture bias. There's mapping error. So it's a combination of these 4 factors that tell us what should be the expected sort of rate of read count. Yeah, and then some samples are extra noisy. Some genomic intervals are extra noisy. So the amount of over dispersion that we add to our noise process is a combination of both sample level and genomic interval level noise. And these are learnable parameters. And ultimately, this bias, which is a very important piece of the model, which every sample and every genomic interval is subject to some bias. And that bias is a combination of some average bias and then some learnable sample-specific bias. And these are like how, for example, GC contents contributes to bias, how mappability contributes to bias, fragment length, and things like that. So every source of bias can have a sample-specific contribution of the bias of any given genomic interval. Okay. All right. Okay, so at this point, hopefully I roughly motivated how we generate the forward process of GATK gCNV, how you can just randomly generates copy number variation events, and then from those copy number variation events, I can randomly generate read depth. But of course, nothing is random. We observe the read depth. So our goal is to, given that observed read depth, how can we go back and infer copy number variation event? And that is called Bayesian inference, which I'm not going to cover today, but there is like some well-defined mathematical procedure that you can take any generative process, condition it on observed data, and then use it to infer the underlying variables that you have introduced in your modeling process. Of key important for for us, just that very specific random variable that we introduced in our model, which is the copy number matrix. Okay, and if you're interested in just learning about like that step, we have detailed it in our preprint here. Excellent. Okay, so I'm done with the hard part. So GATK, you can run it in two different modes. One is called running in the cohort mode, and then the other one is called running in a case mode. The cohort mode, as the name suggests, is that you have, let's say, a cohort of samples, 100 samples, and then you want to simultaneously call copy number events on those 100 samples, and also like model the noise and all the things that we discussed, right? The case mode, as the name suggests, is that if you have already run GATK gCNV in the cohort mode, and now you have a fresh sample, and you want to call copy number events on that single sample, you use the output from the cohort mode to basically just do calling on the single sample. It's kind of very similar to making a PON, right? This is a panel of normals type thing. Except for that, this running GATK gCNV in the cohort mode just doesn't create a panel of normal for you. It produces some model parameters and also copy number calls for the cohort samples. Okay, so you start with the intervals and the BAM files. You give your intervals to this tool, which is called like pre-process intervals, as the name suggests. It gives us like padded and binned intervals and just make sure that the intervals are high quality. You give your BAM files to collect read counts. It's the exact same tool that we saw yesterday. And then if you have like multiple samples, you just invoke this command multiple times to collect read counts for every one of your samples. And then let's say you can visually put the read counts of every one of your samples together into the form of this heat map, which is sort of a matrix that you have genomic interval on the x-axis, and the y-axis you have samples, different samples, and then you're showing the read depth sort of as a color-coded heat map. The next is that you can take sort of your read count collection, like the read counts over multiple samples, and then you can annotate your intervals further if you want, by GC content mappability. This is by the way an optional step. And then filter your intervals. So why do you want to do this? Well, if you have a cohort of 100 samples and then you find that there are some genomic intervals that have very, very small coverage, or like there are some genomic intervals that have a lot of GC content or like extremely unmappable or something, you probably want to throw them away from, right off the outset. So that's why after collecting read count, you want to go back to your intervals and maybe you want to do some filtering of the intervals. All right, now that you have your filtered intervals and then you have your read counts, then you want to determine a ploidy for every contig. That's sort of the baseline copy number of every contig. It's not that the main process. This is just for saying that, okay, this autosome has copy number 2, that autosome has copy number 3 if you have an aneuploidy, and that sex chromosome has copy number 2, something like that. So just a contig level baseline copy number for every contig. And here's the main part. So once you have determined a baseline copy number for every contig, now you want to go back and you want to sort of call those like smaller focal copy number events. And that's the main tool that we have, which is called the Germline CNV caller. You probably don't want to run it on the entire genome in one go because it takes forever. You probably want to shard the genome into smaller pieces and then run many of these tools, copies of the tool in parallel. So long as these shards are like long enough, but like 10 KB, 50 KB long, you're good. You can do this sort of parallel processing. So in practice, that's what we do. You can also like parallelize this both across like samples and genomes. Sort of if you have like a huge cohort of like hundreds of thousands of samples and something like that, you probably want to like do a bunch of parallelization. Okay, so the output from Germline CNV caller is basically the inferred model parameters, the biases of every interval, the noise of every interval, the noise of every sample, all those parameters that we introduced, as well as those small copy number events that you might have here and there. Awesome. The final step is that once you run all these tools, there's this nice tool called post-process Germline CNV calls that takes the output from all these tools and produces a nice VCF file. You can just look at that VCF file. Cool. Now, running things in case mode is simple. So you just need to reverse some of these arrows, right? If you've already run the gCNV in the cohort mode, then you just use these model parameters, you give it as input to Germline CNV caller. If you've already filtered your intervals, you give your filtered intervals as input. If you've already sort of done determine Germline contact ploidy, you just give them as input, and single sample comes in, collect read counts, determine its ploidy, you call copy number events, you post-process your now. It's a much simpler pipeline. So our preferred approach to run gCNV both in case mode and in cohort mode is to use Cromwell and Whittle and Terra. Just a single workflow. It takes a bunch of BAM files to give you VCF files. So you don't have to run all these tools like separately for yourself. It's so much easier. gCNV in general produces a bunch of output, but I'm going to go through these outputs in the upcoming demo session. So let's just not discuss this just yet. So the main output that we care most about is that VCF file, and it's just like any other VCF file. It's just zoom in into one of the records. You have, per usual, the chromosome number, position, some ID for that variant, the reference, alt, quality, blah blah blah. A very important thing that we have here is a number of quality metrics: QA, QS, QSE, QSS. So the last three are the most important ones. QS basically just gives you an overall Phred scale quality for determination of copy number for that segment. QSE is a quality for the end of that segment, like how certain you are that that's where the copy number event has ended. And QSS is a quality for the start of the event. So if you care about how certain you are about the beginning and the end of the copy number event, those are the two qualities you want to look at. If you're in general interested to know like how confident you are about making this copy number call to begin with, you want to look at QS. And in general, like as a rule of thumb, QS is the most important quality score to filter on if you want to do some filtering. Without any filtering, gCNV should produce about 10 CNVs per whole exome sample. And these are like 10 rare CNVs, I should mention. By default, we recommend that you just get rid of all of your segmental duplication regions. You just not call common CNVs with gCNV. We haven't done proper tuning of the parameters of gCNV to actually be able to call common copy number events using it. So the current main use case of gCNV is to use it for detecting rare CNVs. So with the current parameters, it only detects rare CNVs, and you should get about 10 of them. If you see 200 or 500 events for your sample, probably that is a QC fail. Somehow something didn't work. You might ask that why a sample might fail. Well, it could be that it's just like an outlier in your cohort. You know, you have given gCNV a cohort of 200 samples, and that single sample might have used a different exome kit. Yeah, so it's incompatible with the rest of the samples in the cohort. That's why you shouldn't trust the results that you get on that. So the idea is that if you want to run gCNV in the cohort mode, exactly like the somatic CNV pipeline, you want to make sure that you choose that sample cohort to be as homogeneous and uniform as possible. We have some guidelines of how to filter if you're interested in clinical precision, more than 95% sensitivity, more than 97% specificity. If you want to only keep the super high quality events, that these are how you should filter your VCF file. I mentioned those things. Let me also mention this in passing, that if you have a very, very large and heterogeneous cohort, comprised of thousands and over thousands of samples, what you want to do is that once you do your collect read count step, you make that, you generate the read count for every sample. You probably want to do some manual clustering of your read count matrix. Just generate smaller cohorts of samples based on the similarity of their read counts, and then run gCNV on each of those smaller cohorts. Runtime and cost. So it's actually not too expensive. Running gCNV the cohort mode with 200 samples, it takes about 9 hours wall clock time, but you're using 14 parallel compute instances, and it costs about 4 cents per sample. Running in case mode is a slight bit, it's about 50% cheaper, 40% cheaper. It costs about 2 cents per sample, and it takes about 8 hours. But it's not much faster, it's just cheaper. All right, and finally, how does gCNV compare to other tools that are in common use for copy number variant calling? In terms of, okay, so generally, the bigger the event, the larger the copy number event, the more accurate, the more confident we are. So what we're showing here is that here on the y-axis you have recall, on the x-axis you have the number of exons that spans that copy number event. This is for whole exome sequencing data that I'm showing here. And in general, for all the tools, the larger the event, the higher the recall value. It just kind of makes sense. A huge copy number event is just easier to call. In terms of recall, gCNV is tied with basically 3 other tools, SumDep and XHMM. Where gCNV sort of is separate from other tools is in precision. So it's about 20 to 30% more precise than the other tools. And that's really the main advantage of gCNV. So if you're really interested in making actionable clinical calls, that's where I guess gCNV could be extremely useful. And this is a very, very careful evaluation using 8,000 exomes in quartet families. So we have mother, father, and 2 siblings. So it's very, very we have many ways of validating the CNV events that we detect, so that we can come up with these accuracy figures. Excellent. Okay, so that's the end of my. Oh, please. Yeah. So there is a tutorial that I will point you to. It's definitely today it's definitely possible to do that with whole genome sequencing data. So if you have whole genome, you want to call then you're in good shape. With exome, it's a much more challenging. The reason that we don't support calling copy number variant calling on exome data is that exome data is so noisy that disentangling actual copy number variation on copy number hotspots, which can show actually a lot of copy number variability, can have anything from 1 copy to 15 copies. Distinguishing that from actual noise is even like mathematically impossible. So that's why, you know, we don't currently know how to actually tune the parameters to be able to call common copy number variation from exome, but for genome, we do have a set of parameters that work.
As this kind of variant derivative particle, is it worse to use the exome data to whole genome to only target the function of the events?
It is like many of the rare Mendelian diseases, for example, they involve copy number variation of the protein coding part of the of the genome. So if you do have genome data, it's definitely, you know, why not? You know, also in addition to like usual, like, you know, SNP calling, you know, short variant calling, you know, why not also do like copy number variant calling? But if you're on a discovery campaign and, you know, you still haven't designed your experiment, of course, if you have the budget for that, it's best to do like whole genome so that you can see the entire copy number variant. All right, let's take a 10-minute break. Before we do the, and we're going to do the guided demo. If you want to do it as an exercise together with me, you need to do the steps that we did yesterday again, unfortunately. So that is basically you need to go and this time go to that same workspace as before, which was the 2023 Cambridge workshop thing, and open this time 09 GATK Germline CNV tutorial, and then basically grab these commands and then run them on terminal. So let me just do it live. So just copy and then open the terminal. And run. If you're asked questions, just answer yes, yes, or all, something like that. One very annoying thing about Bash is that if it asks a question from you that you have to like give you like yes or no, it stops running the rest of the commands. So in this case, I think since you've already downloaded GTK, the unzip asks a question from you that you want to overwrite or not, and this sort of causes the rest of the commands not to run. So you probably want to make sure that, you know, just everything runs. So just copy this again and just go back here and just run the rest. It's faster for me because I've already done a bunch of steps. All right, so guys, on Bash, we need to just type history. It tells you like the history of all the commands that you've ran. So make sure that the last thing that you've run is this Python -n ipy kernel install blah blah blah. You must have reached that point. That's I really want to make sure that if you run all of these commands, because it has bit me a couple of times that I just like copied and pasted it, and then just put it on the terminal, and then for some reason not all of them got run, and then yeah. But
You want to use Terra in the future, we need to install everything every time? Or how does it
we will fix
exactly work?
this. I mean, even this stuff.
You normally don't.
Yeah, you normally don't need to. This is just for a couple of tools that uses Python, and unfortunately something got broken in
Okay.
the Docker image. We're actively working on fixing
Okay,
it.
okay.
Yeah,
No.
yeah. All right, so if you've gone through this painful step again, you can return back to your Jupyter notebook, refresh, and you should have a new kernel exactly like yesterday. This conda and GATK. If you don't have that, something has gone wrong. So who doesn't have it? And who is still like working on things? 1, 2, 3, 4. Is there an issue, or is it just like doing its thing? Doing its thing or issue? Oh.
Is that a problem? Was that a problem? I moved on, but
Oh yeah. So mind if I
yeah.
so you probably we can just maybe stop this. So if you look at history,
I had a lot of yeses, and then
you had a lot of
yes,
yeses. So you see, you unzip this guy, and then, but some commands got skipped.
right.
like-
Okay, so something got jumped over. I was going line by line, but I must have
Yeah,
I must not caught up there. I think that
this conda and creates. That's the one that you should be good at this.
right. Okay, thank you.
No, you're not good. So did you refresh? Refresh this. Mhm. No, you're good. Okay.
I did this, which all worked,
Mhm.
but then when I go back, now my Terra page is just like this.
Oh, beautiful. Can you refresh again?
Yeah, I have, and then it made me like reload. And then every 10 minutes. But this bit worked. Yeah, no, it's just.
Can you?
Oh, like closing down. and stuff.
Yeah, just go back to your like to the Terra website from scratch. Yeah, like log in here. All right, analyses.
Does it just work now? Is there
It
anything?
looks like it. Mhm. Arnold. Yay! I got it.
That one,
Yeah.
I think it was all that already.
I had no idea what happened. Yeah.
It's just.
Refresh, restart, you know. All right, guys, let's get started. Any other one still waiting? You good? You? Okay.
Okay, I think it was all there. No, it's still. It's
Okay.
okay.
All right. Well, some of the steps are actually kind of like long, so hopefully for those of you who might be lagging behind a little bit, you can catch up. But for the interest of time, allow me to continue. Okay, so today we just want to run GATK, gCNV in the cohort mode and the case mode. The point of this exercise is just to run the tool and maybe just look at some of the results, but we just don't have time to go deep. Like you can do all the things that you did before. You can take these VCF files, you can take them to IGV, you can look at the events, you can inspect the events to see if they make sense. You can do all those. And the tutorials that I've linked, courtesy of Derek, that basically just go through those additional like steps after doing the CNV calling, just loading things up in IGV and looking at them, and then doing quality control, filtering, and all those things. So highly encourage you that if you're interested in this topic, you want to do germline CNV calling in your work, just also additionally go through those other tutorials, and also maybe take a look at our preprints if you're interested in more of the details. Okay, so I just check my kernel. I'm in good shape. I selected Conda and GATK. So I should be good. You start from this cell. You run it. This is the usual stuff, just setting up some global variables. And now making some folders on the local virtual machine. Going to 1.5. We're listing the contents of that workshop bucket. There should be one tutorial 11684 RGZ file. You're gonna go down to 1.6, download that, decompress it, and set this tutorial data path variable to where we decompressed it as a shorthand that we can just use it in the next commands. Everyone with me up to this point? Okay. Let's go to section 2, and that is the part that we should be familiar from yesterday's tutorial, and that's to do the pre-process interval and collect read count. So for whole genome sequencing data, since we have more or less like even coverage in the genome, we probably want to do copy number variant calling everywhere because we can. So that's if you're dealing with whole genome data, you probably want to like run this pre-process interval command. What it does is that it takes your FASTA file, your reference genome, and by default, it will chunk it up into 1 KB sized bins. And those are the intervals that we would be using for doing copy number variant calling. You can go all the way down to like 200 base pair if you want, to have like finer resolution, but you just have like more intervals to process, and just running gCNV takes longer. So it's sort of the trade-off between resolution and breakpoint sensitivity. For whole exome data, what you want to do is that you probably want to take the exome capture intervals that you might have, and then maybe pad them a little bit by 250 symmetrically like yesterday, and then just use those intervals. For this tutorial, we're doing neither. So for expediency, we're just only looking at chromosome 20, chromosome X, and chromosome Y. So we have that as a list, as you can see here, chromosome 20, chromosome X, chromosome Y. And you can give that list as a -L inputs to pre-process intervals, to basically just you're telling this pre-process intervals that those are the contexts that I want to be looking at. Give me like bin length of 1,000. Don't do any padding. You don't want to do any additional padding of these. These are like already like head to tail to one another, these intervals. And you want to create this interval list. So this should take about a minute or two, mostly because the genome reference is on the cloud and it's not local. So it basically needs to read that from the cloud. Second.
And can we just increase that? If we want to just change it So whether you
Absolutely. Yeah. 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 All right, while this runs, let me just give you some comments on some of the parameters of preprocessed intervals. So, like I said, for whole genome sequencing, you want to bin the genome, and the default value for like the bin length parameter is 1,000 kilobase, 100,000 base pairs. So you can increase it, decrease it as you see fit. For exomes, you want to set the bin lengths to 0, but you definitely want to give you the interval, the intervals of your capture kit as a -L input parameter, and then you want to pad them a little bit to make sure that you don't have any like tiny intervals. Because if you just get like this capture interval list from whatever kit that you're using, it definitely contains a whole bunch of like tiny intervals, like 20 base pairs, 30 base pairs here and there. And then collecting coverage over those tiny regions is just going to be too noisy. You're going to like pad them a little bit. You can also exclude intervals if you want by giving like a -XL, like centromeric regions, segmental duplications, or like whatever regions that you're just not interested in. It's taking longer than I expected. All right, it's done on my end, so hopefully it's also done on your ends. So let's just look at what we got. Big rep, we just skip the headers and they just print the top 10 intervals. And as you would expect, you get like 1,000 nucleotide bins of chromosome 20. And if you look at the tail, you should see like something in chromosome Y probably, right? Yeah, so it works as intended. Okay, now that we have our pre-processed interval list, we just go to collect read counts. We have our BAM file here. It's one of the samples from the 1,000 Genomes project. And this is exactly like the command that we used yesterday. It is just going through the BAM file, co-localizing reads with each of those intervals and just counting them. That's it. So pop quiz. You're doing paired end sequencing, right? In paired end sequencing, you have one read here and another read pair here, like they're pointing to each other. Now, let's say you have like an interval, which is right in the middle of the two ends of the paired read. Would you want to count that? Or do you want to sort of give nothing to that interval? See what I'm saying? I don't have. I can draw that for you, but imagine that you have like you have a long insert, yeah, 600 base pairs. You have first in pair and second in pair here and here. Is this writable?
No.
Oh, this? Cool. Can I just write on the wall? Okay, this scenario, right? This is, let's say, left and right read. Let's say you have like 100 base pairs here. You have 100 base pairs here, and of course there's this like, and this is the part of the genomic fragment that we have not sequenced, because in paired end sequencing you're not reading the whole fragment, just reading from left and right and left. And let's say this is the interval that you want to like do coverage collection over, So would you want to, for this specific read, would you want to say that this gets like 1 count, 2 counts, 0 counts? What do you think is the right thing to do? One count. Okay, so it's not very clear, how you want to answer it. My own thinking is you should give it 1 count. Yeah, the reason is that if the mapping is right, if everything is working correctly, we actually have evidence that such a fragment was sampled from the genome. So it should contribute to the coverage of any interval that overlaps with this fragment. You're not doing that in practice. In practice, what you're doing is that you're actually breaking paired end reads into like 2 reads. And for an interval here, we're giving it no coverage. Now, if you have an interval here, this contributes to this interval. If you have another interval here, this contributes to that interval. So we're kind of like doubling the contribution of a fragment. We've actually evaluated it, like what's the impact of different strategies of read count collection, fragments level, read level, and all these things. Didn't have actually any impact on the results. So theoretically, it makes sense that you want to do something like the first, you want to look at genomic fragments as opposed to independent reads, but in practice, it didn't have any effect. All right, so we've collected the read count. So if you just look at it, it's kind of like the thing that you would expect. It's a TSV file. You have your intervals, and then you have like counts, how many reads overlap with that interval. Okay, beautiful. Now what we're going to do is that we're going to go to section 3.1, and we're going to run this tool called annotate interval. Annotate interval, as the name suggests, just adds a bunch of collects a bunch of additional metadata for each of the intervals, like what is the G and C content of that interval, like what fraction of base pairs overlapping with that interval from the reference genome, mind you, not from your reads. Yeah, and then in general, there are other annotations that you want to maybe bring over, like if you have mappability, there are actually some. We actually have references for those in this tutorial, maybe further down below. There's this website, UMAP and BISMAP. They're just like very nice tracks along the genome that tell you about the mappability of each genomic region. You also have tracks you can download from UCSD Genome Browser about segmental duplications, signs, lines, low complexity regions of the genome. So in general, you can imagine that you can add a lot of metadata to each of your genomic intervals. And the more of this metadata you collect, the better, because you can use them for filtering. If a region is extremely unmappable, you probably want to exclude it from your analysis. A region has a exorbitant amount of like G's and C's, you probably want to also like exclude it from the analysis. So that's the reason that we're doing this exercise of annotating our intervals. So by default, you're only like looking at GC content. That's one of the more important criterion for filtering.
Blacklisted regions, is this methodology similar?
Similar to that, yeah, yeah. Come on. Chromosome X is long. Yeah, so if you're interested, for example, in a common list of intervals across widely used whole exome sequencing kits that have been excessively and meticulously filtered for unmappable regions and segmental duplication and things like that, we have that as a supplement in the preprint that I mentioned earlier. So you can just download that interval list and use it in your analyses. That's very useful if you care about like rare copy number events and exomes. Say again? It's one of the supplements of the preprint of the gCNV paper. It's supplement 5 or something. All right, so we got our annotated interval. You can take a look at it. As you can see, the only thing that was added to it was the GC content. So, yeah, cool. Now we filter our intervals using the filter intervals tool. So what you got to do is that you got to give it your annotated intervals, and that annotated intervals at the moment it only has GC content, but you can add, like I said, additional columns to this, and then you can use them for filtering. This filter intervals not only takes this annotated interval list, but it also takes the coverage track from all the samples that you want to analyze from the collect read count step earlier. You just give it to this tool as a -I input. And then it filters your intervals according to some default filtering criterion that I'm going to cover while it runs. So by default, any interval that has a minimum GC content of 10% or maximum GC content of 90% will be removed. If you have a mappability track in your annotated interval list, if anything is less than 90% mappable, like the probability of correctly mapping an interval to that region is less than 90%, we will remove them. Also, segmental duplication content, and if a region has a lot of seg dupes in it, we will also remove it. Like I said, at the moment we don't have those annotation tracks in that annotated interval list file that we give to the tool, so this tool sort of ignores basically these. And the tool also uses that the samples over which we collected read count and we provided to it to also filter intervals that have excessive amount of coverage or very small amount of coverage. So basically it just looks at the distribution of coverage over all the intervals, and then it sort of clips the heads and the two tails of the distribution. So all of these parameters are tunable. You can change them if you think that for your analysis they're too extreme. You can always make it more lenient. So here I'm just looking at how many intervals we started with. We started with like 242,000 intervals. After filtering, we ended up with about 230,000 intervals. So we filtered about like 5% of all the intervals. All right. Yeah.
What are the CSV files in our previous input?
Oh, those TSV files. Yeah. So these are the samples that you remember that we did like coverage collection on step 2. we did like not here, so hold on. Collect read counts, right? Yeah, so we collected read counts over, for one sample, but let's say that we've already done it for 20 samples. That's the 20 samples that it's our cohorts that we want to do copy number variant calling. So those are the 20 samples, yeah. So we've already done that. So as you can see, we're reading them from our tutorial data path. We sort of skipped that step for
Sorry, so you've already done what you did with step 2, you did for each of them.
Yes, I did it 20 times. Yeah. And that's why you want to use Terra and workflows, because you don't want to run the command 20 times. Awesome. So now we're getting into the main part of the gCNV workflow, which starts with determining germline contig ploidy. It's a mouthful, but all we're doing here is that we're assigning a baseline copy number to every contig. Most of the autosomes should be 2. Every once in a while, you might get a 3 if there's a trisomy, aneuploidy, or anything like that. On chromosome X and chromosome Y, depending on the sex, you might get different things. So the tool takes as input a very important table, just called the contig ploidy prior. So that's how you impose your prior belief over what you expect a random population, what should be the ploidy in a random population. So here, for example, we're saying that chromosome 20 in a random population, the probability of copy number 0 is 0, because a person who misses chromosome 20 shouldn't be alive. Like, not as a moral imperative, but as a fact of biology. And you can have like 1 copy of that chromosome. You can have 3 copies of that chromosome with a small probability, 1%, 1%. But 98% of the time, we're saying that you probably have like, 2 copies of that chromosome. Chromosome X, copy number 1 and copy number 2, they're equally likely, female and male. And then, but there's a small chance of having 0 copies, and there's also a small chance of having 3 copies, the triple X phenotype. And chromosome Y, again, high probability of having no copy if you're female, 1 copy if you're male, small chance you might have like 3 copies. So that's I think you can use that table. There's no need to modify that table unless you're working with a special set of samples that that sample set has, for example, a higher amount of like trisomy or something like that. Okay, so what you do is that you give to the germline contig ploidy tool. Let me actually run it while I'm speaking over it. Yeah. It's literally this table. It's a TSV table. It has like these columns and.
Yeah, but where would you get information about that?
Oh, it's in the tutorial. You can basically just take that same table from the tutorial bundle. Yeah. It should be also on the GATK official tutorials. You can probably have like a link to that in some resource bundle or something. I think the easiest way is just get it from this tutorial bundle. That tar gz file. That, yeah. Okay, determine contig ploidy. You give it the interval list, the filtered interval list, the final interval list that you want to use in your analysis. Interval merging rule. You always want to merge overlapping intervals and no other rules. So make sure that if you have like head to tail intervals, you don't by mistake merge them into like a big intervals. If you don't, if for example you're doing genome analysis, if you forget that interval merging rule command, what will happen is that since you sort of bin the genome like head to tail with intervals, by default GATK merges them all. You get one big interval and you get copy number calls over just one big interval, which is not what you want. right? So you got to make sure that you always specify like interval merging rule to be overlapping only. All right. You give like the coverage read counts basically over each of the samples that you want to determine their contiguity. You give it the contiguity prior table, and then where you want to put the output. This thing should be running if if your environment is set up properly, it produces a whole bunch of output. Yeah.
On a single you don't need to have data.
Yeah, but you need multiple samples. Yeah. So what it really does. That. Yeah. Okay. So instantiating the germline contiguity determination model. Yeah, the first time that you run this, it takes a little while because there are a bunch of Python packages that needs to be loaded. Next time that you run the rest of the commands, it should be a little bit faster. Yeah.
We don't choose whether it's case mode or cohort mode. It determines based on our input.
No, you do choose that. So we're actually running it in cohort mode now. Let me see. Hold on. No, yeah, it basically, you're right. So it does that based on the specific command line. So in the case, if you want to run in the cohort mode, these are like the inputs that you give to it, like an interval list, a bunch of coverage tracks, and a contiguity prior. When you want to run it in case mode, you give it the model, and the model is one of the outputs from the cohort mode run. Yeah, and if you give it a model, that it knows that okay, it needs to run it in a case mode. I'm stuck at setting up DA-ADVI. Is anybody else further along than I am? If you go all the way down. Oh yeah, yours is doing the right thing. Looks like I got the slow machine. All right, mine has also started. Okay, now once it starts, you start to see a lot of output. So these are the same steps that I mentioned that is happening earlier. This is like a machine learning type algorithm that it sort of updates certain beliefs. It says okay, this is the noise, this is the ploidy, and then it sort of keeps like iterating between the determination of what is noise and what is actual ploidy alteration to ultimately come up with a self-consistent belief. Excellent. Okay, so I'm done with it. So as output, you get a bunch of things. So, remember that one of the other things that you specified in the command line was an output prefix. That's sort of the prefix that will be appended to every output that this tool produces. So one of those outputs is called calls. So my prefix is like ploidy core 24 because I have 24 samples. In the calls folder, I should have for every sample 0, sample 1, sample 2, all the way to sample 23. If you look at one of this, sample 0, get these files, and then you can just so these are like human readable files, so you can print them. Sample name, it gives the name of the sample, it reads it from the BAM file, and then it gives you a contact ploidy. This sample was determined, for example, the chromosome 20, ploidy 2. Genotyping quality is pretty high, so we're very, very confident that we got it right. I mean, you'd better get the ploidy at the level of chromosomes right. I mean, it's an easy problem. Chromosome X, ploidy 2, chromosome Y, ploidy 0. This is the female sample. Okay, and then we got a bunch of other files. So I just showed the contact ploidy TSV table. This was the content of it. It's a very small table. Sample name, just the name of the sample. Global read depth, that's another one of those sort of inferred quantities. That's just our average read depth of the sample. And then for this sample, this is this sort of mu psi s log. It's the it's basically like a measure of how much noise this sample has at the level of each chromosome. And yeah, this is also related to that. So in the model folder, we get a bunch more files. So ploidy not only produces per sample calls, but it produces a model. And the model is a thing that you would use if you wanted to rerun this determined contact ploidy in case mode. Let's say you have another sample that is similar in terms of sequencing the way it was sequenced with your old samples, and now you just want to do a ploidy call on that. Yeah, so you get a bunch of additional files. The interval list will be here. The average per chromosome capture bias, noise, all those things that you need to know to be able to do a single sample calling will be now bundled into this model folder. So the description of every file is here. If you're interested to learn more about them, you can do it. Okay, so I guess, yeah, so we can just, since I want to run this ploidy in case mode, let's run it in cohort mode one more time, but let's just exclude one of our samples, so that we actually run it in cohort mode over only 23 samples, and we take our 24th sample and then we run it in case mode against this 23 model, 23 sample model. So I'm just running it again. Hopefully this time it will be a little bit faster. Actually, I'm gonna run everything here because these tools take forever. I also encourage you to run everything so that we can speak over them as opposed to just waiting for things to finish. Just go run everything all the way down. Yes, I like that sound. Now, as you can see, the second time that I'm running this determined contact ploidy, I'm using a different output prefix. I'm calling it ploidy cohort 23 because it's a 23 sample cohort, which means that I will get a ploidy cohort 23 calls folder, and I will get like a ploidy cohort 23 model folder. The calls folder would be the ploidy calls for these 23 samples, which hopefully is identical to the ploidy calls to the 24 sample over the same samples. The model folder is the one that we'll be using case mode. Okay. This is done. So now we go to running determined contact ploidy in case mode. The command is slightly simpler, but importantly, as you can see, if you're giving to the model, if you're giving to the tool the path to previously run to previously made model. So this ploidy cohort 23 model is given as input to this tool, and the sample that we left out. Now if you're giving it as a case sample, and you're asking it to write the output to ploidy cohort 23 case. So it's running on my end. While we wait for it to finish, oh well, we can't. We have to. Should finish a little bit faster. Yeah. Oh. It won't. It will run it again in case mode. No. But I mean, at least you will get identical results. Okay. It's done. Like I said, we should get a ploidy cohort 23 case. That's the output prefix, and you should get a calls folder. Inside that calls folder, there's a single folder which is called sample 0, and that sample 0 has exactly the same files that we would get in the calls folder when we run the tool in the cohort mode. You can inspect the call. It's again, it's the same sample that we looked at earlier, and it has the same ploidy call. Chromosome 20, ploidy is 20, chromosome X2, chromosome Y0. All right, one thing that is very important when you want to run this tool, this determined contact ploidy thing, is that you want to exclude on chromosome X and chromosome Y certain regions which are called pseudo-autosomal regions or PAR regions. So these are like, parts of the sex chromosomes that are very homologous to autosomes. And even a female sample will get a whole bunch of coverage over the Y chromosome over those PAR regions. So that confuses the tool because the tool says, okay, hold on. So I'm getting a whole bunch of coverage on chromosome Y. So I'm probably going to call it as like ploidy 1 or ploidy 2 or something like that. So you need to exclude those regions so that a female sample actually gets basically virtually 0 reads over chromosome Y. I think there's a link here which takes you to the intervals. We haven't done it here, and nothing actually broke it this time. But I certainly have seen cases that not excluding. Well, never mind. I've seen cases that not excluding the PAR regions have resulted in making like very strange ploidy calls in sex chromosomes. So it's a good practice to do it. All right. Now that we're done with running determined contact ploidy both in case mode and cohort mode, let's just move to the last part of the workflow, which is the penultimate part of the workflow, which is to run the actual main part of the tool. This is this Germline CNV caller. So unfortunately, gCNV is a very slow and sluggish workflow. You have probably already noticed it. The tools take forever to run. So that's certainly one reason that you want to run things on Terra. You want to shard your genome in many different chunks and run chunks in parallel, so that that your workflow finishes on reasonable amount of time. Like I said earlier, on a whole exome sample, you would expect something about 9 hours runtime. If you just shard your genome 14 ways. If you shard your genome in more ways, it can probably finish a little bit faster. Yeah, but for this exercise, what you're doing is you're just looking at a very small region in I guess that's somewhere in chromosome 20. Only 1,400 bins. So just tiny amounts. Remember that we had hundreds of thousands of bins actually in this example. So running the tool over all those bins probably takes a few hours. Okay, so we have already made that interval list. And by the way, so that 1,400 bins, we are further sharding it into 2 pieces. The first 700 bins and the second 700 bins, just to demonstrate how you do sharding. So let's say we've already done that. We've sharded our interval. And now we have 2 interval lists. One of this called 12, 1 of 2, and then we have 12, 2 of 2. These are 2 interval lists. Each of them contains 700 bins in it. And now we are running Germline CNV caller first, on the first chunk of the genome, and then we run it again on the second chunk of the genome. Every time you're saying that the run mode is cohort, you're giving it the interval list of interest, you're giving it the read count over all intervals. So it automatically goes and sort of subsets the analysis intervals to whatever interval is you provide here, right? So if you've collected coverage over the entire genome, but you want to only model the specific part of the genome, that's how you do it. You just give it an interval list there. Okay, you've given it all of the 24 samples. You need to give it the contact ploidy calls. It's an input from the determine contact ploidy step. Annotated intervals, if you want to do GC bias correction or mappability correction or anything like that, you give it your annotated intervals, interval merging rule as usual, overlapping intervals only, and where you want to put the output and what is the output prefix. So since I'm running the tool twice, well, my output prefix in this case I've chosen to be gCNV core 2412, 1 of 2, and the second time that I'm running it, I have 2 of 2. I'm using that interval list, and my output prefix is also going to be 2 of 2. So once all these things run, I would expect to get a whole bunch of folders. It's 1 of 2 and 2 of 2, the first part of the genome, the second part of the genome, and inside, and I'm going to get a model, I'm going to get calls, and then we're going to look at them. So on my end, the first shard is still running. I think each of these runs should finish in about 2 to 3 minutes. Yep. Just about this output, the most important thing that you want to look at this output is quantity, which is called elbow. In machine learning, elbow is this stands for evidence lower bound. when you're fitting a model, when you're fitting a probabilistic model, when you're fitting data to a probabilistic model, there is this thing, you turn it into an optimization problem, and then there's this thing that you're trying to maximize, and that quantity is called elbow, evidence lower bound. So if you just look at what happens to this elbow from when you start running the tool to when it actually stops, elbow should start with a very small, very large negative value, like, I don't know, like -80, -90, something like that. And as the tool continues to fit data to the model, this elbow eventually starts to converge. So it's now at the -4.6. It's getting there. As you can see, the changes are getting smaller and smaller and smaller. At some point, it determines that, okay, this is good enough, and I'm gonna stop iterating, which is exactly what happened now. Okay, so the first chart is finished on my end, and I'm onto the second chart. The elbow thingy started at around, I don't know, -90, and we expect it to reach the same level. Okay. So while this runs, let me tell you about what you would expect to get. So you will get, this. Here's where we put the output, gCNV core 2412. The reason that we put 12 here, because there are like 12 interesting regions in this part of the genome that we selected. So that's why you have the 12. There's another tutorial that actually goes and looks at those 12 regions. It's linked at the preamble of this document. So I encourage you to go through that tutorial if you're interested. Okay, but inside this like output, now we this main output folder, we get like 1 of 2 and we get 2 of 2. In 1 of 2, it's the first 700 bins. You get a model, you get the calls. Inside the model, you get a whole bunch of files, and these are like all those random variables that we introduced when we were doing the generative modeling. Like, there are a whole bunch of interesting things here. Like, this is, for example, the probability that any one of our genomic intervals was determined to be an active CNV site or a silent CNV site. So if you have included in your intervals some CNV hotspots, you would expect that if you look at this file, you would see a high probability of that region to be determined to be an active CNV locus. Then remember I told you that you have like different sources of bias, like GC content, mappability, blah, 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 blah. So, and those different sources of biases contribute to different intervals with different weights. Those weights are given in this file, like this WTU thing. So yeah, so that's how you basically, that's the type of stuff that you see in the model folder, like the innards of how gCNV works. But for the purpose of like just doing variant calling and analyzing the output, you really almost always don't care about these. You probably want to like skip through all the way to when you post-process the calls from gCNV and you get a VCF file. VCF file is really the thing that we care most about. Okay, so I'm both of the shards are done on my end, as I promised. You get 1 of 2, and inside 1 of 2, we get calls model tracking. Tracking gives us a trajectory of the elbow. So if you want to see if your model converged, you can look at this tracking, and you should see a nice curve that plateaus and says, yay, the model is converged. And then for 2 of 2, you get, again, calls model tracking. Inside the model, we get all those files that I was telling you about. in the tracking, you get the history of the elbow. In the calls for each of your samples, you get a sample folder, and inside each of these samples, you get copy number calls, among other interesting sample-specific things, like the level of noise in that sample, the read depth of the sample, and the contribution of different noise, different bias factors to this specific sample. So, for example, if you want to know whether the sample has a lot of GC bias, you can actually find that information there. Okay. Since we're gonna run gCNV in case mode, to demonstrate that, I'm running the whole thing again, excluding one of our samples. And I'm just calling it cohort 23. And what we're going to do is that we're going to, once this is done, we're going to run gCNV in case mode over the sample that we left out, using the model that we're building by running gCNV in the cohort mode. So on my end, this is nearly done. And then we're going to wait for this. So that was for the first shard. You're running it on the second genomic shard. You're going to wait for this to finish. Oh, the first is already finished. Good. The second just started. And once we are done with that, with running gCNV on the 23 samples, we can do the final step of running gCNV in case mode. So one thing that we're thinking about doing at the Broad is to be able to provide gCNV as a service. And here's what I mean. So, you see a lot of different sequencing centers. They have different capture kits that they're using, and different sequencing machines, and so they all have different characteristics. So it would be kind of it would limit the utility of any tool if every time you want to run it, you need to give it a cohort of, let's say, 100 samples, because sometimes you don't have those 100 samples. Sometimes you only have like one sample you want to call CNV on it, right? So what we're thinking of doing is to collecting a large amount of samples from different sequencing centers and different capture kits, and build these types of models ahead of time. And once you give us a sample, we will try to match your sample to the best possible model that we've built ahead of time, and then use that for CNV calling. We're not quite there yet, but that's something that we have planned, we have talked about. So why did I mention this? The reason I mentioned this is that if you want to use gCNV in case mode, you should apply the same level of precaution. Like, you should make sure that the sample that you're case calling against the previously built cohort is actually a good match for that cohort. Otherwise, you get weird results. And the hallmark of getting poor results when you run gCNV is if you get a ton of CNV calls. Because biologically, we know how many CNV calls we expect to see in any sample. So if you see about that amount, you're good. If you see way more, something has gone wrong. All right. So this is also about done. Beautiful. Okay. Now we're on to running gCNV in case mode, section 5.2. This is the read counts for the sample that we left out when we built the 23 sample cohort. You're not running gCNV in case mode. You're just saying run mode is case. You're giving it the coverage of that sample. We're giving it the ploidy calls for this sample. We're giving the path to the model. and then we're giving it like output on output prefix. Remember that we also need to run this thing in case mode twice, because we ran the tool in cohort mode twice for the first 700 bins and the second 700 bins, which means that we have two distinct models. One model only applies to the first part of the genome, another model applies to the second part of the genome. So when you run it in case mode, you also need to run it twice using the first model to get copy number calls on the first 700 intervals. And second time you get copy number calls on the second intervals. Generally, case mode is a tiny bit faster, 20% faster, but it's not actually too much faster than running it in cohort mode. Cool, it's finished. The first running it in case mode. Actually, it was pretty much faster. See, it took about 2 minutes, but 1.75, 1.78. Whereas running it in cohort mode over all those 22 samples, it took about 3 minutes. So it's about 50% faster, at least in this test case. Yeah, and if you don't want to get all this output from gCNV or from any other GATK tool, you can set the verbosity level to debug. Instead of like setting it to debug, you can set it to info or something like less verbose. We're running for this tutorial in the debug mode, and that's why you're getting so much output. Yeah, if you just like remove that verbosity command, you would get much sparser outputs. Awesome. So the second part is also done on my end. Calling the case sample against the second 700 bins. It took about a minute. And if you look at what we get in output in the case mode, we get calls on tracking. We don't get any model because we're not building a model. We're using a model, right? So we get calls on tracking. Tracking again shows the trajectory of learning, and the calls is the folder that gives us like the actual copy number calls. And it has the same files that we had before. So gCNV has a lot of parameters. For example, one of them is called max copy number. So if you expect to have like copy numbers that are like more than 5 or something, you're gonna sort of change it from the default value that is 5. But typically, most de novo copy number gains are just like 3, sometimes 4. So capping it to 5 is not a terrible idea. If you want to like use gCNV to look at common CNV loci, sometimes copy number over common CNV loci can reach 20. So in those cases, you probably want to increase the max copy number to the appropriate value. If you want to run gCNV in a very sensitive mode for calling both common and rare CNV events on whole genome sequencing samples, not whole exome, these are the parameters that you need to change. And it gives you really reliable rare and common copy number events. And then there are a whole bunch of other things that we don't have time to go through, but you can do it at your own time. Oh, by the way, I said that if you want to like run gCNV, you can like scatter your interval into like multiple shards. You don't have to do it manually. There's a tool that does it. Interval list tools is a GATK tool that takes your interval and scatters it for you. All right, and the last step is actually very simple. Post-process Germline CNV calls. It takes your models. If you have 10 shards, it takes 10 models. If you have 10 shards. Again, it also takes 10 calls. So you give all the models and all the calls. And you need to tell it that which of your contigs are allosomal contigs as opposed to autosomal contigs. So chromosome X and chromosome Y. So it needs to treat them properly. And yeah, you also give it the contig ploidy calls, and then it gives you 3 things in output. One output is a denoised copy ratio. If for some reason you don't care about the copy numbers, you actually want to look at the copy ratios, but with gCNV level denoising, it gives you that. Some people actually do want to inspect copy ratios. They don't trust the copy number calls, which is a good practice. You should never trust any tool. And then genotype segments, it has to give you like copy number segments with genotyping qualities and copy number calls. And genotype intervals, and that's like an interval level copy number. So if you want to know the copy number of every single one of your intervals that you provided, you also get that. So the difference between that output, that sort of interval level output and segment output, is that the interval level output, you get actually like 1 row in your VCF file for each interval that you provided. The segments VCF file, you actually get as many rows in your VCF file as like the number of copy number segments that we detected. Constant copy number segments, right? Running it in case mode is very similar. Actually, it's almost the same thing. It has the same command. Yeah, and then finally we can inspect. You remember that one of the samples we left out. So we have one sample that we have 2 sets of copy number calls. One when we included it as a part of a cohort, and once we sort of called it against the 23 sample cohort. So the last part of this exercise to actually compare the 2. So what we're doing is that we're using GATK variance to table to take our VCF file and then turn it into a nicer table, just retaining only a few things. We do it both for the when we included the sample as a cohort, or when we treated the sample as a case against that cohort, and we get 2 nice tables that we can just load in Python. Both of them, they have like 1,400 lines because we had 1,400 intervals. For every interval, you get a copy number. That's kind of what you expect. And over all these 1,400 intervals, we have agreeing copy number calls over 1,396 intervals. So there's only 4 intervals that we determined a different copy number when we treated the sample in case mode. And that is expected because when you have a small cohort, only like 24 samples, there will be a difference between a cohort of 23 samples and 24 samples. So the more samples you have in a cohort, the more reliable the tool will run. So if you ask me that which one of the 2 I would trust more, I would trust the copy number calls when we included that sample in the cohorts, because you're actually learning something from that same sample as opposed to just taking your learnings from the 23 samples and applying it to the 24 sample. In general, we see that the performance of gCNV improves up to about 150 samples, and then it plateaus. So the larger your panel, the better. After about 200 samples, there's nothing to be gained. So if you already have 200 samples that you want to sort of joint call together, you're good. You don't need to, like, if you want to get better calls by throwing in another 500 samples, you're not gaining anything. So that's sort of really where things start to plateau. Awesome. Where to go from? Okay, so as you noticed, I didn't really take any deep dive into the results. There's a VCF file with a whole bunch of things in it that you can do filtering on, you can visualize, like look at the distribution of genotyping qualities, and you can do all the fun things with it. And there are 2 tutorials that actually does this thing, and also like takes those VCF files and then visualizes them in IGV next to your reads, so you can actually see that, oh, there's a deletion here that gCNV detected, and there's also like a loss of coverage, and there are breakpoints. So we can just those two tutorials just do those things. So highly recommend that you do that, but unfortunately we don't have time to do it today. Cool. Any questions? Yeah.
Sorry, I missed this, but how did you determine the number of shards to use?
Great question, actually. So typically, you want to include as many intervals in a shard that spans at least 50 kilobase of your genome, at least. Because that's typically how much of the genome you need to see to be able to learn the biases. Of course, the more intervals you can fit into one shard, the better. So the whole idea that we're sharding the genome is to be able to run gCNV in parallel. Yeah, because otherwise it takes just longer. So the complexity of the model is like linear in samples and linear in number of intervals. Like the more intervals you have, the longer it takes to run. Yeah. But of course, you cannot like shard the genome 1,000 ways, because every one of your shards will be so small that you cannot learn anything from that shard, right? So it should be some nice intermediate scale shards. And the other reason is memory. Like if you have like 1 million shards and you have like 10,000 samples, it just doesn't fit into memory. So you do need to sort of shard both in samples and in genomic intervals. Any other questions? Awesome. So we wanted to do the next topic before lunch, but I think this is a good time to take a break. We'll return back to our final topic. It's a totally different topic compared to what we've covered. It's about single-cell RNA-seq analysis. Yeah, so that's an area that we're getting like deeper into at the Broad. So we thought that we'll let you know about some of the things that we've done. Awesome. So Derek, when do we want to reconvene?
Yeah.
Sounds good. Started with the last topic of the workshop. So it's a very different topic. In fact, it doesn't even have much to do with GATK. But that's one of the main things, one of the key topics that we're doing at the Broad, and we're developing methods for at the Broad. And we expect to do more of these things at the Broad. And I'm pretty sure you guys also have heard about single-cell RNA sequencing or using it in your work. Everybody's doing it. It's the new cool thing. So we thought that we'd talk a little bit about that. and tell you maybe a little bit about some of the things that we've been doing at the Broad along these lines. But today's talk is going to be like kind of, for the most part, like introductory for like folks that have not seen single-cell RNA-seq or have not done single-cell RNA-seq or not familiar with the methods and the pipelines. So this is sort of the level of the talk. Yeah. So if you're only like a single-cell RNA-seq data analysis guru, this might not be too useful for you. Okay. So let's start to dissect this topic. So why do we want to study RNAs to begin with? So we have a central dogma that DNA gives you RNA, RNA gives you proteins. So if you study the DNA, which is what we've been studying for the past 3.5 days, then you probably learn everything that you want to learn from your tissue. So why even bother looking at RNAs? Why bother even looking at other things like proteins, for example? Well, the reason is that central dogma is a dogma. It's not actually quite true, in the sense that DNA and RNA and protein, they're like feedback loops across all 3 levels, that sort of this clean separation of feed forward, DNA gives you RNA, RNA gives you protein. That is kind of like an oversimplification of how cells actually function, right? So, and the reality is that your genome is not a FASTQ file. It's not even like a linear piece of, it's not even a linear molecule in your cell. It's basically a very complex three-dimensional structure coded by various regulatory RNAs and proteins. So that is more like how a piece of your DNA looks inside the cell. You have parts of it that are exposed. These are like accessible parts of the chromatin. You have parts of the DNA that are like wound around histones, and then histones are these like protein structures that sort of kind of hold loops of DNA together. And then histones themselves, they have states. They can be modified. They can be easy to like open. They can be like sort of hard to open. And then you have transcription factors, you have remodeling complexes, you have like so many different RNAs and proteins that are sort of interacting with the DNA all the time, right? So the state of a cell is, and therefore the function of a cell is determined by this sort of highly dynamic RNA, DNA, protein complex. So, and that's how, like, if you have like different cell types, right? If you have a T cell, we have a B cell, we have an epithelial cell. They all share the same DNA, but the state of those cells are different because the DNA is in a very different, the DNA RNA protein complex of those different, distinct cell types, they're just distinct from one another, right? So if you want to understand like cellular diversity, if you want to understand cell function, if you want to understand cell dysfunction, we need to go beyond DNA. DNA is just a code, right? It's a code that we're trying to crack. So we need to look at the result of that code. You need to look at the output from that code to be able to understand what that code is doing. So that's why we need to study RNA, and that's why we need to study proteins and other things. This is a little bit more of the same. So here on the left, I'm showing like parts of the gene regulation, that how from one DNA you can get different cell types, because the DNA is in a very different state. You have like different enhancers, transcription factors bound to enhancers and promoters, and then basically depending on what are the things that are coding your DNA, the propensity of expression of different genes will be different. So so we need to basically study these things in order to understand how cells function. Here on the right, I'm showing the canonical signal transduction pathway in eukaryotic cells, and you have all these like receptors that are like floating on the cell membrane, and you have like this sort of signaling cascade which involve a whole bunch of different proteins, and ultimately they find their way into the cell nucleus and result in like gene regulation and all those things. So if you want to understand like how cells respond to stimuli, or the plasticity of cells, how they go from one state to the other state, we need to measure the sort of the products that of the DNA, which again are like RNAs and proteins. Okay. Now you may ask, why? Okay, so RNAs are cool. You need to understand RNA, but why look at things in this with the single cell resolution? Well, there's so many interesting questions that we have that they require having single cell resolution. So one of them is, for example, development. How we go from the DNA to an egg, to a blastocyte, to an embryo, to a fetus. This super complex process is one of the sort of holy grail questions of biology. So if you want to understand this question, we need to look at biological systems with single cell resolution. Another interesting question is tumorigenesis, how tumors get formed, complexity of tumors, how they maintain their metabolism, and how they grow, and how they sort of evolve. These questions again require having single cell resolution. And finally, like tissue remodeling. Like here, I'm showing like a normal heart and a dilated heart. You have like dilated cardiomyopathy. It's a symptom. It's a disease, and on a very coarse level, the phenotype is that the heart is dilated. But how the heart is dilated, you know, what cells are involved in this process, or like what type of cell circuits are involved in remodeling the tissue from a normal phenotype to dilated phenotype, again, requires us to look at the system with single cell resolution to be able to model cell circuits. So here's another way of like bringing home the same message, that here I'm just showing like a piece of tissue under a high resolution microscope. Like every one of these dots that you see is a cell nucleus, and you know this is how we could be looking at tissues. But if we don't have single cell resolution, we would be looking at the tissue more like something like this, just a mixture of DNA, RNA, and protein from every cell, kind of like a homogenized goo. So there's not much you can actually infer about how the system is working by looking at this sort of homogenized thingy. This is another analogy that I like a lot. I learned it from Aviv Regev at the Broad. So when I look at the tissue has a lot of cellular diversity. A single cell assay that actually gives you the state of every single cell is more like, gives you something that looks like a fruit salad. A bulk method that basically gives you some overall RNA content of your tissue gives you more like a smoothie, right? So from that smoothie, you might be able to infer what fruits were used to make that smoothie, but you cannot understand like a diversity of different fruits. You cannot understand whether you had a bunch of bad strawberries and a mix of good strawberries and things like that, right? So the more the resolution you have, the better you can map out diversity of cell states, and that's one of the ways that we learn more about biology. Okay, so single cell RNA sequencing, as the name suggests, is basically just measuring RNAs in cells at the single cell resolution, and it has led to an explosion of data. It sort of started in around like 2009, 2010 by this paper from Tang, that they actually just pipetted one cell and made a library from a single cell. And for the first time, they showed that you can measure RNA from a single cell. It's a very difficult thing to do, right? Because a cell doesn't have too much RNA, right? So to make any type of library, you need to have like, tens of, I don't know, like, not necessarily, but typical protocol, you need to have like tens of nanograms of like genomic material. You don't have that much in a single cell. So you need to be able to amplify the signal in the single cell. So that was a feat on its own, but then it sort of started there, and now this doesn't even cover like 2023, but in up to like 2017, we were already in a place that we could do single cell RNA sequencing in about like 100,000 cells. So today there are companies that they have actually commercial assays that can give you measurement over like tens of millions of cells. Yeah. So it's definitely like one case of like an exponential growth. Moore's law. This is an actual Moore's law as opposed to the one that I showed you yesterday. Okay, so and just to tell you like two cool things that people have done with single cell RNA sequencing. There are just many more, many of them, but these are just two that I kind of like a lot. What you see here is a map, is a two-dimensional map, and this map is built in a very specific way. Every point on this map is a cell, and two cells are close to one another in this map if the RNA content of the two cells are similar to one another. Or, in more technical terms, if the transcriptional state of the two cells are close to one another, you put the cells close to one another in space to make this map. So like, for example, this cell and this cell, they're sort of very close in terms of their RNA state, but this cell and that cell are sort of far from one another. So the way people have made this nice graph is that they took zebrafish and they looked at the embryonic development of the zebrafish. They basically just took like 6 different developmental stages of zebrafish, from like super like early development to sort of like mid embryonic development that you already have formed some of the layers, and then you color them. You color the cells that you harvest from each of the zebrafishes. So the basically 6 different zebrafishes. You color them by like gray, purple, orange, red, green, and blue, and then you put all of those cells together and make that map that I told you about. And very beautifully, you actually see formation of a tree. So you go from like this sort of primordial germ cells, and then you have like these bifurcations and more bifurcations, and at the very outer layer, you actually see more and more like differentiated cell types. Like you have endoderm, you have mesoderm, you have like neural plate border, and so on and so forth. So this like nice textbook picture of like developmental trajectory as a tree that cells sort of differentiate and differentiate and differentiate. You can actually see that in the data, which is beautiful. The other one, this is a project. It's not in like same year. It's called Tabula Miris. So what they've done is that they took a single mouse and a team of like 30 people dissected that mouse in 1 hour, and each of them collected one organ from the mouse or mouse. And then they basically did single cell RNA sequencing on every organ of the mouse, resulting in about 500,000 cells. And then they basically built this atlas. This is an adult mouse. And that's why you don't see a tree structure. It's the same type of map. But imagine this map is just showing like the crown of this tree, because it's an adult mouse. And now you can actually see very nice formation of like clusters for different cells. You have fibroblasts here, you have microglia, I don't know, you have like granulocytes there. So every major cell type forms a cluster of points together, because that's the way these maps are made. So one of the key things that single cell RNA sequencing has enabled is to actually build this sort of catalog of cell type diversity. People have identified many new cell types, because there are actually cell types that, in terms of like their morphology, the way they look under the microscope, they're identical. But when you look at their transcriptional state, you actually see that they're very different. That's how actually people discovered many new cell types that just didn't know about them before. You just look at the tissue, say, okay, the same cell types, but according to our single cell RNA sequencing, they're very different. And they have like different functions. Okay, so how does single cell RNA sequencing work? It's a relatively simple protocol. So you start with sample collection. Let's say you take a heart, and then you resect a piece of tissue from the organ of interest. You rinse it, you mince it, you digest it, so that you basically get rid of the extracellular matrix. You isolate the cells, and then you filter it. You get rid of all the proteins. And now what you get is that you get a cell suspension, right? A buffer that sort of contains a whole bunch of cells in it. You inspect it. If it has like too much debris, you wash it again until like it's a nice clean suspension of cells. If you need, you incubate them. If you need, you sort them. For example, if you care about specific cell types, You can do FAC sorting, which is a fluorescent technique for like just letting specific cells that express certain like surface proteins, they only like pass through. Ultimately, the result of this step, this sample preparation step, is that you get a nice vial containing cells. Now, you have your vial of cells. What do you do with it? Well, you could either do the low throughput method, or you can do it in the high throughput. The low throughput method is, as the name suggests, it is very, very hard to do. So you need to pipette individual cells to put them in different wells in a well plate. A typical well plate has like 384 wells. So I don't know how long it takes to do that manually. I mean, there are definitely like robotic liquid handlers that can do that a little bit faster, but in general, like the throughput of this method is very, very low. So the main sort of exponential growth of this technology started when people realized that they don't need to do this manually. They can actually do it in a very high throughput way using tricks from microfluidics. Okay, so how does the microfluidics way, the high throughput way of doing single cell RNA sequencing work? Remember, our goal here is that we want to effectively put individual cells in individual chambers, and those chambers could be either wells of some well plate, or it could be droplets, or it could be like partitions. I don't know. You need to somehow partition space. And in each partition space, you need to just put like one cell and make a library from the RNA of that cell, so that then we can go and sequence those RNAs. Because if you mix all the cells together and make one big library out of that, you lose the cellular identity. So we need to sort of partition space and somehow barcode those partitions in a way that we can then go and sort of figure out which RNA came from which cell. So that's really the name of the game. So how did we do it with microfluidics? The first step is this. You can very reliably make droplets with microfluidics. All you need to do is you need to flow oil in one channel, and you flow water in the other channel. It's the same rate, and it naturally forms droplets. Very, very, you don't need to do anything crazy, because oil and water to just form an emulsion, right? So that's interesting. If you can form droplets, what if you just trap individual cells in these droplets? Okay, and that's exactly what we do. So we flow oil, and then we flow our cells, and now each of these droplets gets one cell. But that's not enough, because if you just have like cells trapped in individual droplets, how do you want to turn that into a library? To be able to turn that into a library that you can then put on your sequencer, you need to be able to sort of lyse that cell, which means to release the content inside that cell. You need to convert the RNA inside those cells to DNA. You need to sort of barcode those DNA so that each droplet has a unique barcode. And then that's the library they want. So inside each droplet, we don't need just a cell. We need something else. So what we need is a bunch of enzymes, like reverse transcriptase that converts RNA to DNA. We need a bunch of buffers, because enzymes don't work in all types of media. And you need to have some barcodes. And those barcodes are sort of somehow we want to be able to transfer those barcodes to the DNA that we're making from the RNA. Okay. So what we do to this end is that in addition to flowing cells, we also flow these things which we call barcoded beads. And I tell you exactly what's inside those barcoded beads. But ultimately what happens is that you get inside each droplet a cell and a barcoded bead. The cell contains all the RNA that we care about. This barcoded bead contains like millions of copies of the same barcode. Now, somehow we need to transfer these barcodes to the RNA of the cells. Here's actually like under a microscope what a droplet looks like. This is the cell. And this is like that barcoded bead. Okay, so what's in that barcoded bead? That's the fun part. The barcoded bead has an adapter. We always need to put adapters because we want to do PCR amplification. Whenever you want to do PCR amplification, you need to put some sequence there that you can use it to prime PCR reaction. So that's that. You have an adapter. You have a totally random sequence called the UMI. UMI is Universal Molecule Identifier. It's a totally random sequence. Like every one of these sort of barcoded things, the barcoded oligonucleotides that we have in the bead, they have the UMI. And then you have a cell barcode, and the cell barcode is unique. The cell barcode is the same for all of these primers that we have in the barcoded bead. And ultimately, we have a poly T. T. Yeah. So now what happens is that once the cell is lysed, the RNA sort of leaks out. Now, in particular, mRNA, the messenger RNAs, the ones that we care about, they have a poly A tail. So the poly A tail of the mRNA hybridizes with the poly T tail of this oligonucleotide, this guy. And now what happens is that a reverse transcriptase, which is an enzyme that converts RNA to DNA, comes and basically starts from here and converts this part, like synthesizes a strand of DNA. Yeah, and then something else happens, which is called template switching, which is something slightly technical. I'll explain later. Is that the reverse transcriptase switches and then synthesizes the second strand. So this is how effectively what you get out of this is that you get a double-stranded DNA, which has basically the same sequence as the RNA, but it also has a unique cell barcode so that we can then group them using the cell barcode and say, aha, all of these guys come from the same cell. And then they also have this totally random sequence, UMI, that we can use for counting. Because if you have, let's say, 5 copies of the same RNA, since each of those copies gets one of these totally random UMIs, if you see like 5 different UMI sequences, you would say, oh, okay, I have like 5 different copies of that RNA, right? So even if you take this library and you amplify it, you do PCR amplification like 1,000,000 times, you can always use this UMI to do accurate counting of how many copies of the RNA you have, because every RNA gets a totally random UMI. Okay, so just a little bit more detail about how actually conversion of RNA to a barcoded cDNA happens. So the first step is that this is sort of, let's say, a zoom in of our barcoded bead. It has like these little hairs, and each of these hairs is one of these single-stranded RNAs typically. Sometimes they're like single-stranded DNAs in different protocols. So it has a poly T tail. This poly T tail hybridizes with the poly A tail of the mRNA. And now reverse transcriptase comes, and the reverse transcriptase synthesizes sort of this first strand of our DNA. And conveniently, sort of now it starts off from this barcoded primer. So the barcode is already part of this molecule that we're making. Now, this is something I don't know like how people came up with, but this is a very genius idea. They found a specific reverse transcriptase that once it reaches the end, the 5' end of the RNA, it adds a CCC. So you might ask, why do we need this CCC? It's actually pretty cool. Once you have a CCC, then you can put some primer inside the same droplet that has GGG. So that GGG hybridizes with the CCC, and now you can use that basically sequence as a primer to synthesize the second strand. So that's how you make a double-stranded molecule. Remember, so whenever you want to sort of synthesize a strand of DNA, you need to have a primer. So we have the primer for synthesizing the first strand. That's the primer that we put in, but we also need to have a primer to synthesize the second strand. We didn't have that primer. But since we had a CCC at the end, we just put in our primer, which has a GGG tail, hybridizes, and starts sort of priming reaction to synthesize the second strand. This is called template switching. This idea came way before single-cell RNA sequencing. It's called like smart sequencing, SmartSeq. And that's one of the key ideas behind single-cell RNA sequencing. Okay. Now here's the full library structure of one of the RNA, like one of the fragments that we get from this protocol. Oh, by the way, I forgot to say that once you basically make this barcoded cDNAs in individual droplets, you can now break the emulsion. You can just break each of the droplets and mix the contents together because they're barcoded. They're barcoded. You can always, once you sequence them, you can sort them by their barcode, and then you can say, okay, all of these fragments came from this cell, all of those fragments come from that cell, right? So the identity of each droplet is now associated to a unique barcode. So that's why you can do a lot of the steps of this protocol in bulk. Okay, anyhow, so this is sort of the structure of the library fragment. We have our usual like Illumina adapters and primers on that end, another Illumina adapter, some sample index, another adapter for like doing the actual short read sequencing. So these are like a universal things. We have the same in any library that we want to throw at an Illumina sequencer. But specific to single-cell RNA sequencing is what we have in the middle. So we have a cellular barcode, and then we have a UMI. And like I said, the cellular barcode is unique to each droplet. The UMI is totally random. And then here we have the reverse transcribed sequence of the RNA in DNA form. So what we do is that we do paired end sequencing. We get read 1 and read 2. Read 1, we do a few cycles, in this case 28 cycles. The first 16 cycles gives us the cell barcode. The next 12 cycles gives us the UMI. So that's read 1. On read 2, we primed read 2 from this TrueSeq read 2 sort of primer, and we sort of read the RNA sequence. So the FASTQ file that we get from sequencing this library, read 1, effectively gives you the cellular identity and the molecular identity of each of our RNA. Read 2 gives us the actual RNA sequence. Okay. So some numbers is good to have in mind. A typical experiment, typical sort of short read sequencing gives us something about like 100 million to 500 million reads. We typically like process 5,000 to 20,000 cells at a time using the standard, like the most like widely used commercial assay from 10x Genomics. mRNA to cDNA conversion efficiency, the step that sort of converts RNA to DNA, unfortunately, is very inefficient. The efficiency is only 10%. A typical mammalian cell has about 300,000 RNAs. What we get out of this sequencing run is something close to 30,000 RNAs. So we only see like 10% of all the RNAs. It's still good data, but the data is going to be very sparse and very noisy, because it's like you have like this lot of like molecular complexity and you only randomly see 10% of it. So naturally it's going to be noisy. You know, just so that, you know, that UMI sequence doesn't remain a mystery, the idea is this. It's actually a very cool idea on its own. In the case of this specific chemistry that I'm showing, the UMI sequence is 12 nucleotides. Now, with 12 random nucleotides, you can represent 4 to the 12, which is about 16 million different sequences, right? Now, a typical mRNA in a mammalian cell has about like 1 copy per cell. The most highly expressed mRNA definitely has less than 10,000 copies. Like that's really a high bar, right? So let's just take the most highly expressed mRNA ever. It has 10,000 copies. We have 16 million like random sequences, UMIs. The odds that the same random sequence is assigned to basically two different copies of the same mRNA is pretty low, right? So in all likelihood, every mRNA, every actual physical mRNA copy in the cell gets a totally random UMI. So you might ask, why do that? Here's why. Let's say you had a cell that had 2 copies of TP53 mRNA, and it had 1 copy of the EGFR gene, EGFR mRNA, right? You make a library, you do PCR amplification, you sequence, you have all the biases that we've been talking about, right? And somehow, let's say it so happens that EGFR is a very, very amplifiable gene. PCR just likes to amplify it, So what might happen is that single copy of the EGFR gene, we might actually see 4 copies of it in our data because it was like very, very amplifiable. But that TP53, we might see like 2 copies of the first one, and we might see like 1 copy of the second one. Yeah. So if you just do naive counting, we might say that, okay, I have like 3 copies of TP53, but I have 4 copies of EGFR. But if you now look at the UMI sequences, you see that all the copies of the EGFR that you see, they have the same random UMI sequence. And then you will conclude that they should all be PCR amplicons of the same mRNA, because again, the odds that 4 different copies get the same random sequence is pretty low. So that's where we use UMIs. You just use it very, very effectively for deduplication. Yeah? So we don't need to do mark duplicates anymore. Just look at the UMI sequences and then just count how many UMI sequences we have.
One of the issues with the single RNA is synthesized to CDMA. In many cases, you don't synthesize the mature, cold mature.
Yeah.
So, if this pose any issues, these the size of the segments of each.
Yeah. That's a great question. Yeah. So the question is that many of the mRNAs that we have in cells, they're not mature. So mRNA has a life cycle. When they're sort of initially transcribed from a gene, it is called like in a mature mRNA, it has like introns and exons. It can be also pretty long, like upwards of like 10 kilobase, sometimes even like 200 kilobases long. But then there's this process which is called like splicing. There's this machinery called spliceosome that comes and basically just takes chunks of this mRNA out, the introns, and then you get sort of this mature mRNA, which is typically much, much, much shorter. It only has a few coding regions. So the question is that, what if you just grab one of these immature mRNAs? Does it, cause some type of an issue when it comes to like doing analysis, data analysis? The answer is that generally no. The reason is that even though those mRNA fragments are much, very, very long, what we always do is that we fragment it. Part of this protocol is doing fragmentation and then size selection. So effectively, we only get basically just some part of, either an intron or an exon or, just a UTR part of the mRNA. We just see some piece of the mRNA that is like short enough that you can throw on the sequencer. Now, unless you're looking at some very confusable genes like HLA genes, IG genes, these were like, VDJ and those, weird regions in immune cells that you have a lot of homology and diversity, you might not be able to like uniquely map that mRNA to specific genes. But 99% of the cases, you can actually, even by observing a short fragment of the mRNA, you can uniquely map it to the gene. Now, you might not be able to tell exactly which isoform it is, like, is it like exon 1, exon 2, exon 3, or exon 1, exon 2? So that's a harder one. But if you just want to know what is a gene identity, that's an easier problem that often doesn't cause a problem. Okay, so hopefully we have like an overall understanding of the protocol. It's converting RNA to DNA and then throwing the DNA on the usual sequencer. So analyzing single-cell RNA-seq data has 3 stages. Primary analysis, secondary analysis, tertiary analysis. Primary analysis is the same thing, is the same primary analysis of genome sequencing analysis. All we want to do is that we want to, like, from the library that we've made, we want to get some FASTQ files. Yeah. So we can just use the exact same tools that we have for converting, let's say, BCL files to FASTQ files. Secondary analysis is domain specific to this data type. And generally, the idea is that we're going to go from FASTQ files to some quantified features. So what do I mean by quantified features? Well, one of them is, for example, a BAM file that has one record for every mRNA. And as additional tags in that BAM file, you would know what is the cellular barcode and the UMI of that mRNA. Yeah, so you kind of want to take the read one and read two. You know, from read one, get the cell barcodes and the UMIs. From read two, you get the mRNA sequence. You align it using a transcriptome aligner to get sort of the BAM alignment record, and then sort of combine them together to get a nice analysis-ready BAM file. So that's one part of the secondary analysis. And the most important part of the secondary analysis is to take that BAM file and extract even more granular feature, like less granular features from the BAM file. For example, you want to have like a matrix of gene expression. Like this cell with this barcode expresses this gene with this many copies, that gene with that many copies. So basically making a matrix of cells by genes. And in each cell and each gene, you just put like an integer number, and that is how many copies of that gene that you see in that cell. So making that matrix from FASTQ files, that's part of the secondary analysis of single-cell RNA-seq data. And finally, we have the tertiary analysis, and that's anything that you would want to do on that gene expression matrix. You can do some quality control. Maybe you want to do some denoising, normalize it because maybe different cells have different sequencing depth. We select important features, get rid of some boring genes. Dimensionality reduction, some genes are co-regulated, so maybe you want to treat them separately. You want to identify groups of co-regulated genes, correcting for batch effect, clustering them, identifying how many different cell types you have in that experiment, doing some differential expression analysis, like which genes my T cell expresses more than my B cell, that type of thing. Maybe you want to do trajectory reconstruction if you're looking at a developmental system, looking at mouse embryos or something. Maybe you're interested in, reconstructing trajectories of morphogenesis and visualizations. There's so many things that you can do with this data. You can make nice visualizations. So let's review the secondary analysis in a nutshell. I've already spoke over this 10 times, so it should be pretty fast. So this is sort of the structure of the reads that we get, right? We get our cell barcodes, which I'm just using different colors to look at different cell barcodes. And we get these random UMI sequences that we use for counting. And then we get our mRNA sequences. So the very first thing that we do is that we align the mRNA sequences to a reference transcriptome using a transcriptome aligner. And there are many tools for transcriptome alignment. There's STAR, there's HiSat2, there's Minimap2, and many, many more tools. So you just pick your favorite tool and then align the mRNA to the genome. The difference between a transcriptome aligner and a genome aligner is that transcriptome aligners allow for gaps. Remember that mRNA, especially mature mRNAs, they have exons only, and exons are only 2% of the genome, which means that between one exon and the other exon, if there might be a huge gap. So the aligner must allow these types of gap in the alignment instead of soft clipping your read. Yeah. So that's really the main difference between transcriptome aligners and genome aligners. Now, the next step is that you sort your reads based on their cell barcode, UMI, and gene. You sort of group them and then you sort them so that all the cells with the same, all the reads with the same cell barcode appear together. And next you sort them by UMI, so all the cells with this, all the reads with the same UMI appear together, and then you sort them by gene. Yeah. So once you've done the sorting, now all you need to do is to group them. You say that, okay, I see 2 copies of TP53. They have the same UMI and the same cell barcode, so I just count them as 1 copy because it has the same UMI. And then you see that, oh, okay, I have EGFR, 1 copy. I have HBB, 1 copy. So you just basically, after you're doing the sorting, you identify how many actual copies of each mRNA you had. And once you've done that, it's just as easy as putting it in a matrix form. So you form this matrix of the cells by genes, and just record the number of how many copies of that gene you see in each cell. So I've called it like genes by droplets as opposed to genes by cells, because some of our droplets might actually not contain a cell, might actually be empty. And I'll talk about that in a second. Remember, we're flowing cells from one channel. We're flowing our barcoded beads in the other channel. And then things, some stars must be crossed and aligned in the right way so that you get exactly one cell and exactly one barcode in one droplet. Most of the time, you see no cells. Sometimes you actually see two cells. So all these weird things happen. It depends on the size of the cells. It depends on the density of the cells. So there are guidelines, for like how to set the density of the cells properly for each device so that you get the correct flow. Yeah. But ultimately, it's some of it is unavoidable. Some things that I glossed over is that your cell barcodes and your UMI sequences, like any other sequencing readouts, they might contain error, and you need to correct for those errors. Why is it important? Because if you don't correct for, let's say, an error in the cell barcode, you might then, for example, erroneously conclude that that read comes from a different cell, right? So you need to sort of correct for errors that might happen in cell barcodes. The same thing for UMIs. For example, this pink UMI, this magenta UMI, if you had a barcode error, it would appear like as a different UMI, and then you would count it twice, because you remember we're counting unique UMIs to count unique RNAs. So again, we need to correct for those things. And there are many tools that people have written for secondary analysis, the step of going from FASTQ files to matrices. Cell Ranger is one of them. That's from 10x Genomics. 10x Genomics is the most widely used commercial device for doing single cell RNA-seq sequencing. There is another tool called Star Solo. Basically, it's a single cell version of Star, which is almost identical in operation to Cell Ranger. Like it's 99.95% identical. The results are to Cell Ranger, but it's 10 times faster. And then there are other tools like Alvin and Alvin Fry and Callisto bus tools that are like 1,000 times faster than Cell Ranger. Cell Ranger is sluggish. It's the reference implementation that I actually use myself, but it's just very slow. So there's this nice paper that sort of compares like many different secondary analysis pipelines. We have our own pipeline at the Broad. It's not in these. It's discussed in this paper, but it's not on those graphs. Called Optimus. Under the hood, Optimus actually uses Star Solo, so it's the results are identical to Star Solo. Yeah. Yeah, no. So in my opinion, the most accurate one is Alvin. And the reason is that it handles multi-mapped reads a little bit more carefully than Cell Ranger and Star Solo. In Cell Ranger and Star Solo, multi-mapped reads, by multi-mapped reads, I mean basically a cDNA that you can align equally well to two different genes. So you don't know exactly what gene it comes from. So Alvin has a bunch of interesting, heuristics, some EM algorithms that actually tries to quantify the abundance of this multi-map reads. So yeah, so if you're looking for accuracy, I suggest that. Cell Ranger actually is kind of like, let's just filter. If you can't do something about, if you can't make an easy decision, we just filter. So, yeah. All right, so how about we take a 5-minute break now before we do the tertiary analysis stuff.
Yeah. Yeah.
All right, let's slowly get started with the rest of this presentation, and then we'll directly jump to the exercise after this. So no more break, just power through. And I'll try to be less verbose. Okay, so we covered secondary analysis. I remember that we had like 3 analysis stages: primary, secondary, tertiary. Tertiary analysis is basically, now that we have our count matrix, what do we want to do with it? So yeah, so the general answer to this question is that what do you want to do with your count matrix? This, oh, by the way, gene expression, people refer to that as, you know, they refer to it as, you know, many different things: counts matrix, gene expression matrix, you know, so you see them, you see both all the time. I refer to it as like count matrix. Okay? So once you have your count matrix, which is the matrix of cells or droplets by genes, then what you want to do with it, it totally depends on the biology that you're after, right? But there's some steps that are pretty much common, regardless of what biological question you're trying to answer. So you want to do some maybe data pre-processing. You want to identify which droplets contain a cell, which droplets do not contain a cell. And then you want to do maybe some cell quality control. Some of the cells are like, you know, dead or dying, or just, you know, not nicely sequenced, not deeply sequenced, something like that. You want to like maybe throw them away. You want to normalize gene expression, because different cells actually get sequenced at slightly different depth. So you get more reads from some cells, you get fewer reads from another cell. So you want to sort of equalize the level of the number of reads that you see from each cell. So then dimensionality reduction and clustering, you sort of wanted to identify some distinctive features of different cell types, and then make those maps that I mentioned in the beginning of the talk, those 2-dimensional maps that shows similar cells next to each other and dissimilar cells sort of far away from one another. Differential analysis and cell type assignment. Differential analysis is basically just, you want to answer like these cell cluster compared to the other cell cluster, what genes are overexpressed or underexpressed. You know? And cell type assignment is basically, now that you have like these clusters of cells from your data, and remember that each cell is a row in the count matrix, right? Once you basically say that, okay, so these rows in the count matrix clustered together, these other rows of the count matrix clustered together, you want to give a label to it. Is it a T cell? Is it a B cell? Is it a pancreatic cell? Is it a hepatocyte? Yeah. So that is another step that is very important. So that is sort of, you know, once we're at that stage, you can consider it as like an analysis ready count matrix, then which, you know, you can do like further downstream analysis on, depending again on the biology that you're after. Maybe you're looking at some case control. Maybe you just wanted to look at the heterogeneity of cells in a tumor. I don't know. You know, there's so many things that you might be doing. So let's quickly go through these steps. So let's start with like, the first box, which is data preprocessing and cell calling. Remember that this is how the protocol worked. We had these barcoded beads, and then we co-localized a barcoded bead with a cell, ideally inside individual droplets. In reality, some of the droplets do not get a cell, but even if a droplet doesn't get a cell, it doesn't mean that there is no RNA in that droplet. Why? Because remember that that step that we were like preparing our cell suspensions, we took like a piece of tissue, we digested it, and then we washed it, and we repeated it again and again and again. Every time you do it, you rupture a cell. Cells are actually very flimsy. So, the more you manipulate them, the more cells you rupture. And then once the cell opens up, it leaks this mRNA into the cell suspension. So that's the medium that the cells are suspending on is actually full of all types of crap, cytoplasmic debris, proteins, and mRNA and all those things. So of particular interest for us is what we call ambient RNA. And those are basically the RNA that are floating in the soup that makes our cell suspension. So even if a droplet doesn't get a cell, it gets a scoop of that soup, and which means that some of the rows of our count matrix are basically an observation of that soup. It's not an observation of a cell inside the soup. It's an observation of the soup itself. So what we want to do is that we want to identify those rows, and we want to say that we don't want them, and we only want to select rows that correspond to actual cells. Now, in addition, sometimes we want to say that, well, we see 7 copies of that gene in this actual cell, but out of these 7 copies, maybe 1 copy is actually from the soup. So we not only want to identify which rows of the count matrix corresponds to actual cells, but we also want to remove the contribution of that ambient RNA from our cells. So the net effect of like this type of pre-processing and cell calling is that we get a sort of a cleaned up count matrix that has far fewer rows, because most of the rows in a count matrix, they do not correspond to cell containing droplets. And also some of the counts might change, because if you remove the contribution of the ambient RNA, then some of the counts would go down. So that total depends on how clean or how dirty the cell preparation is. If you have like a very clean, nice cell suspension with few ambient RNA, you don't remove too many counts. But if you have like if you use a very harsh protocol, maybe sometimes when people want to extract cell nuclei, you need to rupture every single cell and only enrich for cell nuclei. So you get a medium which is rich, full of like cytoplasmic material. So in those cases, correcting for ambient RNA is very important. So how do we do cell calling in practice? How do we basically mark the rows of this matrix as containing cells or not containing cells? The easiest way, and that's the strategy that is implemented in Cell Ranger directly, actually, is to just count how many UMIs, or basically how many distinct mRNAs you see in each droplet. Yeah, a droplet that contains a cell should contain more RNA. And a droplet that doesn't contain a cell, it only has like a sample of the soup. Hopefully that soup is not too full of RNA. Otherwise, something has gone wrong with the experiments, right? So simply by just counting how many RNAs you see, basically just summing this matrix in column, like row-wise, you see a nice separation. Like in this toy example, maybe you see like 37 RNAs here, and only 5 here, 32, 3, 31. So you might say that, okay, anything above 30 is probably like a cell-containing droplet. Anything less than 10 is probably like a soup. More systematically, the way you do it is that you make this plot, which is called the barcode rank plot. You rank the barcode from having, in terms of the total RNA content. And you put that rank on the x-axis. On the y-axis, you put the total RNA count in that droplet, and then you make a log-log plot, because you have just so many. You have like 5 orders of magnitude difference in terms of like the RNA content in the cell, and you have like millions of droplets. So if you show it in a linear plot, it doesn't look good. So you do the log-log plot, and what you often see is a structure like that. So, the way you interpret this structure is that these ranks, anything, below, let's say rank 10,000, these probably are cells because they have a lot of mRNA. But then you have this sort of sharp cliff in this sort of cumulative distribution thingy that now you see a whole bunch of droplets. And remember, this is a log scale. So this is close to like, hundreds of thousands of droplets that do contain some RNA, but at the level of maybe like 200, 300 counts. So probably like all of this ranks after like 10,000, you might say that, okay, those are the ones that you want to cut out. Yeah. So effectively what you're doing is you're trying to look at a cliff. You're trying to identify like a cliff on this top plot. So this is the traditional method. But we have a favorite method called Cellbender. And Cellbender does this much more rigorously. So it doesn't use this traditional method that I told you about. It actually uses one of those like gCNV type methods. It's a generative model, and it tries to actually estimate from that soup, what is the profile of the soup, and then use that to better identify soup containing droplets, and also use that information to clean up your count matrix. So that's what we recommend. It's available on Terra as a workspace. It's a very simple workflow. It takes your count matrix as input. It gives you cleaned up count matrix as output. The next step is to do quality control. So even if a droplet contains a cell, or even if a droplet contains something that is not like a soup, doesn't mean that it's a good cell. Doesn't mean that it's even a cell. What it could be is that it could be like a piece of a cytoplasm. Or if you're working with neurons, it could be that you're just, the droplet got like a piece of the dendritic arbor of the neuron, or maybe it got like a long axon, So especially cells that are big and they have like, just projected in space, they provide you, especially once you're like, any protocol that you use to dissociate a tissue, you're just generating all these debris. Some of them are large, some of them contain mRNA, but they're not cells. Maybe there's no cell nuclei in it, right? So you always got to do some additional quality control over what you have determined to be non-empty, because not every non-empty droplet is a good cell. So unfortunately, there is no universal way to do quality control. It's very, very domain specific. If you're working with heart cells, you're probably looking at specific things that are like very specific to hearts. Looking at neurons, you're probably like looking at neuron specific features to make sure that you have those in your data, right? There's some things that are like semi-universal. For example, mitochondrial genes. If you see too much of the expression of mitochondrial genes, you can conclude that that's a very stressed cell, because cells under stress, they tend to change their metabolism to a state that they just want to proliferate their mitochondria. There's another reason that might happen is that cell apoptosis also begins with a cascade that starts in mitochondria. So if you see a lot of the expression of mitochondria, like if you see like 20% of your counts are just mitochondrial genes, you might say that okay, that's probably like that's not a good quality cell. It's a very stressed cell. I mean, maybe you're interested in analyzing stressed cell. See, that's the problem. There is no hard and fast rule. Any rule that I say that you can use it to do quality controllable cells, you can actually come up with an exception for it. So it's kind of like an art. And this is where biology happens in a sense, right? Doing quality controllable cells is part of the research in a sense. Okay. But again, I mean, the standard things that people do, I have to tell you, there's just like cells that express too much mitochondrial genes, droplets that contain like an outlyingly high number of mRNAs. If you see that most of your droplet contains like 5,000 mRNAs, but you have a couple of droplets that contain like 100,000 mRNAs, you can probably conclude that something has gone wrong. Maybe multiple cells, are encapsulated in that droplet. Maybe there was a collision between cell barcodes. All those things can happen. All right. Then you want to do some normalization. After doing your quality control, normalization is actually pretty easy. You take your matrix of gene expression, you just sum the total counts. So X and G, let's say this is the element N cell number N, G gene number G. You take like every element of your count matrix X and G, you divide it by the sum over G. So basically that's the, for every row of the count matrix, you sum over the columns to get the total RNA, and then you divide all that, that entire row by the total RNA. So what this gives you is a fractional RNA expression. Instead of like having like absolute RNA expression, this is like, a fractional RNA expression. So the reason that this is important is that if you sequence two cells at different depth, the absolute number of RNAs can be different, but the fractional number of RNA will be the same. Yeah, this is kind of like very similar to the, remembering the CNV calling. Once we got our read counts, we divided, we normalized them by like the total count. That's kind of like similar to that. Then you multiply by big numbers up like 10,000 so that you can interpret this number as like fraction of the number of RNA copies in 10,000 RNAs. Sometimes people use like 1,000,000 here, and then they call this number TPM, transcript per million. Like if you observe like 1,000,000 RNA, what fraction of them are that? So, but you choose like a large number here, doesn't really matter all that too much. And then you take a log. And the reason you take a log is because gene expression often covers 5 or 6 orders of magnitude. There are genes that are like highly expressed, and when they're highly expressed, they're really highly expressed. So if you just work with just raw gene expression data, or even like raw fractional gene expression data, you're going to be dealing with like very, very small numbers and very, very large numbers. So when you take a log, you sort of compress the dynamic range, just work in the log space, just makes your analysis sort of easier. But you remember, you can always like invert all of this process. And if you want to like access your raw counts, this is an invertible transformation, this is an invertible transformation. This is not an invertible transformation, but this is an okay transformation. There's no harm in doing that. And finally, you z-score it. So z-scoring is sort of the standardization. So you subtract the mean gene expression, and then you divide by standard deviation of the gene expression, so that you effectively turn gene expression into a number effectively between -1 and 1. If it's 1, it's relative to in distribution, it's lying on the right tail of the normal distribution, right? If it's -1, it's lying on the left tail of the normal distribution. So this type of like z-scoring allows like all the genes to be on this sort of a level playing field, so that highly expressed genes and lowly expressed genes do not necessarily dominate or like get submerged in your analysis. Okay. Next, what you want to do is that you want to do something called dimensionality reduction. Again, at this point, we have our count matrix, which is nice and normalized. You have 30,000 genes. Working with 30,000 genes is difficult. And not all of those 30,000 genes are interesting. Ribosomal genes, mitochondrial genes, housekeeping genes, the genes that are they're just necessary for cell function, but maybe you as an analyst, you don't care about them. Or maybe you're looking at a specific cell circuit that just doesn't involve like these boring, ever expressed genes. So what you want to do is that you want to select interesting genes. So you might ask that, okay, so what are interesting genes? But there are basically 3 criterion for being interesting. That's a standard workflow. Different people have different ways of doing this. You want the gene to be not too lowly expressed, because if it's too lowly expressed, it's probably very noisy. Our measurement of that gene is probably not reliable enough. You don't want the gene to be too highly expressed, so you don't want like this tail. And the reason is that often highly expressed genes are boring. They're mitochondrial and ribosomal, and we don't often care about them because the ribosomal gene and mitochondrial genes do not determine the identity of cells. At this point, we're looking at finding genes that might actually help us identify the identity of a cell. Okay, so this sort of leaves sort of genes that are expressed sort of in the middle, not too highly expressed, not too lowly expressed. But now you look at those genes and you select the ones that are very variable between droplets. If a gene is sort of constant, all the droplets sort of express those genes at similar levels, that's probably like a boring gene. It's not informative. But if you find a gene that has high variance between droplets, it means that it's probably expressed in some cells, but it's probably not expressed in other cells. So that's an informative gene that you want to keep. This step is called highly variable gene selection, HVG. You typically go from 30,000 genes to something about like 500 to 5,000 highly variable genes. And you often just work with these genes. Then sometimes you then go back to your entire genes, but this is what you start your analysis with. Now, if you want to further reduce the dimensionality, because 5,000 genes is still too much, yeah? And there's a good reason for that. Many genes in cells are co-regulated, which means that their expressions are like tightly correlated with one another. So out of those 5,000 highly variable genes that we select, there might be like groups of hundreds of genes that are like so strongly correlated with one another that you can treat them as one group, because maybe they're like part of the same cellular pathway. Yeah, maybe there's the same transcription factor that is like regulating the expression of all of them. All right. So the way you do that is that you do the good old principal component analysis. That's exactly what principal component analysis does. It gives us combinations of genes that co-vary. Yeah. So we typically go from 5,000 highly variable genes to something about 20 to 30 principal components. And each principal component, naively, it's like one group of co-regulated genes. Okay, now we want to even reduce the dimensionality further, because we want to make a two-dimensional map. Now, remember that at this point, once we do the PCA, we're in a 50-dimensional space. Every cell has 50 features. You can't visualize 50 features. Maybe you can select two of these features, feature 1 and feature 2, and like plot one versus the other. But what about the other 48? Yeah. So you need to find a way to make a two-dimensional map of a 50-dimensional space. So that's analysis is called nonlinear dimensionality reduction. And there are two widely used algorithms designed for that. One of them is called TSNE, the other one is called UMAP. TSNE is T students stochastic neighbor embedding. UMAP is uniform manifold approximation. The key idea of both of these algorithms is the same. The key is that it's an optimization problem. You want to find positions so let's say that you want to make a two-dimensional map of all of your cells. So for every cell, you have an XY coordinates. You want to determine these XY coordinates of every one of your cells, right? But you want to do it in a way that if two cells are close on your two-dimensional map, then you want them to also be close in that 50-dimensional space. Yeah. So in a way, you want to collapse a 50-dimensional space to a two-dimensional space in a way that you can sort of capture the global topology of your data, so to speak. So nearness and sort of this proximity and distance in the 50-dimensional space, you want it to be basically proximity and distance in the two-dimensional space. So, you can imagine that, there are some very nice visualizations how these algorithms work. I encourage you to check them out. They can take a point cloud of a giraffe or something, and then you can sort of collapse into two-dimensional space, and then you get a two-dimensional like giraffe example. So it allows you to like gain intuition about how these algorithms work. Yeah. So sometimes it's actually kind of not trivial how to do that, because I don't know, like there's some things that can only make sense in higher dimensional space. Like if you have like a knot, something is just like intertwined in a high dimensional space. If you collapse it in two-dimensional space, it doesn't make sense anymore. So you do see some of those weirdnesses when you're making these maps, but that's the best we've got. This is just for visualization. Remember, when you're making this map, it's just to gain intuition about the structure of your data. It's not to do any analysis directly on the space. You always do your analysis either in this space or in this space, or even in the raw gene expression space. This is just for making nice visualizations.
Might understand the variance and what is going to be more to the variance. Another is that collapse together. Can you use, when you have a fight point to understand that, how the different variances contribute in different directions of different stage? So that it might do that and together where actually the variance continues. So what is the component and not what is the best strategy?
We'll cover that a little bit, yeah. But really the idea is that, yes, you got to look at how much variance each of your principal components explains. And then if there's a natural like cliff, you might say, okay, these like first 50 PCs or like first 30 PCs, they explain most of my variance. And then I also want to like neglect the other PCs because they're just not explaining much variance, yeah. So there are heuristics like that. Unfortunately, they don't work very well with single cell data. When you look at the variance explained by principal components in single cell data, it's often like just it's a plot that sort of decays without any knee. So you have to just put a cutoff somewhere. Yeah. So, and there are other algorithms for dimensional reduction, like non-negative matrix factorization, NMF, which actually does give you one of those elbows. So those are like more appropriate, I would say, for analyzing single cell data. And what I'm explaining today is not the state of the art of single cell analysis. This is the standard single cell analysis, right? This is something that you do as a first step, and then sometimes, just go back and refine components of this. It's like a quick and dirty analysis. Okay, so now here's a cool thing. So once you make these two-dimensional maps, you often find beautiful structures like that. You see that cells naturally form clusters. And this is exactly what you would expect from the biology, because any tissue that you pick, either it's like a blood draw, or it's like a piece of lung or heart or anything like that, there are like many, many different cell types in that tissue, right? There's epithelium, there's blood cells, there are immune cells. And each of those cells will naturally, all the cells that are of the same type, they probably express the same genes. So you would expect those cells to sort of group together. And that's exactly what these sort of TSNE or UMAP plots show about our data. We're getting towards the end. So now once you have done your dimensionality reduction, you see these clusters, right? Now you actually want to formalize this. You see them, but you want to actually give a label. You want to say, this is cluster 1, cluster 2, cluster 3, cluster 4. Like, how do you do that? So the way you do that is by taking your gene expression data, looking at it either in the raw gene expression space or in the principal component space, doesn't really matter also that too much. And then we look at the connectedness of cells. So typically what you see, connectedness, I mean, like in terms of like spatial proximity, you see like for any given cell, who are the nearest neighbors of my cell? Yeah. And then you make this graph, a graph of nearest neighbors of every cell, right? And that graph naturally forms communities. What is a community? A community is a group of nodes, in this case, a group of cells that are like very interconnected with one another. They're very close to one another, but they're distant to other communities, right? And they're like very fast algorithms for once you have this graph to basically determine communities. One of them is called Louvain, and the other one is called Leiden. They're both pretty widely used and pretty fast. In fact, I think some of the authors of these methods, they're even the same. And yeah, so that's how you actually identify cell clusters. Finally, once you have your cell clusters, you say, okay, so these are my cells. cluster 1, oh sorry, cluster 2, cluster 0, cluster 1, and now you want to maybe do differential analysis. What I mean by that is that you want to say, like, take cells in cluster 0 and compare the level of expression of every single gene of cells belonging to that cluster with the rest, with the other cells. And that sort of gives you like a score for every gene, kind of like a plot like that. Here you get like different genes, and then here you get like scores. So you see that because like these genes are like very distinctly higher expressed in cluster 0 compared to cluster 1 and cluster 2. And you do this analysis for every cluster, and sort of you get a list of interesting genes. These are genes that sort of mark the identity of these clusters, so to speak. And then what you do is that you do Google search. You search like CD79A, and you see that people say that, oh, CD79A is expressed in B cells. So then from that, you might conclude that maybe this cluster is a B cell cluster. Okay. So obviously, Google search is not the most appropriate way of doing it. You can ask ChatGPT. In fact, it does a really, really good job if you just give it a list of genes and ask ChatGPT to give you what is that cell type, because it has read all the biology literature. So it actually knows cell identity from just a bunch of genes. But we have done something probably fancier than that I'm going to show you in the live session to automate this step of assigning cell types to subclusters. Awesome. The other things that you could do is that you can make beautiful visualizations. You can pick your favorite genes, like NKG7 is like a marker for natural killer cells. And then you see it's highly expressed over cells in this cluster, is not much expressed everywhere else. Cystatin 3, this gene is highly expressed typically in monocytes. So it sort of lights up in monocytes. So you might conclude that, okay, this is like a monocyte and all that. You can also make these other plots. You call it dot plot. It's a very cool plot. On the x-axis, you see genes. On the y-axis, you see your cell clusters. And at any position of this two-dimensional grid, you have a circle. The radius of that circle tells you how many cells in that cluster actually express that gene at all. And the color intensity of that circle tells you what is the magnitude of expression. So you see basically two pieces of information in one plot. So for example, you might say that this PPBP gene is expressed pretty much in every megakaryocyte at very, very high levels. And maybe here you can conclude that, I don't know, like LYZ gene is expressed in monocytes, in CD14 monocytes and FCGR3A monocytes, but it's expressed more here. Like all the cells do express it, but the cells that express the monocytes, the CD14 monocytes actually express it more copies. Awesome. Also to just show you remember I told you Cellbender that it does a pre-processing of your data. It sort of did two things for us. One of them was to identify cell-containing droplets. The other one was to remove ambient RNA contribution from our data. So you can actually see how Cellbender, what it does to a typical sample. So this sample is a PBMC. PBMC is peripheral blood mononuclear cells. So when you're like doing a blood draw, when you centrifuge it, when you spin it, there's this layer here, just between granulocytes and plasma. This layer is a layer that contains many different immune cells. You isolate it, that's your PBMC. And that's the one that you're doing single cell RNA sequencing on. So here's how a couple of different marker genes, their expression is shown in different cell types on the raw data. And if you just take the same raw data and analyze it and sort of pre-process it with Cellbender, here's what you get. We can go back and forth between them. So something very interesting has happened. Like if you look at this like S100A8 or S100A9 for LYZ and cystatin 3, there's a huge difference with and without Cellbender. I can tell you exactly why this is happening. The reason it's happening is that these granulocytes, as the name suggests, they're very eager to degranulate. And when they degranulate, they release a whole bunch of lysosomes, which they contain actually a lot of these LYZ genes and cystatin 3 genes and like S100A genes. So they actually enter our ambient soup that contains the cell. So if you don't correct for them, you actually might erroneously conclude that every one of your cell types actually express those genes, which is a violation of known biology. We know that these genes are actually only expressed in certain immune cell lineages. So yeah, Cellbender actually does something really good to our data. We recommend it. All right, and finally, so that's the end of this presentation. We sort of covered the standard workflow for single-cell RNA-seq analysis. After this, you probably want to do some more advanced and application-specific analyses, like doing trajectory analysis, like if you want to reconstruct trajectory of cell maturation, for example. Covariation analysis, if you have like samples with different labels like sex and disease, or like, I don't know, you want to identify cell types that co-vary with these labels. And then single cell EQTL analysis, which is another very common analysis. So you want to like combine genetic information with gene expression information. Maybe you want to know, having a variant in your genome, is it linked to overexpression or underexpression of some genes? That is called the EQTL analysis. And so these are the other things that you probably want to do, depending on the nature of your research. Here I've just for your reference, I've put like a list of tools for tertiary analysis for each component of tertiary analysis. There's some one-stop shop toolkits. There's some specific tools for like cell calling, batch correction, ambient RNA removal, gene regulation, network inference. So if you ever want to do some of these works, you can always refer to the slide. This is relatively accurate up to 2023. This field evolves very, very quickly. If you look at this slide, probably in 2 years, you'd be laughing. So yeah. All right. So let's not take a break and let's directly jump into the live demo if you don't mind. Yeah. Question? Okay. So this time our live demo is actually doesn't require running that horrendous script. So just log in to your Terra account. And you want to go to analyses, and then you want to select 10 intro single cell RNA-seq tertiary analysis. Okay, while it's loading, I can only try to cover part of this, introductory stuff. So here, what we want to do is that we want to do some, we want to run the tertiary analysis workflow on a very commonly used, commonly studied single cell RNA-seq sample, which is a PBMC, peripheral blood mononuclear cells. It's a very well-known sample because we know exactly what cell types we expect to see in this sample. And what we want to do is that we want to do cell calling, we want to remove ambient RNA, we want to do data pre-processing, dimensional reduction, cell classification, all the stuff that we talked about, we want to quickly go through. Yeah. And in general, there are two major sort of one-stop shop toolkits for interactive analysis of single cell RNA-seq data. One of them is called SCAMPI. is from Fabian Theis's group in Helmholtz Institute. The other one is called Seurat. So depending on whether you are an R person or you're a Python person, you would use either ScamPy or Seurat. ScamPy has pi in it, so it's Python. Seurat, R. Both of them have like very similar functionalities. The implementation is slightly different. Seurat tends to be slower. ScamPy is slightly more scalable. So if you have like a data set containing like 100,000 cells, you can easily analyze it with ScamPy, but with Seurat, you'll probably run into some memory issues. But there are some things in Seurat that are more accurate, like differential gene expression analysis is much more accurate to do in Seurat. So, you know, depending on what you want to do, you may want to use Seurat. It's spelled as like Seurat, S-E-U-R-A-T. I think it's the name of a French artist. Okay. So the kernel you want to use here is just a vanilla Python 3 kernel. You don't need to do any particular setup. If you have like 4 CPUs, you should be good. Yeah. All right. So the very first thing that we want to do is, let me just clear the outputs because I just ran this. You want to install a couple of packages, pandas, numpy, scampy. And then we want to install this specific package. It's called cell annotation service. And that's the thing that I advertised earlier, that it's the work in progress project that we have at the Broad for automating cell type annotation. So hopefully we will see a live demo of that. I've actually never tested that. It's a service, which means that you submit your data to it and it gives you the results back, as opposed to just running it on your VM. So today is actually going to be fun because we're going to stress test it. It might break. I don't know. We'll see. Hopefully it won't. All right. So you should have installed like all these packages. Now we download some data. This is the data that we basically just get directly from 10x Genomics website. It's a public data set that they have put out there for people to play with. It's 10,000 cells, 10-ish thousand cells from PBMC. And if you just list the contents of your data, you should have a bunch of things like that. Now, what you want to do to use Cellbender for like pre-processing your single cell RNA-seq data and doing cell calling is that you want to take your raw feature matrix, that's like the output of Cell Ranger that they have not already done filtering. They have not already like marked specific droplets as being empty. You want to take all of your droplets and then you want to give it to Cellbender. So that takes about 30 minutes to run, and that's already something that I've done. We have a Terra workspace for that. It's very easy. You just put your count matrix there on Terra. There's a workflow, just run it, and then you get the output back. So in the interest of time, I've already done that. So I'm just downloading the results from Cellbender to this notebook. It's yet another one of those sort of count matrices. Okay. So when you run Cellbender on your data, it produces a bunch of output files. It gives you like a nice HTML report and a PDF file. And then, of course, your count matrix, which is denoised, and the droplets have been removed. The empty droplets have been removed from it. All right, so let's just get into the analysis part. We import a bunch of packages. And then we define the location of some type, some cells. This is just a raw count matrix. This is the Cell Ranger filtered count matrix. This is the Cellbender filtered count matrix. All right. So as you can see, all of these files have the extension of H5. This is an HDF5 file that contains the count matrix in it. This is the standard output from Cell Ranger. So Cellbender has the same output format for interoperability. Now let's load all of these count matrices, the raw data, the Cell Ranger filtered data, and the Cellbender filtered matrix. And we'll just look at them. So as you can see, I'm calling like this var names make unique on each of these loaded files. This is to make every gene have a unique name, because whoever made this data set, they, for some of the genes, they use the same name. So it confuses ScamPy. So by calling like make names unique, you just add like an _1, _2 to every gene so that it's unique. All right. So if you wanna, once you load one of these H5 files, you just can print it, and then it tells you at least a dimension of the data. So if I print the raw data, it is telling me that I have like 6,794,880 droplets by 33,538 genes. That's all the droplets that this machine has produced. It doesn't mean that I have 6 million cells. Most of these droplets are empty. Okay, so when we print the count matrix that is filtered by Cell Ranger, we get about 12,000 droplets. This is a little bit closer to what we expected, because this experiment supposedly they loaded 10,000 cells. So you should expect to see about 10,000 cells in the output. And then when you see the output from Cellbender, Cellbender gives you slightly more than Cell Ranger. It gives you something like about 13,000 cells. That's typical. Typically, Cellbender always gives you more cells so that you have the opportunity to go back and filter them if you want. It's a little bit more conservative. It doesn't want to throw anything that might actually look like a cell. And in fact, once you look at the cells that are only unique to Cellbender, that are not in Cell Ranger filtered data, they're actual cells. So it's good that we're keeping. All right. So what we want to do is that you want to, I guess, work with Cell Ranger, or you can do Cellbender. It doesn't really matter. I recommend that you use Cellbender. So just, you just select your ANData Cellbender filtered, and then you just call it AData. One thing that I forgot to tell you, this file format that contains this gene expression matrix is called ANData, annotated data. It's basically just a gene expression matrix with a bunch of annotations on the cells and on the genes. So yeah, that's why we call this AData. So it's that. Okay, so what's the anatomy of one of these ANData files that we've loaded? It's basically something like this. You have your count matrix in the middle. Cells are on the rows, genes or variables are on the columns. Then for every gene, you have like a data frame that includes metadata for every gene. What's the name of that gene? What is the ensemble ID of that gene? And you can put additional information there. What's the length of this gene if you want? Every metadata related to genes should go here. Metadata related to cells would go under observations. Like initially, it's just the barcode of the cell, as we will see in a second. But if you derive certain things from your cells, like if you just count total like RNA in each cell, you probably want to put it in the obs, because it's something that is related to each and like a row of a count matrix. Okay. So if you just do like adata.x, it actually gives you access to this underlying matrix. As you can see, it's a matrix has like about like 13,000 cells by 33,000 genes. Just to get acquainted with this matrix, let's just count the total number of non-zero entries in this matrix. So the way you do that is that of the total entries in this matrix is just number of rows by number of columns. The non-zero, you can just call NNZ, just call NNZ on any sparse matrix. It gives you the number of non-zero elements. And now let's look at the ratio of the two, just to see like how many entries of this count matrix is non-zero. When we do that, it turns out to be about like 5.8%. That's very important. only 6% of this huge matrix has some non-zero entries. That's how sparse single cell RNA-seq data is. And remember I told you that the efficiency of reverse transcriptase is only 10%. That's kind of a reflection of that. Well, part of the reason is also biological, because not every gene is expressed in every cell. These are immune cells. So there's certainly like certain genes that are not expressed. But even immune cells, you would expect to, you know, to have like a lot more genes than just 5%. Okay. If you look at the adata.obs, you look at the sort of obs data frame, you see that at the moment, it only has just an index. And that index is just the barcode of the cell. You can see it's a 16 base pair sequence. That's the cell barcode sequence that we used in the protocol. So that's sort of the unique identifier of our cell. Now you can add stuff to the obs, like I said. You can just add like a total counts column to this obs matrix, and you can sort of set it to take your sparse matrix, you sum it over the column dimension, flatten it, and then you just put it in total counts. And now if you just print obs again, you see that not only you have like an additional column there. And this column like tells you like what is the total number of RNA in each of your cells. So that's a typically like that's a general flow of doing tertiary analysis. You start with like a bare bones count matrix, you just add things to it as you go along. You embellish it with derived quantities. Okay. Now, SC is SCAMPI. So SCAMPI has two types of things. It has like PL, some methods under PL package, and you have some methods under TL package. PL is like plot, TL is like tool. So anytime that's you want to do some processing on your data with SCAMPI, you probably want to call a method under TL. When you want to plot something, you call a method under PL. So, and you can, it has a very good documentation, SCAMPI. You can just look at the definition of like all these commands. So one of those plotting tools is like highest expressed genes. So you can ask to just make a plot of the highly expressed genes in this data set. And here's what you get. MALAT1 is one of those housekeeping genes. It's always highly expressed. MT. These are all mitochondrial genes. It's a housekeeping gene. This is a receptor domain gene, mitochondrial, some housekeeping gene, ribosomal protein, all ribosomal protein. So you can see that some of the our highly expressed genes are actually boring. That's what I said earlier. Now you want to maybe do some filtering. SCAMPI has really good abilities for filtering. Maybe you want to get rid of cells that do not express too many genes. A cell that expresses only like 200 genes, for example, is probably not a nicely observed cell. Maybe we just didn't sequence it well. So you want to look for cells that have good complexity. And you also want to filter genes. If a gene is not expressed in, let's say, 5 cells out of these 10,000 cells, there's not much you can do with that gene because it's so poorly observed that you cannot make any statistical conclusion out of it anyway. So you just filter it. Again, so these numbers are just you make up as you go. So you can return back to your analysis, and then you can include more or you can include less. It's really interactive. Now, what we want to do is that just move along, and then you run this command. Just add another column to your var data frame. Var was basically the annotation that you had over the gene columns. And now what we do is that we just, you want to determine which genes are mitochondrial. So we say that any gene that its name starts with MT, let's just give it a flag that it's a mitochondrial gene. And once you have that flag, then you can ask SCAMPI to, PP is pre-processing, to calculate some QC metrics using those mitochondrial genes as sort of QC variables. Effectively, what we're telling it is that just tell us what is the count of those mitochondrial genes. And once you run those commands, you can see that ScanPy automatically adds a whole bunch of things to our ANData file. So we have total counts that we added, number of unique genes by count, total mitochondrial counts, percent of mitochondrial gene counts, and number of cells that express this gene by at least one copy, whether it's a mitochondrial gene, number of cells that express that gene, and total counts, and I don't know, you get a whole bunch of things. Now you can actually look at the OBS data frame, see that ScanPy added all these derived features. You can look at your var data frame, and you can see that ScanPy has added all these additional gene level metadata. And now you can make a bunch of plots based on these things that we calculated. For example, here we're plotting the total RNA in cells by the percent of mitochondrial genes. So for example, just look at cells from here. These cells express like 40% mitochondrial genes. Like out of all the genes that this cell expresses, 40% is mitochondrial. And that cell has only like a very, very small amount of RNA. So that thing that we call cell is probably just a jumble of mitochondria. We want to remove it. So you probably just want to remove all these cells. And maybe you also want to remove this point and this point and this point because they're kind of like really outliers in this distribution. So that's the QC, that step that I mentioned earlier. So let's just do some of that QC. Get rid of all the cells that express more than 20% mitochondrial genes. And let's get off all the droplets that have like too many genes expressed because they're probably like doublets, they're 2 cells. Now you can see that our ANData should be like a smaller object now. If we started with something like 12,000 cells, we're down to 10,000 cells because we got rid of a whole bunch of cells. We started with like 30,000 genes. We're down to like 19,000 genes because we removed a whole bunch of genes that are not expressed or they're lowly expressed. Now we do the data normalization. It's as simple as calling normalized total on your ANData. And then you call log1p, that's logarithmizes your data. So everything is now in a logarithmic scale. And you call this command highly variable genes. It determines your highly variable genes according to the same criterion that I mentioned earlier. It looks at genes in the middle of the road that have a lot of dispersion between droplets. And it can make this plot of highly variable genes. In particular, here you have like the mean expression. Every point here is a gene. The mean expression of the gene is in a logarithmic scale, is in the x-axis. And the total variance of that gene across different droplets is in the y-axis. So probably these genes that are marked black, these are the ones that we want to keep. Because they're not too lowly expressed, not too highly expressed, and they have a lot of dispersion. Now, we're going to even do like more filtering of this data. Like we want to, for example, subset all of the genes to just highly variable genes. If we do that right away, we just lose our data. So what we do is that we just keep a copy of our data before we filter, before we restrict it only to those highly variable genes. We store it in the raw. So you just do ANData.raw equals ANData. So it basically stores a copy of itself in a specific attribute, which is called raw. And you can always restore your data from that raw attribute if you want. So here's the one that we're doing actual filtering. Remember, at this point, if you just print your ANData, it has about 19,000 genes. But now, if you're slicing it by highly variable attribute, and now we only have like 17,000 genes. Those are the ones that are highly variable. Finally, we scale it. That scale is the one, is what I said, that it's so z-scores it. You subtract the mean expression, you divide by standard deviation, so that all the expression values are sort of order 1, from -1 to 1. All right, dimensionality reduction and clustering, very simple. You just call PCA. It does PCA. And in fact, if you look at your AND data after you call PCA on it, you see that it has under this obs_m. These are matrix observations. It has added something like XPCA. That's the PCA of our X being the underlying matrix. Now you can look at this PCA. It's a two-dimensional matrix. If you look at the shape of this matrix, it's like 10,000 by 50. So we have only kept 50 principal components. You can plot the cells in terms of the two PCs, PC1 and PC2. And you can already see, even in terms of the first two principal components, there's some clustering going on. So even the first two principal components are useful for separating identity of our cells. But we do expect this data set to have a lot more cell types. You know, from this, you can identify maybe like 1, 2, 3, 4, 5, maybe 6 clusters, right? But we expect to have like this, I don't know, 10 different cell types in this data. So you're going to look at different higher dimensional PCs. Maybe there is a specific cell type that is only like distinguished by looking at PC3 and PC4. And that's what I said, that making these two-dimensional plots is just not enough. You need to be able to squish that 50-dimensional space using a nonlinear method to a two-dimensional space. Going back to your question about explained variance in principal component analysis, you can also call this like PCA variance ratio, and you can make this plot. And this plot basically tells you how much variance of your data each principal component explains. You can see that maybe there is a little bit of a knee here around like 30. So 30 should be enough, maybe. Okay. The next step is that, okay, so now that we put our gene expression data in this PCA space, we want to build a neighborhood graph. And that's a graph that tells us that in the principal component space, what are the neighbors of each of our cells? So we basically effectively are under the hood making a graph like that. Every node of that graph is a cell. Every edge is like the distance between one cell to the other cell in the PCA space. You do this by calling I don't want to start. Should be pretty quick to run everything. As I wait for my kernel to run things again, any questions? I know that the pace of this is kind of too fast, but I hope that you will return to this notebook again if you're serious about single cell analysis, and hopefully the second time around and third time around things start to make more sense. I have maybe looked at the same tutorial maybe 20 times up to this point, and I still sort of return back to it. So it's a good resource to have. Okay. Oh, it died again. Does it happen to any of you, or you're good? Let me try with another kernel. That could be an issue. Yeah, maybe 15 is not too, yeah. If it failed again, I'll increase it to 30. Yeah, that it could be a memory issue. Anybody here has like 15 gigabytes of RAM and don't have this issue? Doesn't have this issue? You're good. You're good. You can always have a backup. Anybody here with a laptop that you could just plug in and use their laptop instead? Yeah. Let's see if it failed again. I'll get yours. Yeah. Oh. Yeah, it's dying on my end. It's probably something with my environment. Yeah. That's the good thing about doing things on Terra, because you can always grab someone else's laptop and you're all working on the same thing. So. Okay. Oops. Thank you. Awesome. Thanks. All right, so now we have our okay, so we made our neighborhood graph. If you just print your AND data again, you see that it must, you have like two new things in your AND data. One of them is called distances and connectivities. So connectivities, well, if you just, for example, look at your distances, you see it's a matrix of shape, number of cells by number of cells. So that's the pairwise distance between any cell to any cell. If you look at connectivities, that's a binary matrix. It just tells you like what is the, well, not necessarily binary matrix. It's a different type of distance. It's a topological distance. It's a slightly like more complex notion of distance, but it also ultimately tells you about like how close the cells are to one another. That's sort of the graph that we're talking about. All right, so now you want to embed that graph into 2D. That's the step that graph effectively is a 50-dimensional object. You want to squish it into 2 dimensions. So one algorithm for doing that is called UMAP. So you just call UMAP on your AND data. And then you should get this additional X underscore UMAP in your obs M. And that is a matrix which should have the dimension of number of cells by 2. So every cell is now in a 2-dimensional space. You can plot the cells in UMAP and you get something like that. A lot more clusters. This is kind of like what we expect. Now, every one of these clusters is probably like a different cell type. All right. So now you can highlight your favorite genes on this UMAP, like cystatin 3, NKG7, PPPP, and you can see that these are like lit up on different clusters on your UMAP. And you can like color this either in terms of like the scaled and logarithmized gene expression, or you can do this like in terms of like the raw gene expression. So you have access to all of those things. Now, the next step is to cluster the neighborhood graph so that we can have cell clusters. And when you run this Leiden algorithm, you should get on your obs like a new column called Leiden. And if you just print your obs data frame, you see that now you have like this additional Leiden column that for every cell, it gives you like an integer number, like 8, 8, 8, 8. I don't know. So basically all of these cells belong to the same cluster. 6, 6, which means that these cells belong to cluster number 6. So there should be about, I don't know, like 10 or 20. Well, in fact, or if you just color our UMAP by Leiden, you can see that we have identified about like 20 clusters. So arguably, some of these cells, this is like over clustered, like maybe this is just one big cluster that Leiden has like broke into 2 clusters. So you can change, there's a resolution parameter in your Leiden algorithm. You can mess around with it and it can gives you like fewer clusters or more clusters. So inspect your data. If you think it's over clusters or under clustered, you can just go mess around with the resolution algorithm. Cool. Now, finally, we want to assign cell types to our cell clusters. The standard method, like I said, is to do differential analysis. So you just you call this rank gene groups and use Leiden labels, so that what effectively it does is that it compares every Leiden cluster, every cell cluster effectively with the rest of the cells, and it scores the genes. So, and the result of this analysis is this dictionary, which you can convert into an easily viewable data frame. Just run it. Yeah. So as you can see, for every cluster, like cluster 0 versus rest, cluster 1 versus rest, cluster 2 versus rest, and so on and so forth, every gene is scored. So for example, like cluster 0 expresses S100A8, S100A9, S100A12 very much. I can tell that's a monocyte. Those are monocyte marker genes. Yeah. And that's typically how you do it. You just pick those like top genes, and then you refer to your biology knowledge to see what cell type you're dealing with. Okay. Yeah, and like I said, you can convert that into like a nice table if you want. So for every cluster, you get p-values, you get log fold change, like how much that gene is overexpressed in log space. And yeah, so you get this big table for every cell type and every gene. So now I have actually a, since let's say I'm working a lot with immune cells, I know my marker genes. So I know that these are markers for monocytes, these are marker genes for, PDCs, plasmacytoid dendritic cells, and so on and so forth. And then I can make a dot plot, type of plot that I told you about, based on these marker genes that I provide. And it should nicely give me a concise summary of the expression of those genes that I have specified before in different cell clusters. So indeed, you can see that these groups of genes, they're highly expressed in monocytes, S100A9, S100A8, S100A12, L by Z, CST3, and so on and so forth. Now, if you did this analysis on the raw gene expression data as opposed to Cellbender corrected gene expression data, you would get this. And the difference between this dot plot and the other dot plot is that there are a whole bunch of genes that are expressed at low levels everywhere, but Cellbender sort of very nicely removes those. So like if you didn't process your data with Cellbender, you would, for example, conclude that LYZ is expressed everywhere in all cell types, but it's just expressed more in monocytes. That's wrong. CSA3 is only expressed in monocytes. So that's the type of conclusion that you want to make. And Cellbender sort of helps you make that conclusion. So here's the final step that I don't know, it might work, it might break. So this is the experimental tool that we've been working on for automatically, this is not a prank anymore, for automatically determining cell types. So what we're doing here is that you're just giving a copy of our raw gene expression and data matrix to this service, and this automatically returns cell type annotation back to us. So let's see if it works. So it's like submitting chunks of cells in thousands to this service. It's called Cellarium Cas, Cell Annotation Service. And you can just feel free to run along. Yeah, so what is happening here is that we have like made a large list of reference cells, 4 million cells, and the plan is that we're going to extend them to 40 million cells soon. And so what is happening here is that every cell that you give to the service will be compared to each of those 40 million cells, and then the annotations over those reference cells will be summarized, and a consensus of that will be returned to us. Yeah, so It's all human. So what you get here is the consensus of the biology community. So instead of doing a Google search yourself, you're doing a search over this data set. Okay, so it definitely worked for me. It finished under a minute. Can you show a raise of hands if it also worked for you? Yeah, great. So now I can just show those annotations directly on our UMAP plot. So this service adds another label to your ANDATA file. It's called CAS per cluster cell type labels, cell type. And then every one of your Leiden clusters will now have a actual cell type label. So for example, you have like CD4 positive helper T cell, CD8 positive alpha beta memory T cell. So for example, these are like two different types of monocytes. I would say these are classical monocytes, and these are just monocytes. These are naive B cells. These are like memory B cells. So that's yeah, we're still working on this. It works kind of well for immune cells because we have a lot of immune cells in our atlas. Hopefully by the end of this quarter, we'll add a whole bunch of cells from all other like tissues, and then it would be something that we could use all the time for cell type annotation. But that's the end of the live demo. I hope that you got something good out of it. It was not like a GATK thing, a totally different type of workshop module, but it's an interesting topic. I personally love single cell RNA-seq analysis. That's what I do. All right, thank you. Please, yeah. Yeah, so it's a thing. It's called like it's a microservice. It's a serverless hosted application. So the more you submit to it, it sort of self-replicates to be able to manage the load of requests that you give to it. So the good thing about this is that it's relatively fast compared to something like Cromwell, because Cromwell needs to sort of spin up a new VM every time. Spinning up a VM in the way that we're doing it here is much faster. So it's kind of like more responsive, but at the same time, you're not paying for a server that is always running if nobody's using it. It's a cloud native application type thing. Cool. Any other questions?
Okay, everybody. Well, we're at the end. So first off, I just want to say thank you all for pushing through and for attending and paying attention and asking great questions. It's really been great for us to have this experience with you. And also, before we wrap up, I wanted to make sure that before you go, you remember that to say this link, especially if you have it on your desktop computers that are here. If you save this link someplace, you will have access to all of our presentation materials, along with the agenda and the Google Docs that we have here for you. It has a lot of links in there that are handy that you can use, along with all of the workspace links that we've been using throughout this workshop. There will also be links in, there should be a link already to the 3 previous days of this workshop, the recordings of those. Obviously the 4th day will be available too. We're still recording that now, so whenever that's done, that will be available for you to see. And obviously I have been, every time I've been up here and the other instructors, we've been mentioning that there are articles and documentation available at the GATK website. GATK is a package of software tools, but I think one of the strongest features of GATK is that there's just so much available information about it and documentation that's being written about it. So I just wanted to go very briefly to show you, I guess to prove to you that this is the case. So here we are on the GATK website. And so I briefly covered on the very first day the type of information that was here, but I just want to highlight that if we go to the Getting Started section right here, we can click on this. And there are lots of documents here, of course, documents that you can search through, but I specifically want to call out the best practices workflows set of documents. If you click on this, it will take you to our best practice workflows for mitochondrial short variant discovery, somatic short variant discovery, germline short variant discovery, and a series of others. All the types of information that we've been covering throughout this workshop is available on the website. And so we've been giving that information to you, but let's say you that you're not actually going to use the GATK tools immediately after this workshop, and some of the information is hazy when you actually start using the tools for yourself. Just remember that the information is available online, and that should serve as a very good refresher for you when you actually are starting to use this for yourself. Another one of the features that I really need to highlight, we've briefly touched on it before, but on the website, we do have a GATK forum, and the forum is a very good resource for you to use, not only because there are questions that other people like you have been asking there and have been answered by the developers, but if you do find yourself encountering a bug or not knowing how to solve a particular issue that you are finding yourself encountering, you can, of course, ask us on the forums, and we will try to get out to you as fast or as quickly as possible. So it's a really important resource. And the last thing I want to highlight is the tool index. If you visit the tool index, you will have access to the tool information for each of the different versions of GATK. If you have an updated version of GATK to the most recent version, which is 4.4, then you would just go to the current version. But I know that depending on what download you have on your, let's say on your local server, it might be a little bit older. You can always access the previous versions. But if you are using the tool for the first time, I encourage you to find that tool in our tool documentation index. For example, let's say you're looking for haplotype caller. Just find it, click on it, and then you will not only get an overview of what the tool does, how to use it, you'll also get information about how to actually call a haplotype caller in this particular case. And if you scroll down, you can get information about what the required arguments are in order to use it, along with a long list of all the optional arguments that you can use to tweak this tool for your particular use cases. So all the information is there. And of course, if you we have been trying to answer as many of the questions that you've been giving to us in the Q&A doc that was made available to you at the start of the workshop. But if you find yourself with some additional questions after the fact, then feel free to email us or reach out to us in some other way, and we'll try to answer your questions. So, returning on, we have tried to give you a lot of information that is available to us in this amount of time, but now it's your opportunity to give us information. So I'd like you all to go to this website. This is our survey that we host at the Broad Institute. This link should also be available in the agenda on your computers, and it should only take about two minutes to complete, but this will give us feedback on the workshop. What did we do well? What did we not do well? What did you like? What did you not like? And what would you like to see in the future? And all of this information is really important to us. We actually do read all of your comments and suggestions, and we take it to heart. So please take the next 2 minutes to go to the site and fill out the survey.
You can do it on the computers on the desks, and if you're doing it on the computers on the desks, there's a shortcut on the browser as well. You can click the shortcut on top of the toolbar on the browser.
Thank you.
Or it's in the Google Doc.
I still hear typing, so I don't want to stop you. If you haven't completed the survey yet, please continue. But I want to move on very quickly and also say that there's another survey for you to fill out. I'm sorry, but that you've got 2 of them. But this one is going directly to the organizers, going to Hugo and the rest of your team. So this will go directly to people here at the University of Cambridge, and I'm sure that they would appreciate your feedback, not directly on us as the instructors, but more on the content of the workshop and how it well, how it does what, how it affects you if you want more of this content in the future. So please go to this site. The link should also be in the agenda, and it should be bookmarked on your local machines as well. So if you're already done with the previous survey, if you could go ahead and do this one, that would be fantastic. Thank you. Again, you don't need to stop immediately. There is going to be time to continue before we leave, but I just wanted to again reiterate that we really do thank you for coming to the workshop and being engaged. That's really the most important thing for us. And finally, before we wrap up, I want to give Hugo an opportunity to give it any additional information that you wanted before we leave.
So just a couple of things to wrap up. The recordings to the sessions are on the Google Doc. You probably already picked up on some of the ones from the last few days. We'll add the day 4 there at the end of today. The links will be available for a month. The videos will be available for at least a month. You can download them if you want to keep a record. Just all we ask is that you don't publish them anywhere. They're for you. They're not for public use, as it were. One thing I wanted to mention is I've got some links there for other courses you might be interested in signing up for the mailing list if you want. But one thing I recommend is if you're at Cambridge, to join our University of Cambridge Bioinformatics Slack channel or Slack workspace. It's a very friendly Slack workspace. There's currently 200 people on it, so it's kind of growing all the time. And it's a great way to ask for help with any bioinformatics questions you have. There's no silly questions or anything like that. The idea is really to help people that need help. And sometimes those people are people that might have experience as well or no experience at all. And it is active. So this is one of the channels, is the HPC channel for people that use the HPC at Cambridge and want help. And this one was, on Wednesday, Mariana asked a question at 12:41. And then at 12:47, Andres was giving her a reply. And then they exchanged 16 messages with each other to solve the problem. Yesterday, James asked a question. And instead of paying attention to these guys, I 10 minutes later answered the question that he was asking, and we exchanged a few messages to solve the issue. So there's a few of us there that are quite active. So anyone's welcome to ask questions. At the moment, we don't have, there's also some social stuff. So this week they were actually going out for lunch. Oh, and by they, I mean anyone can go. And apparently there's 2 Yori restaurants in Cambridge, and they went to different ones. So that's funny. So we don't have a ton of channels because we want to avoid having too many channels. But if it starts to emerge that it would be useful to have more specific channels about topics, for example, you've been on this course and you might feel like it would be nice to have a genomics focused channel, we could always do that. I might put out a call on the general channel to see how many people would like to have dedicated channels, say for transcriptomics, genomics, something like that. But in any case, if you post a question on the general channel, that's fine. Don't feel inhibited about asking questions there. So yeah, so do join that Slack workspace. In particular, if you're at Cambridge, it actually doesn't, if you're not at Cambridge, you can also join. It doesn't stop you from joining. But I guess it's more relevant for us here. I'm also, I've been playing around a little bit with having Cromwell run on our HPC. And I think I've managed to run a Hello World whittle, submitting a job to Slurm. I'm gonna like play around with it a bit more, and then I'm gonna try and document that somewhere. Possibly in our, or initially, I'll probably do it on our HPC course materials. We've got a section on Cambridge HPC stuff. I might put it here initially, but I might share this on the Slack workspace when I document it somewhere. So that's something else to just kind of for you to keep an eye out for, and another motivation to join the Slack workspace, I guess. Apart from that, any questions before we finish? Okay, so let's give a big thanks to Derek, John, James, and Mehrtash, who have done a great job. Thanks a lot for coming, guys. It was really, really great. I really enjoyed it. I hope everyone else enjoyed it. And there was a little bit for everyone, I think, in this workshop. If you'd like to, we could also do like a group photo outside for those of you that want to be in the photo. And then we can share it maybe on your Google Drive. That might be a good place to share the photo. Yeah, we might put it on the Google Drive of the course. All right, otherwise we'll stay here for a while. So if you want to grab any of the instructors, feel free to grab them for like specific questions that you may have for your data. And thanks a lot, and maybe I'll see you around.